apply this to my patient care. Of course, we have functional imaging, and these are some examples from one of my patients where I apply fMRI, DTI, transcranial magnetic stimulation, and also direct electrical stimulation that can be pinned with numbers of positive stimulation sites. And although we use this in practice, I still maintain most of my approaches to direct electrical stimulation. One point to notice is that despite the vast amount of information, still there is no consensus with regards to the anatomy and the function of the white matter tracts. And one example is the arcuate fasciculus. This is from the paper of Anthony Dick, which again is in the faculty, where it shows that the arcuate fasciculus, as originally depicted, is the same paradigm we use 150 years later in medical schools and residency programs. Other groups, for example, Catani, have the three-segment model. Glasser has the two-segment model, and a Japanese group has an indirect connection of the Broca's and Wernicke's areas. Therefore, this is possibly the most well-studied language tract, the arcuate fasciculus, for more than a century and a half, and still there is no consensus as to how we can define it and how we can test its function. The cortex is very important for us, and although we all accept a connectivity model, we accept a network model, we cannot avoid the fact that we have to go through locality of brain, and even if we disrupt the network, it has to be a local disruption of the network. So the cortical parcels were defined by a huge number of scientists and clinicians, including Brodmann, who's been working with Oscar and Cecilia Vogt in Berlin in the beginning of the century. And you can see this is the original monograph of Brodmann from 1909. And this is one of his original diagrams where you can see the area 4, the motor area, and the area 3, the sensory area. You see how the cellular profile changes from one parcel to the next. And although the science has evolved, even with the evolution of functional techniques, the work of Brodmann is quoted more and more. And although it has been surpassed in many aspects, still, I believe it is a staggering amount of intellectual contribution. He initially parcellated the brain to 43 parcels, although the number is 52. There have been a few gaps, which are more common in animals than man. Then the votes said that actually Brodmann got it wrong. It's not 52, it's 180. Then we had the von Economo and Koskinen in the 1920s, where they said the parcels were 107. And more recently, the articles from Van Essen, which again we have the pleasure to welcome tomorrow, that we have 210. Of course, many of these models have not been validated in clinical practice. So when we look at the brain, we look at the sulci, and these are some of the common landmarks. The anterior horizontal and anterior ascending branch define the pars triangularis. And of course, we have the subcentral areas, posterior ascending, posterior descending parts. In my institution, we have established a group where we try to analyze the location, the function, and the testing. And I have to say that this is not some academic exercise, but for our patients who needs to have a tumor removed while preserving the function is a very, very important issue. So some of the paradigms you have developed, and of course you continue to get input from other colleagues, is that the pars triangularis between the anterior horizontal and anterior ascending branch is mainly involved in word retrieval, and we test with semantic, syntax, grammar, and object naming. And I hope that tomorrow Greg Hickok will describe to us the neural networks of syntax and grammar. The pars opercularis, which is posterior to the anterior ascending branch, is mainly related to word generation. And we test this with the ability to generate words. The pars orbitalis, it's a lot less well-defined. And we feel that possibly has both phonological and semantic functions. The posterior part of the pars triangularis and the pars opercularis were considered as the traditional Broca area, but we know that these concepts are very fluid and vague and might even not exist, as we oftentimes remove what is an anatomically defined Broca area for tumors, and patients don't have any deficits. Other areas, other cortical parcels, are a lot less well to define, and this includes the premotor areas, both ventral and dorsal. One of my MSc students did her entire thesis on the anatomy of these parcels. And after scanning the entire literature for about a year, it is obvious that there's no clearly defined cortical parcels. The dorsal premotor is supposed to suppress motor activity, also has to do with temporal coding, response selection, etc. The ventral premotor area, which is occupying a slender area at the inferior part of the precentral gyrus and also extends to the pars opercularis, can be involved in articulation. And we have patients when they have a small but very critical stroke in this area, they cannot articulate. The Brodmann area has been quite difficult when you look at traditional diagrams because there are these small squares and dots. It's difficult to understand. So we just use this color map. We try to transform it in a way that makes more sense to us. And this is how it looks like on the left. The medial part of the frontal lobe is a lot less well described. And of course, here we have the parcellation of the cingulum, which is very important as critical segment of the limbic system. For example, last Thursday, we saw a young patient with a lesion, a ganglioglioma, in this very critical part of the brain, including the para-olfactory and subcallosal gyri. But these are very difficult to identify on scans. Professor Ribas very eloquently described the parcels of the parietal lobe. And you can see here the Brodmann area 40 and 39, supramarginal and angular gyri, respectively. But as you realize, there is this gyral continuum described by many epilepsy surgeons in particular. And therefore, the critical point for us when we test is where one parcel stops and when the other parcel starts. Brodmann said is the cellular profile. Vogt said is the myelin profile. We have neurochemical profile. We have different other profiles. But for us, we use direct electrical stimulation. How would you test the supramarginal gyrus? Again, our group, based on the work of Kathy Price and data from stroke, we apply a segmentation of the supramarginal gyrus into 4 quadrants: anterior ventral, posterior ventral, anterior dorsal, and posterior dorsal. And you see that most of these relate to the motor control of speech, particularly the anterior ventral, repetition of the pseudo words that can be useful in assessing the auditory components of the short-term memory. And the posterior dorsal is related to the word production. The angular gyrus, again, has multiple functions, and there have been segmentation models that we're still trying to define. And these are involved in remembering past experiences or future experiences, but these are components you cannot test during surgery. And of course, more easy to test, more practical to test is sentence reading and arithmetic fact retrieval, which can test the short-term memory. If you say, what is 15 minus 3? You have to hold these numbers in your memory for a second or two. So this is a very reliable test of a short-term memory, while multiplication, for example, would not be useful in this regard. The corpus callosum, although it's the largest white matter fiber tract on the brain, is not that well studied. Of course, we know the split brain syndrome by Roger Perry, who got the Nobel Prize, and his apprentice, Michael Katzanika. But it's very difficult to test this intraoperatively, and as a matter of fact, we have no paradigms. You're all familiar with the Whittleson scheme, where he divides the corpus callosum into thirds and fourths and geometrical shapes. But for those who study anatomy know that the brain doesn't operate on strict lines and dichotomies. I think the model of Hoffel and Fram from 2006, it's a lot more relevant, where the gene is composed mainly from prefrontal, premotor, motor sensory, and the splenium, we have the parietal, temporal, and occipital areas. And for us, it's very important because if a tumor is affected by the corpus callosum, we tend to resect as safely as possible. And you know that the anterior half is a lot more forgiving than the posterior half and the splenium. So we do need to develop these paradigms. There are many ways to segment the superior longitudinal fasciculus. With some of these, I do not agree, but I present them out of completeness. And of course, I would like to emphasize that the arcuate fasciculus is not part of the SLF. the SLF is a parieto-frontal tract. The arcuate fasciculus is a temporo-frontal tract. So based on some of my initial dissections that were done at the Department of Anatomy, and I would like to thank Professor Carey for his license to do my dissections there. You can see that the superior longitudinal fasciculus, number 3, is the shortest, is the most superficial one, and connects the supramarginal gyrus with the pars opercularis. The main function of the SLF3 is phonological processing. And this is important because, as we saw earlier, the pars opercularis is linked to phonological processing, and therefore all these functions are related. And most of the times we do infer the function based on the areas of terminations and origin. The SLF2 is a bit longer, is a bit deeper, and is a bit superior than the SLF1, and connects the angular gyrus with the middle part of the middle frontal gyrus, and is mainly related to the visuospatial attention. If you damage this, you have neglect. The way we test this in surgery is through line bisection task. This patient has gave permission for the video, but still I wanted to block the face. So you can see that they mark the middle of a series of lines. And while we stimulate, if the visuospatial attention is affected, the patient will mark the 1/4 of the line, thereby ignoring 50% of the line. The arcuate fasciculus, as I previously explained, has multiple models, but I personally prefer the anatomical segmentation into the ventral and dorsal arcuate fasciculus. The ventral one originates from superior and middle temporal gyri, makes a turn in the supramarginal gyrus, and terminates to the inferior frontal gyrus. Its main function is phonological. And again, you can see the same thing, the same theme. You can see supramarginal gyrus. This is the area where we have the SLF3, and you see the inferior parts of the frontal gyrus, which is all phonological. The dorsal arcuate fasciculus is taking the outside lane, starts from the middle and inferior temporal gyri, goes through the angular gyrus, which is the navigation center of the brain, and terminates to the middle part of the middle frontal gyrus, and therefore is more important for semantic and lexical function in the dominant side. This is a cross-section where it shows that the dorsal and ventral arcuate fasciculus, or any arcuate fasciculus for that matter, are being sandwiched between the SLF3, which is lateral and superficial, and SLF2, which is medial and deeper. The insula is very critical for us because it's an area of many important structures. And you can see that the central sulcus of the insula separates the short gyri, 3 short gyri, 2 long gyri, transverse and occipital and accessory gyri. And the central sulcus is in line with the line of the central sulcus of the cerebrum. The posterior part of the short gyrus is what is called a Dronke's territory. And I used to give this talk in various conferences and talk about Dronke's territory. And I'm thrilled that we do have Nina Dronke herself to be talking about this particular region later on. So I won't go into the topographical anatomy of the superior limiting sulci, Lyman point, etc. We have recently published our technique on insular gliomas, which is quite challenging, in a book by Alfredo Quinones from the Mayo Clinic. Once you remove the insular cortex, then we go to the extreme capsule, which is short association fibers between the adjacent operculi and the extreme and the cortex of the insula. And you can see here the projection of the putamen and the thalamus. The Dronke area, and I'm sure Nina will have a lot more to say about this, is mainly involved in motor planning of speech and polysyllabic words, for example, spaghetti, gravity. Patients who have deficits in this area tend to have an effortful try and error speech. They eventually get it right after many unsuccessful efforts. The ventral anterior area of the insula is related to empathy and emotional behavior. And I did have a young mother not long ago who said to me, I just feel nothing, I have no empathy for anything, which was quite spectacular. Once you remove the extreme capsule, then we see the claustrum. And the claustrum is a very slim area and is related to two important structures, the external capsule. The external capsule is composed of two parts, and please stay with me for a second. I know it gets a little bit complex, but it's not that complex. So the ventral part has association fibers that go from one end to the other, while the dorsal part has projection fibers that go from top to bottom. And you can clearly see that the claustrum has a dorsal part, which is a little bit bigger, but still difficult to see, and a ventral part, which are even more difficult to see in the sections because there's some small fragmented nuclei embedded into the uncinate fasciculus and the eye of the inferior front occipital fasciculus. So putting everything together, you can see that the extreme capsule is composed of the ventral part, which is the IFOF, and the uncinate fasciculus that becomes very narrow in this area above the limb insula, which surrounds the middle cerebral artery. And then we have the dorsal claustrocortical fibers that are particularly condensed in the parietal area and the SMA area. And these are very important in the multimodal sensory innervation. Crick and Watson, who did the double helix of the DNA at the later part of his career, was involved in neurobiology. And he wrote a paper with Christoph Hoch, which again will be with us tomorrow, where he said that the dorsal claustrocortical system might be the seat of consciousness because it can integrate so many different areas. The globus pallidus is deeper, is a bit harder and a bit smaller. And clearly you can see the internal capsule just medially to the globus pallidus. So the IFOF traditionally has been implicated in semantic processing, although the scientists, including Catani, who doesn't believe that it is involved in language, but rather in sensory isolation, something like a reticular formation for the cerebrum. But we do see semantic errors when we stimulate it, and we do see semantic paraphasias when we damage it. So in our mind is very critical for the semantic speech. And tomorrow, Gregory Hickok will discuss the dual stream model, and I'm sure that they will have the opportunity to discuss this further. You can see the uncinate fasciculus, how this shows it, but both of them are become joined here, just above the limb insula. The uncinate fasciculus is important for face recognition and place recognition, and we test this clinically during stimulation with famous faces and famous places. This is some of the work we've done to identify the Brodmann areas involved in the IFOF. And again, you can see this is the limb insula, this is the pars triangularis that Professor Ribas talked about, and this is some of our dissections that demonstrate these principles. Similarly, the uncinate fasciculus. The ILF you can identify in surgery at the inferior temporal gyrus. And again, this is the Brodmann areas that it connects. This is the Latinis model of 3 segments of the ILF with the dorsolateral, the fusiform, and the lingual parts. And of course, it's very important for visual agnosia, for reading, for naming, and interpretation of emotions. But during testing in awake patients with brain tumors, we use mainly reading. The MLF connects the temporal pole with the supramarginal, with the angular gyrus, and is important for visuospatial attention and placement of sounds in space, although it has been implicated in semantic functions. Closing, the corticospinal tract, of course, has only a very small components. The internal capsule has a very small component, which is the corticospinal tract. And these are some of the other fibers that are contained within the internal capsule. Now, as a surgeon trying to understand the anatomy of these fiber tracts, you will come across a number of terms which are below the level of the anterior commissure, including the anterior perforating substance, the ansel lenticularis, the medullary lamina and reticular nucleus. And of course, additional nuclei include the nucleus accumbens and the basal nucleus omeianate, part of the limbic system, which is very important in this inferior to anterior commissure area include the diagonal band of Broca, the septal nuclei, and a number of other structures which are very important when you try to put everything together, aiming to have a holistic image, a holistic concept of the brain and the fiber tracts. So that was a super fast review of key tracts and the way we test them in awake patients. And I would like to thank very much my team for the help and support. And if you want to ask any questions, please feel free to reach out. That's my email. And as I said, we'll have the opportunity to discuss this further during the discussion area. I'm conscious that we already started being a little bit behind schedule, but we'll try and catch up. So moving on, I would like to introduce our next speaker, Juan Carlos Fernandez Miranda, who's a professor of surgical science at Stanford. He directed a virtual laboratory. Some of his anatomical papers are absolutely legendary, and I've been studying them over the last several years. And this is why I also invited him to talk to us today. One of the particular reasons is that Juan Carlos applies both anatomy and tractography, and I felt that he would be an ideal person to somehow compare and contrast these two modalities. So Juan Carlos, thank you so much for being with us today.
George, thanks so much for the invitation, and congratulations for putting together such a phenomenal symposium. It really is an honor for me to be here, and especially sharing the screen with so many of giants in the field that I've learned so much over the years. Follow the contributions and their papers that influence the field so much. So what George asked me is to talk about some of the fiber tracks, especially related with language. I want to start by introducing the members of my team in the lab, because a lot of the work you're going to see is being done with them and by them. My journey in this field started when I was a very young resident, and I learned from these giants in neurosurgery that the most important knowledge we need to acquire as neurosurgeons in training is microsurgical anatomy. And for that, you're required to dedicate full time in the laboratory. This is something that for those out not in the field of neurosurgery is surprising, but it's not standard. Most neurosurgeons do not spend time in the laboratory training to learn anatomy and surgical techniques, but the giants recommended it. So that really influenced me. And when I was a young resident, I spent any time I could, any spare time, to go into the laboratory. I was trained at that point in Madrid, Spain, and I would try to find old brain cerebellums and go under the microscope and try to dissect and follow the teachings of Professor Roton in the field of microsurgical anatomy. Right at that time is when Dr. Ture, along with Professor Jasenfield, published this paper on fiber dissection technique. And this was really life-changing for me because it was a technique to study the brain in a detail that I didn't think it was available before. I was a bit frustrated, honestly, as a neurosurgery resident, excited to do brain surgery. And at some point, you see the brain surgery, it all looks, just going to the brain and all the white matter looks the same, and you cannot tell what is what. But with this technique, you start understanding the fiber tracks within the brain. And this technique, as George explained very well, you frozen the brains, and then you just use wooden spatulas to dissect, or some other microsurgical tools under the microscope. And then these are some of the first dissections I did at that point, 2002. And at the same time, DTI was studying, and I was fascinated that I could not only then dissect the fiber tracks on brains, but I could also actually identify fiber tracks with imaging in patients that I was going to be involved in operating. So that was a fascinating time. I was extremely lucky to have the opportunity to join Professor Roton then after a few years in Gainesville, Florida, to continue learning and investigating anatomy. And I spent 2 years in the laboratory there, full time, after I finished my residency, investigating and training in microsurgical anatomy, and a lot of the time I spent on doing fiber dissections. And what we try to do is correlate those fiber dissections with fiber tractography. This is the first paper we published together, and I'm happy that yours mentioned it, because it was a coincidence. I published on this anatomy of the claustrocortical fibers because I thought it was not well understood. It was for me interesting to study. And at the same time, in a separate way, Professor Koch, along with Francis Crick, they published this paper saying that the claustrum perhaps could be the site of consciousness. I'm not sure I endorse that, but it definitely was an interesting coincidence. And the claustrum does have a lot of cortical connections. But over than that, we actually worked really hard in putting together this article that became a classic, because we studied basically all fiber tracks with dissection, and we correlated with fiber tracking. And I think it was the first, or one of the first comprehensive papers on all the white matter tracks, and actually comparing both techniques to see what are the benefits, the limitations of each technique, and how they complement each other. Then I later moved into the University of Pittsburgh, where I spent a decade, 10 years. And I was extremely fortunate to establish my laboratory there and have tremendous partners, among them, you see Sudhir and also Frank Ye, who you're gonna hear from more later. But we together founded this lab and continue investigating fiber tracks, especially from a structural point of view. And Frank Ye is a phenomenal researcher that helped me understand the differences between the different reconstruction methods, and went from DTI to high definition fiber tracking, a technique that allows us to really isolate the fiber tracks with great accuracy. For example, this is the central semi-valley fiber tracking, which is an area where there are multiple fiber crossings that the technique can actually now solve. And you can compare DTI with the HDFT, or high definition fiber tracking. And this was groundbreaking for me, because DTI was not that accurate, still is not that accurate, but with more advanced diffusion imaging techniques, you get great accuracy. And we also focus on validating this imaging technique and applying into our surgical patients. So validation of the technique, you can see here, for example, how you can actually really replicate nicely all the sulci and the gyri just using diffusion information and reconstruction of fiber tracks. And again, reconstruction along areas of multiple crossing. Now it became the next generation, and this is work from Max in my laboratory. And you can you see the beauty of the dissections and how people like Max have taken this field of micro dissection to the next level. And we can really study in great detail the dissection, the fibers and its connections within the brain using micro dissection. Of course, this is not a extremely accurate method, because there is always error in your dissection, possibility of error, but at the same time, does not allow you to investigate multiple fiber tracks, because you need to destroy one as you get deeper and deeper. But you can see the accuracy of these dissections and the beauty of them. And the field of fiber tracking has really evolved into very accurate reconstruction of fiber tracks. This is work by Sandeep in my lab. We've worked together for several years. And I like to, how both art and science form a confluence of what our field is. Neurosurgery is the art of micro dissection, and at the same time, the science of connectomics coming together. And I believe we need both as neurosurgeons. You can see how beautiful these dissections can become when you are an expert in doing them. And again, I believe both fields, anatomical fiber microdissection and computer-based fiber dissection, are key to evolve the field and our knowledge. Over the, about almost a 20-year career of this, have been my lines of research, and I'm going to focus on the first two, and maybe in the next talk we'll focus on the other two lines of research. And I was very happy listening George and about arcuate fasciculus and SLF. One of the things that really bothered me a bit over the years is the conflict with the terminology of different fiber tracks. And I think as an anatomist and surgeon, it's important that there is an agreement on the nomenclature of fiber tracks, and we need to all make an effort for this. As George very well said, the arcuate fasciculus, its own name says is an arch track. That means that it has like a C shape that goes from frontal to temporal. SLF, however, is a longitudinal track, meaning it goes from anterior to posterior or vice versa, and is mostly a frontoparietal track. And that's a very important difference. Those tracks should not be studied together or should not be put together. They are different entities with, we believe, different function. You can see here some of these dissections we did, and then this is you can see how we're dissecting fibers of the SLF first, longitudinal fibers, and then we see the arch-like fibers of the arcuate fasciculus a bit deeper. Over the years, I was very interested in studying fiber tracks one by one. The same as we study anatomy for example, the skull base, we study different areas or different vessels. I wanted to study different tracks and the details of each of them. This is, for example, studying the arcuate fasciculus, and you can see the left and the right side are, very different, not only on the volume or size, but on the connectivity pattern that the left hemisphere has. And this is based on a template of over 500 subjects from the Human Connectome project. And we can study the different termination of the arcuate fasciculus. We can, for example, segment this in 4 different areas based on the frontal areas of termination. You can see that the organization of the different termination points in the temporal region. And this is the beauty of this fiber tracking. And we've correlated with anatomy. And when we published this paper, I really endorse this view of the ventral and dorsal arcuate fasciculus with the concept of an inner ventral pathway that is mostly phonological and an outer or dorsal pathway that is more robust, and it has to do with semantic system. And this was initially described by Glasser and Rilling, and not many other researchers support this view. And I do, and I think this is, in my opinion, the most accurate, that better represents the arcuate fasciculus anatomy. Then we have the superior longitudinal fasciculus, the SLF. And again, this is a longitudinal track by definition. So it doesn't connect with the temporal region. It goes to the parietal area. And the parietal area, we have both the supramarginal and angular gyri. And we segmented this in 2 tracks, again, in ventral and dorsal SLF. And we saw some variations and perhaps some dominancy of the dorsal one on the right hemisphere, because it seems to be related to visospatial function, which might be more dominant for the right hemisphere. And you can see here the maps of connectivity with the bellow for them. And another problem in our field of connectomies is that a lot of the nomenclature is coming from the animal studies. And the monkey brain and the human brain, of course, are quite different. And the SLF in the monkey brain was described as having 3 different segments. However, in my opinion, this does not apply and should not apply to human brain anatomy. I think we should abandon, in my opinion, SLF 1, 2, and 3, and we should use ventral and dorsal SLF. I think SLF 1, for example, is actually a misnomer, because this track, it belongs to the cingulate fiber system. If you really superimpose the SLF 1 with the cingulate fiber track, is adjacent to it or just above it. And most importantly, is a track that is medial to the corona radiata. So it doesn't make sense to me to group 3 different subsegments that are, one of them is in the opposite side of the hemisphere, completely separate, not medial, but not lateral, but medial. So I do not endorse this SLF1 classification personally, and I think we should correct it. Another interesting track was the middle that we work and publish on, was the middle longitudinal fasciculus. And this track for many years was not well described, or was not even described, because previous DTI studies showed that the arcuate fasciculus actually was forming almost like a C shape. And this C shape was a really false continuation between fibers of the arcuate with fibers of the middle longitudinal fasciculus that are located just underneath. Now, with more advanced fiber techniques, fiber dissection and tracking techniques, we can isolate both fiber tracks much more accurately. And we can see that the middle longitudinal fasciculus goes underneath the arcuate. And because it goes underneath the arcuate, you can follow it, and its terminations are not really, in my opinion, in the angular gyrus. That's not what we found, although papers still describe it that way. The termination of the middle longitudinal fasciculus are more in the intraparietal sulcus and in the superior parietal lobule. And that is important because the function of the superior parietal lobule and inferior parietal lobule, where the angular gyrus is, are very different. We propose that this fiber tract is related to audiospatial localization. It's like the where pathway of the auditory system. And therefore, it has something to do with language, but is more related to a location of sounds in a space. Another important tract that was described over the years, and it's exciting that these things were happening as I was growing as a surgeon and as a researcher, is the frontal ascent tract. Interesting tract that connects the SMA area, supplementary or pre-supplementary motor area, to the ventral premotor cortex for the most part. And we believe is related to speech initiation, and it can cause slattering and decrease verbal fluency. And actually, we've mapped this in surgery, and it's fascinating seeing how patients struggle to initiate a speech when you stimulate into these areas. But at the same time, we have the frontal ascent tract, and the frontal ascent means oblique. And again, the fiber tracts are described based on their orientation, for the most part, longitudinal, arcuate, ascent means oblique. So we have this other, what I like to call the temporoparietal ascent tract. And is this connection here from supramarginal to angular in the temporoparietal junction. And this is a very important tract, but it's being called multiple ways. Vertical arcuate fasciculus, which I think is a total misnomer, because how can you be a vertical tract if you're an arch-like tract? So another name that I think should be abandoned and not called posterior. Posterior A arcuate fasciculus makes no sense to me, but it has been also been called posterior arcuate fasciculus. But this really is like a vertical oblique tract. And I would propose calling it a parietal or temporoparietal ascent tract. And we'll see later the potential importance of this tract in a case we did with intraoperative mapping. But we also studied this tract, and we saw that it has two components, ventral and dorsal. We did not find a laterality for this particular tract. We do believe it relates with language in the dominant hemisphere, and integration of multiple modalities also is this temporoparietal junctional area is so important for integration of multiple modalities of also. And there is, in addition, a vertical occipital fascicle or occipital ascent tract, if you want to call it too, that is located more posterior. And here is an overview of many of these tracts all together. SLF, arcuate fasciculus, middle longitudinal, temporoparietal ascent tract, and how they all come together. And of course, it's sometimes not easy to differentiate them, especially during surgery when we're doing mapping and tumor resections. It's so difficult to know which tract are you really stimulating. And that's one of the issues we have in our field, and we'll describe this a bit more in detail later. But how do we know which tract are we stimulating, and therefore how we can assign function to a specific tract? And I think that's still a lot of work to be done in this area. And again, other composition of multiple fiber tracts, showing, for example, the middle longitudinal fasciculus going underneath and going towards the superior parietal lobule, not to the angular. Imagine for the middle longitudinal fasciculus to go to the angular, it would have to make such a turn here. And it's all covered by the arcuate fasciculus and by the parietal ascent tract. So it really makes no sense. And this tract goes truly into the superior parietal lobule based on our studies. Just to finish, we'll cover a bit on the ventral language tracts, and I put it with interrogation because there's still some doubts on the true role of these fiber tracts on language function, but I hope to share some light with this. And I know the other speakers are going to talk about these tracts in more detail, so I'm not going to cover in great detail, but especially when I talk about the inferior frontal occipital fasciculus, that, as you know, goes from frontal to occipital or parieto-occipital. We have the uncinate fasciculus anteriorly and the inferior longitudinal fasciculus at the base of the temporal lobe. You can see the composition of these fiber tracts all together. And this is a step-by-step dissection of the inferior frontal occipital fasciculus. We can actually isolate this fiber tract and see its connections in the brain, and even taking it as we were doing fiber tracking reconstruction of this brain. But we also studied this track. We tried to segment it into different parts. But the things we found is, for example, this is a non-naturalized track. And also, we did not find any connections of this track with the temporal lobe or the angular gyrus. They all go posterior to occipital and parieto-occipital regions. That means that it's not involved in language. Well, we don't know. I'm not sure we do, but we have evidence from mapping intraop that it has to do with semantic function. And again, although we are not so accurate in knowing which track we're stimulating, there is evidence of its role in the semantic system. But more from an anatomical point of view, there is another point of controversy here. Why do we call it the inferior fronto-occipital fasciculus? Because in theory, there is a superior fronto-occipital fasciculus, but this track is another, I think, mistake in the literature where either fiber tracking techniques that were not very accurate, like DTI, or translation of knowledge from animal studies, make us find this superior fronto-occipital fasciculus, which actually we do not find in the human brain, as far as I can tell. So we did this study where both doing dissections, and there was a previous dissection paper also showing this, but there was no previous fiber tracking paper showing that the SFOF, the superior fronto-occipital fasciculus, in fact, is the false continuation between 3 different tracks. These are thalamic fibers with the stria terminalis, and they form a continuation, and they form this track that really doesn't exist. So if the superior fronto-occipital fasciculus does not exist in the brain, why do we call it the inferior fronto-occipital and not just the fronto-occipital fasciculus? But probably that's not such a relevant notion, but it's important that we are very critic when we do our dissections and our fiber tracking reconstruction on what we find, and with that, where we make sure that we validate our findings and we do not give for granted every fiber tracks that we are finding, because some of them are actually pseudo tracks and artifacts. I want to finish with this very interesting study that I did with my collaborators, Frank Ye and Will Hula at University of Pittsburgh. And this is an NIH-granted study over 5 years. And this is just the first part of our results. But what we did is we wanted to solve this controversy on which fiber tracks are related with language. And one of the things we can come up with is, okay, let's study a patient with a stroke, aphasia due to left hemisphere stroke. And Frank developed this local connectome matrix or connectomics study where you can correlate DSI data, diffusion data, with what Will Hula could do, the indices of semantic and phonological ability based on testing like picture naming. And, Will Hula showed me this, what they call the semantic weight and the phonological weight. So you can categorize speech deficits on patients based on being more phonological or more semantic. And what we did is correlating those measures, those behavioral tests, with the diffusion imaging data. And this is what we find. We found very interesting that, up here you have the phonological related fiber tracks, and down here the semantic related fiber tracks, or more heavily oriented towards phonological function or semantic function. You can see that the arcuate fasciculus is on both. And this supports the theory that the arcuate fasciculus has a ventral and dorsal component, or at least is a track that is involved in both semantics and phonological function. Because some authors say the arcuate track is not involved in semantics, it's just phonological, but this goes against that notion. There are other tracks involved, interesting how corpus callosum and cingulum shows up here, but especially let's look at the arcuate. And then if we go to the semantic tracks, it's interesting how we are seeing actually the inferior fronto-occipital fasciculus right here, and part of the middle longitudinal fasciculus too. And the uncinate. So if we put these two together, and this figure shows in blue the fibers that are more related to semantic function, while the green shows the fibers that are more related to phonological function. This, shows that there is almost a disposition of the tracks in the brain in a ventral and dorsal arrangement. And this is quite fascinating because this is a pattern that is repeated, all the time, ventral and dorsal arrangements, with the ventral being more semantic, the dorsal being more phonological. And maybe this goes along also with the dual S3 model that we'll hear more about later. So I believe this emphasized or provided further support of the role of the arcuate in both areas, but also of the role of the ventral tracts, IFOF and uncinate, on semantic function. So to conclude, we studied surgical anatomy of the fiber tract using fiber microdissection. You can see how this technique has really evolved to great level of detail. And similarly, DTI has evolved into high definition fiber tracking. So we can do really accurate computer-based fiber dissection. I believe it's very important to really be consistent with the nomenclature of fiber tracts, AF, SLF, parietal tract, etc. Also, we continue trying to investigate function of fiber tracts that are not fully known. And I think connectivity studies can really help in this area. Thank you very much.
Thank you very much, Juan. That was absolutely stunning collection of images from your personal work. These images that you presented from Max, if I remember, are they still prepared with the Klinger technique, or you have some modifications you want to share with us?
The specimens are prepared the same. What is a bit different is the tools that Max employs are, microforceps, like jeweler forceps for the most part. So it truly becomes microdissection technique, as you see, and is the amount of time and the talent he has doing this.
It looks like work of art. So thank you very much for this. I believe Anthony has a question to ask.
Oh yeah, I actually had two. I'll ask the one I put in the chat, which is so we have this SLF3, which is well described in the macaque, which is basically a PF, area PF, inferior frontal connection. And in the human, you can identify a similar connection as you showed, but you don't like that terminology. You'd rather call it the dorsal part of the arcuate. I didn't understand that. What's the difference between those components, I guess?
You mean for the SLF?
SLF3, the third subcomponent of the SLF3, which is sort of a direct inferior frontal, inferior parietal connection.
Sure. The SLF, we would call it, SLF3 would correlate with the ventral SLF, and the SLF2 would be the dorsal one.
So you would say they're different things or the same?
I would say they are different.
The SLF3 and the ventral arcuate?
Yes.
In what way in terms of their connectivity profile?
So because the arcuate would connect, I think that the terminations in the inferior frontal gyrus are quite similar for both. But what it makes them different is the termination on superior temporal and middle temporal gyri for the arcuate versus termination to the supramarginal gyrus for the SLF3 or ventral SLF.
Okay, that makes a lot of sense. Thanks.
Yeah.
And
as you know, for me, it's all about being trying to be very meticulous about this anatomical terminology and making these classifications, making homogeneous across the board. Thank you.
I suspect Jeremy might have a comment if he's still online.
Sorry, are you? Is that to me, George?
Yes. So, Jeremy, I think Anthony also put down a written question and says that what can we learn from the comparative anatomy? For example, if the SLF3 in macaque is the PF inferior frontal connection, and if it is beneficial to maintain the terminology for the human homologue.
So, yeah, I'm listening to this with great interest, and the work is gorgeous and beautiful to look at. I do think, this is helpful hearing this. I was going to just restrict my talk tomorrow just to the language system, but I think I'm going to have to broaden that to a larger consideration of the fiber systems, because there are some real issues to discuss here. I think that the 2 points that came up in the talk, one has to do with SLF1, the other with SLF3, and then the issue of the inferior frontal occipital fasciculus. So these are not trivial points, because there are some true species differences, perhaps. And then the other point that I think is critical here is to consider that the tracts are concretizations of a larger fiber system network that are the conveyors of information from one cortical area to another. So if you lose track of that, or if you look at the fiber tracks themselves, outside of the connections, you may find that one gets misled. SLF1 has nothing to do with cingulum bundle in any way, shape, or form. And SLF3 connects very different areas than the arcuate in the monkey. So I think we have to be careful about being humble and restricting ourselves to what we know to be data in terms of the monkey versus what we are looking for in the human. And that's where we can discuss terminology, connectivity, and then potential relevance for function in the human brain.
Mm. With regards to the SLF1, I would agree also that I don't think it's related to the cingulum bundle, and I have dissected this many times in cadavers. But I hope that we can discuss this a little bit later.
Yep. Yep.
And Anthony, Anthony, you said you have one more comment.
Just really quickly, because I don't want to get into the other talks, but for the IFOF, so Juan, you were arguing that this is a continuous pathway from occipital to frontal. But my understanding of these very large fiber systems is that at times you have fibers entering and leaving the fiber system along the course of it. So with the blunt fiber dissection technique, even the micro surgical technique, can you identify that this is not the case, that this large fiber system is not composed of smaller sort of contributions from the temporal lobe as well? Does that make sense?
It does. It makes perfect sense. I mean, I think many of us thought exactly the same as you are doing. The fact is, we are restricted to the limitations of the technique. As you were wisely saying, all I can say is with the techniques we have, we do not see those temporal lobe connections. And however, we see it for other tracks. We are very accurate on depicting U fibers and seeing the little branching of many fiber tracks, but we do not see that for the IFOF. That does truly mean that there is no temporal connectivity? I don't know. It means that the technique does not show it.
Also, your comment about the misnomer of the IFOF as inferior fronto-occipital fasciculus is very interesting, because I believe that the superior fronto-occipital fasciculus exists only in monkeys, and perhaps Jeremy can again help us with that. And I believe Ugur Ture has done some work where he demonstrated that what is considered to be superior fronto-occipital fasciculus is actually SLF in humans. And I also believe that the IFOF does not exist in the monkeys, so they don't have inferior fronto-occipital fasciculus, but they have only superior fronto-occipital fasciculus. Jeremy, you can help us with that one?
Yes. So and I saw in the chat about SLF2 and SLF3. I think it's critical to bear in mind that we're calling these tracks based on what they're linking in the cerebral cortex. And that's what the track tracing in the animal model allows us to do. It allows us to, for example, distinguish the inferior longitudinal fasciculus from the sagittal stratum, which is something that Desjardins had difficulty with. Now that we have the connectivity, we can do that. We can distinguish extreme from external capsules because we know what they're connecting. And in terms of the SLF2 and SLF3, they're linking parietal frontal, but quite different areas, and the bundles lie in different locations. And that's, I think, the bottom line is the monkey, I think, is ground truth. And then we have to know, based on that, what could be species difference in monkey versus human, and then all the things that people will talk about in the coming lectures. What are the gremlins hiding in the analysis with DTI? What are we getting right? What are we getting wrong with this spectacular new technology? And what's right and what's wrong from the dissection techniques now that's any different than Herbert Mayo had, or Desjardins had, or Minot or Gall back in the early 1800s? How much better is it now than before? Are we having the same problems? The inferior fronto-occipital fasciculus, Marco Catani and I went backwards and forwards on this for a long time. Is it a conflation of different tracts and therefore not real in the human? Is it a new tract in the human? I think that's an important question, because its absence in the monkey is something we have to deal with. Because then the question is it a real thing in the human, or is it a conflation of tracts that's not really a connection? Is there truly an occipital lobe to frontal lobe direct connection? That's an interesting question that I think we have to think about carefully.
Thank you. So time to move on, despite this super interesting discussion. I would like next to invite Chris Koutsanakis, who is a neurosurgeon and anatomist in Athens. And Chris, quite remarkably, has established his laboratory from scratch, and he showed initiative and a lot of scientific curiosity to start dissecting brains, and has recently published a quite substantial number of papers. So I asked Chris to discuss with us the language implication of the middle longitudinal fasciculus, which is poorly understood, and I hope that he will show us some equally beautiful images. Chris, many thanks for joining us today.
Thank you so much for inviting me. It's an honor and a pleasure to be part of this great society. Just to, as I'm a surgeon, this talk is about anatomy. Just a moment, I cannot share my screen. Okay. Professor Ribas said about this long-standing motto in anatomy, that anatomy to physiology is like geography to history. It describes the theater of events. So you cannot understand function and physiology without anatomy. I think that we are in a point that can enhance this long-standing motto. And apart from understanding, anatomy can redefine, reshape function. And at the end, you can integrate function and anatomy to what we call today functional anatomy. And the methods that we use in modern functional anatomy are basically in humans are basically these two: DTI tractography, which offers in vivo subcortical dissections, and the clicker technique, which offers ex vivo subcortical dissections. And I think that these two methods have supported the neo-association theory in what we call the clinical anatomical correlation method. What we call odotopy today is what Geswind put forward as the neo-association theory. So in modern neuroscientific research in humans, usually what happens is that DTI tractography, which offers indirect evidence, sets the pace, and then the Klinger technique, the quiet matter dissection technique with direct evidence, comes as a validation tool. So usually, again, these two methods have good concordance, but there are some exceptions. And one of these exceptions is the middle longitudinal fasciculus, is an exception. So it was very recently identified in humans, just in 2009, by this study by Nikos Makris and colleagues, through DTI MRI studies. And what the authors proposed at the time was that the connectivity of the MDLF is between the temporal pole through the superior temporal gyrus to the angular gyrus. And because angular gyrus is a language-specific cortex, they hypothesized that the function is related to language. The main function of the tract is related to language. Since then, and from 2009 till now, we have 11 focused original studies in the literature. 6 of them are mainly DTI studies. 4 are white matter or white matter and DTI studies, and we have only 1 awake brain mapping study. And the disagreement, the point of friction, is the connectivity pattern. What is the connectivity pattern of the MDLF? If it goes or it terminates angular gyrus and the supramalleus gyri, and as a consequence, what is the functional significance of that? So if it goes and it terminates to the angular gyrus, it may have a role in language. Very interestingly, all DTI studies support a termination pattern in language-specific areas, which means angular and supramalleus gyri, and therefore they have hypothesized a role in language networks. Whereas white matter studies, and the only one awake brain map study do not support this concept. So that's what was our again our vision. Our target is to see the anatomy and untangle the anatomy of the MDLF, and this was our paper published just 1 year ago. What we did is that we primarily used the Klinger technique in 20 hemispheres. We did focus white matter dissections, and then after the Klinger technique, we did a DTI tractographic methods in 35 normal individuals from the Human Connectome project. And what we did is that we first traced the the bundle, the fascicle, and then we try to see what is happening with the segmentation pattern of it. And I will give you an example of how we do the anatomical tracing through the white matter study. So you can see a left hemisphere and the cortical anatomy superimposed. This is the first step of the white matter dissection. You can see these very nicely U fibers that connect adjacent areas. And after dissecting and after dissecting the U fibers, removing the U fibers, you can see the SLF-AF complex, I will say, because there is a lot about the SLF-AF complex in the literature. So we see this bundle, which is the first bundle that we see. And after dissecting very carefully the SLF-AF complex, you can see fibers that are coming from the temporal pole towards the parietal lobe. And this is, if you can see the trajectory and the morphology and the silhouette of the tracks, the SLF is the green one, and the MDLF is what is seen is highlighted with a red color. Moving the dissections forward and posteriorly to the occipital lobe, we can have a bigger view of the middle of the fasciculus. So we can see here the MDLF going to the superior parietal lobule, to the parotoccipital area, and just under the vertical occipital fasciculus. And so this is the silhouette of the MDLF. Again, you can see a tract, fibers coming from the temporal pole through the superior temporal gyrus, going towards the superior parietal lobule, the paroccipital arcus or paroccipital area, and the occipital lobe. And during the study, it was evident and consistent in all hemispheres studied this segmentation pattern. So we found a 3 segmentation pattern of the MDLF, and we named it MDLF1, MDLF2, and MDLF3. So this is MDLF1 coming from the temporal pole to the superior parietal lobule. And as you see, we have, you can see some white straps under the bundle, which seems that which says that we can retract the bundle and pull it off the brain. So this is a specific white matter tract. So this comes from the temporal pole to the superior parietal lobule. This is highlighted in red. The second part of the MDLF comes again and originates or terminates in the temporal pole and goes to the parotoccipital area. So this is again, you can see it over the white straps, and this is highlighted in blue. And the third part comes again from the temporal pole and from the superior temporal gyrus from the temporal pole through the superior temporal gyrus to the occipital lobe. This is again highlighted in green. And this is diagrammatically what you can see as a segmentation pattern of the MDLF. You can say that the MDLF1, MDLF2 can be one part or one segment, or you can again say that it is two segment configuration. I think that's the same thing regarding axonal connectivity. In our study, there was a very good concordance between DTI and white matter dissection. So what we found during the dissections was evident again with DTI studies. What we didn't find is that we didn't find any termination pattern, or the termination pattern was very, very minimal to the angular gyrus. We didn't find any significant leftward asymmetry, both in DTI and white matter. And again, from the literature, we know that in the one Awake My Brain map study, there was no language interference during the stimulation of the tract, which is very superficial. It's a superficial trunk. So in my opinion, and I think that it is almost safe to say that the MDLF has no big role in language. I think that because there is a tight anatomical relationship between the MDLF and the transverse gyri, the gyri that are in the superior temporal gyrus, which are the primary and secondary auditory cortices, and because the tract terminates to specific subregions of the temporal pole, which are believed to subserve auditory function, we can say that the middle of the fasciculus may have a role in auditory function. And in parallel to the what and where auditory pathways, to the what and where visual pathways, we now have supporting evidence for the what and where auditory pathways. So the what auditory pathway is processing auditory object identity and characteristics, and it usually is related to the anterior auditory cortex, whereas the where auditory pathway processes the location of the characteristic of sound and is related to the posterior auditory cortex. So the anterior auditory cortex is what we call the Hessel gyrus, and the posterior auditory cortex is what we call the posterior, is the gyrus posterior to the Hessel gyrus in the superior temporal gyrus. So if we can match axonal connectivity to putative function of the tract, we can say that MDLF, the first segment, the MDLF segment 1, because it goes through and traverses the prime auditory cortex, it can be a part of the what auditory pathway. Whereas the MDLF, the second part of the MDLF, which crosses the second auditory cortex, can be related to the posterior, to the where and auditory pathway. This is in line, our hypothesis is in line with a very, very recent paper published by Latini and colleagues in Protein Anatomy just 1 month ago, January 2021. And we have almost the same hypothesis, both in the segmentation pattern and the functional implications of the tract. So coming very quickly to the inferior longitudinal fasciculus and some quick facts about anatomy. It's a major occipitotemporal white matter tract. It usually, you can see 3 segments of the tract: the dorsolateral occipital segment, the fusion segment, and the lingual segment of the tract. The connectivity for the dorsolateral occipital segment is from the superior occipital, middle occipital, and inferior occipital gyri to the temporal pole, mainly middle, inferior temporal gyri, and fusiform gyrus. And the fusiform segment comes from the posterior fusiform gyrus and connects this gyrus to the temporal pole again, T2, T3, T4. Hemispheric asymmetry is debatable across studies, so we don't know if it's more on the left or on the right. I'll show you some examples how we can trace the dorsolateral tract of the ILF through white matter dissection. So this is the left hemisphere again. These are U fibers, and here just under the U fibers you can see this tract here that forms what we call the dorsolateral tract segment of the ILF. And again, this is the inferior part of a left hemisphere. That's the U fibers just below the cortex, and this is what we know as the fusiform segment of the inferior longitudinal fasciculus. Again, connecting the occipital pole to the temporal pole. Again, in terms of connectivity and functional anatomy, the ILF connects the visual cortex to the temporal pole and through the visual object form area, which is an area where we recognize objects, and through the visual word form area, which is an area where we recognize words, especially on the left side. So if we damage the tract from the visual cortex to the object recognition area, we have what we call a visual paraphasia or formal paraphasia. Whereas when we damage the tract from the visual cortex through the visual word form area to the temporal pole, we have what we call alexia. And alexia is when we have difficulty in reading, so we have reading deficits. And visual paraphasia is when we choose the wrong target word, but which has visual similarity. So it's knife for nail or cat for mask. So the words have visual similarity, but we choose the wrong target word to say. And this is what we call the visual or formal paraphasia. You must have in mind that these two functions have the double dissociation. So you can have alexia and without visual paraphasia and the other way around. So again, this says that there are two segments of the tract in each function. I think that this again is what is very close to what we say about the ILF, again by Latini and colleagues. This is I think what is evident in the literature about the segmentation and the functional hypothesis of the tract. And I would like to thank my colleagues in the Athens Magnetic Laboratory, and thank you for your attention. Thank you.
Thank you, Chris. That was very interesting work, particularly with regards to the segmentation of the MLF to 3 segments. The previous work from Roton's lab was 2 segments, anterior and posterior, through a line through the preoccipital notch. What would be interesting to see is the relationship between the IFOF and the MLF. And according again to Roton's work, on the anterior half, it's supposed to be superficial to IFOF, and the posterior half intermingles with the sagittal stratum. Are you able to provide any special relationships based on your own dissections?
You mean the ILF and the IFOF?
MLF and IFOF.
The MLF and IFOF, yes. So it's clearly shown in dissections that the MLF is above IFOF. So it's more superior to IFOF in every case. They intermingle when they go backwards towards the sagittal stratum, mainly in the occipital lobe. But in the dissections, you can see the MDLF lying above the IFOF in all cases. And I think that this is very evident in the literature as well. So it's above IFOF.
And with regards to the origin, some authors or researchers feel that it originates from the superior temporal gyrus. Some include the entire temporal lobe, entire temporal pole. Have you got any views on that?
I think that what we found in our study, and I think that it is again in line with most white matter dissection studies, is that the MDLF initiates or terminates in the superior temporal gyrus, in what we call the temporal pole of the superior temporal gyrus in a way. So it's mainly, in my view, a bundle that transverses the superior temporal gyrus from the temporal pole to the superior parietal lobule, perioccipital area, and occipital lobe above IFOF in every case that we've seen in the lab.
Thank you.
Thank you.
So now we'll move to the last talk of this session, which is the anatomical basis of language and cognition. And I would like to welcome Eduardo Ribas, which is Eduardo's Guilherme's son. And I know Eduardo again for a few years. He has learned a tremendous amount of anatomy, and he has spent quite a bit of time with Roton, with Ugur Ture, who reinvented the idea of wide fiber dissection, and also with Dufault in Montpellier, studying the function of the language. So Eduardo will discuss with us the central core of the brain, the area between the insular cortex and the ependymon of the ventricle, which is super important for language tracts. So Eduardo, thank you very much for joining us.
Thank you. Thank you for inviting me, George. And first of all, I want to say that I'm truly happy to be here and to see all these speakers together, all of them have influenced me so much during my studies and continue to influence me through their works. And just as Dr. Juan said, I truly believe that the neurosurgeon or anyone who is seeking deeper knowledge into neuroanatomy has to do some time into fiber dissection or into a neuroanatomy lab. And I did my time with Professor Albert Roton, just like Dr. Juan. And most of my dissections that I'm going to show are based on this fellowship that I did with him, that I am forever grateful for that. So my mission today is to talk about the central core. So here's a lateral view of the brain. We see in the middle the Sylvian fissure. And if we open the Sylvian fissure, we'll see the lateral surface of the insular cortex that is divided from the rest of the brain by its limiting sulci. It has one anterior limiting sulcus, one superior limiting sulcus, and one inferior limiting sulcus. It's interesting that the insula is the lateral aspect of a block of structures that is at the morphological center of each hemisphere, and that's very different from the rest of the brain. While the brain has gray matter on its surface and white matter on its inside, this block of structures, they have nuclei of gray matter that appear to be encapsulated by white matter. And usually this place of the brain is studied by this axial cut that we'll have in every neuroanatomy book, where we have this alternation of gray matter and white matter that appear to be in layers. So coming from outside in, coming from lateral to medial, we can name all these layers. The first is the insular cortex, so it's gray matter. Then we have extreme capsule. Then we have claustrum, external capsule, lentiform nucleus, and internal to the lentiform nucleus, we have the internal capsule, so divided into 5 segments. So here's the anterior limb, genu, posterior limb. Heterolentiform would be posterior to the lentiform nucleus, and sublentiform inferiorly, we cannot see here. Medial to the anterior limb, we have the head of the caudate nucleus. It is also a round nucleus that's wrapping the thalamus. So at this axial cut, we just see the head, but it's wrapping around the thalamus, and here is the thalamus medial to the posterior limb of the internal capsule. So we can call this region the central core, but I want to reinforce that the central core is more one archaeological, architectural point of view of this place, because we are putting together structures that have very different origins. So we are putting cortex together with claustrum. We are putting claustrum together with the other basal ganglia, just as Dr. Juan said. They have pathways that are completely different, but they are seen together as basal ganglia. We are putting the thalamus, that's the most important part of the diencephalon, together with all these other structures. But morphologically, seems nice to understand that each cerebral hemisphere will harbor on its morphological center one central core, and then we can understand the position of the other cerebral lobes in relation to the central core. So we can see the frontal lobe anteriorly, parietal lobe superiorly, occipital lobe posteriorly, temporal lobe inferiorly, insular cortex on its surface, and then the limbic system, the limbic lobe, wrapping around the central core. It's also interesting that inside the central core, the white matter has been called as capsules, but they are really going or arising or just going through the central core, but they are going towards the other cerebral lobes. And so they have to pass into this small space here, limited laterally by the insular limiting sulci and medially by the lateral ventricle. So here they have all the capsules have to be together in order to continue to the other cerebral lobes. Here we are making this cut deep to the insular limiting sulci and opening the lateral ventricle. And in this cut, we are cutting all these fibers that inside, they are called the capsules, but they are continuing outside the central core toward the rest, all other cerebral lobes. And here we can see the limbic system wrapping around the central core. So we have an outer C or a cortical C, mainly formed by the parahippocampal gyrus, isthmus of cingulate gyrus, and cingulate gyrus terminating in the septal nuclei. And internally, we have a subcortical C that is formed by the hippocampal formation that's mainly formed by the fornix that ends in the hippocampus. Sorry, it's the other way around, the hippocampus that ends in the fornix. But what we are doing here, we are opening the choroidal fissure, this inner C of the limbic lobe, this subcortical C. It's already detached from the central core, and we can just increase this detachment by enlarging the choroidal fissure. This is this fissure that's between the thalamus laterally and the fornix medially. Here with my instruments, we can open it superiorly, posteriorly, and inferiorly, just to show how the hippocampus and the fornix can be detached really from the thalamus, but naturally they are already detached. Anterior to the head of the hippocampus, we have the amygdala, and the amygdala is not naturally detached from the central core. The naturally continues itself with the central core, particularly with the globus pallidus. I'm going to show this later. So here is the outer C of the limbic system. So we can see the cingulate gyrus, Here's the isthmus of the singular gyrus, parahippocampal gyrus. And if we turn this piece of brain to the other side, we can see what's inside. And we see that inside the parahippocampal gyrus, we'll have the amygdala anteriorly, then the hippocampus and the hippocampal fibers getting together to form the fornix. And the fornix has this C-shaped because it's wrapping, it's really going around the thalamus that would be on this space here and laying on its flat surface here that's called the subiculum. Okay, so we can see that naturally the amygdala at the head of the hippocampus, they are anatomically detached already. And here is a superior view to see the hippocampus, and it's famous shaped into a foot of a lion. He's the pes hippocampus. And in this space here, we would have the thalamus resting on the surface here, that's the subiculum. And of course, if we see from the subiculum medially, we can see that between the subiculum and the fornix, we have this vertical displaced gyri. This is the dentate gyri, also part of the limbic system. Here's another preparation. We can split the sylvian fissure and make this deep cut deep to the insular limiting sulci, and we'll reach the lateral ventricle. And you can see how this piece of brain, how the central core can be really detached from the other cerebral lobes. And if we progress the dissection even medially and open the choroidal fissure, then we can separate the limbic system as well from the central core, but not the most anterior part of these limbic structures, mainly the amygdala and the temporomedial parts, because they really continue into this ventral part of the central core. So some people can use other lines to do this connection, but one line that I learned with Professor Wen how to do it is to connect the inferior choroidal point to the ICA bifurcation and to make a cut there, and this cut will be laterally and parallel to the anterior choroidal artery, and will cut the amygdala from the central core. This is one stage of the temporal lobectomy. Me as a neurosurgeon have to do sometimes. Other people will look to the optic tract and make this cut, but this is just to show that the amygdala continues itself with the central core, and to do this detachment, we have to make a cut. Here's one anterior view of the brainstem with the two central cores on its top, just like a two-headed dragon. And each cerebral hemisphere will harbor a central core, and the other cerebral lobes will be placed around the central core. Frontal lobe anteriorly, parietal superiorly, occipital posteriorly, temporal inferiorly. And we can see here the limbic hippocampus, because the limbic structures will be around, and the insular cortex superiorly. There is not a natural division between the central core and the mesencephalon, but most anatomists will see the division between the mesencephalon and the diencephalon being the inferior aspect of the optic tract. From fiber point of view, we can dissect the brain from lateral to medial. So we'll first find U fibers under the all cerebral cortex. Then we'll find the longitudinal, the superior longitudinal fasciculus that Dr. Wan, Dr. Jeremy had already talked a little bit. And just to show that these fibers here, the SLF is not inside the central core, it's a perisylvian pathway. Then we come to the insular surface, and we can see that the insular surface can be subdivided into two compartments. One anterior compartment made by short insular gyri that will converge to an apex, and a posterior insular compartment that will be made of long insular gyri that will not reach the apex. If we decorticate, if we remove the insular surface, we will reach the extreme capsule. These are the short association fibers that are connecting together all insular gyri. But as George said, they are continuing underneath the opercula, underneath the insular limiting sulci towards the opercula. Okay, so they will also connect the insula to the frontal, parietal, and superior temporal gyrus. If we continue even more medially, now we begin to see the claustrum. There's this beautiful article from Dr. Juan that says about the dorsal claustrum and the ventral claustrum, being the dorsal claustrum more organized into this sheet and more compact thin layer of gray matter. And here we can see this antero-posterior disposition of 3 fiber bundles. So from anterior to posterior, we see first the uncinate fascicle connecting the frontal orbital surface to the temporal pole. It's passing really just behind the Lyman insula. Then we have the IFOF, and we had this discussion already here, if the IFOF is the most proper name. I know the group from Professor Yashigi and Dr. Ugorture prefer to call occipitofrontal fascicle because there's not a superior one, and they believe the information comes from posterior to anterior, so they will prefer the name occipitofrontal. But a lot of papers do have this acronym, inferior frontal occipital fascicle, and I put this name here. From the claustrum, we have a lot of fibers arising from the claustrum, and they are positioned superiorly to the IFOF, and these are called the claustrofugal or claustrocortical fibers. And we can see this, all these 3 bundle fibers here, they are all superior and inferior to the claustrum. They are superficial and deep to the claustrum, so they contribute to form the extreme and the external capsule. Okay, it is important to know. We remove the claustrum here, we see the putamen, and if we remove some of the IFOF fibers, we begin to see the anterior commissural fibers. They are connecting both temporal regions, so they're coming from one temporal region to the other temporal region and passing inferior to the putamen. Here I resect the putamen already. I'm seeing the globus pallidus. Just as George said, it's more pale, it's more dense. It can be see on the fiber dissection, and we can see the anterior commissural fibers passing here on this canal that is the ventral aspect of the globus pallidus. This is the graciole canal. So fibers from the commissural fibers are coming laterally and then opening itself into the temporal lobe. If we remove the globus pallidus, now we see everything that's medial to the putamen, that's medial to the lentiform. These are the internal capsule fibers. They have this radial disposition because they are projection fibers, and most of them have to get inside the cerebral peduncle, right? So they have to converge to get into the cerebral peduncle. So they have this radial appearance. And this special place here that is anterior and inferior to the commissural fibers will not have at least so much internal capsule fibers here. So the lateral gray matter that belongs to the putamen will merge with the medial position gray matter that belongs to the head of the caudate, creating a nuclei called the accumbens, or other people will name it the ventral striatum, because these are striatal nuclei, and the gray matter laterally belongs to the putamen and medially belongs to the caudate, and together they will be called the accumbens. So it is this place here, marked here. There will not have internal capsule fibers here. Here's a more far away view. There we can see the internal capsule fibers. They continue outside as the corona radiata, and posteriorly the sublentiform and retrolenticular fibers. They will terminate at the calcarine fissure. These are geniculocalcarine fibers, part of the optic radiation. If we go deep to the optic radiation fibers, we have a more vertical position fibers. These are the callosal fibers, the tapetum of the corpus callosum. This is not inside the central core concept, it's outside. So just to show it's outside and mostly lateral to the ventricular atrium. Now we already opened the temporal horn of the lateral ventricle. We can see the hippocampus here, and then I just enlarge the choroidal fissure and detach the central core. The central core has a special pattern of vascularization. Most of the brain has the pattern of vascularization that's made from large vessels over the cerebral brain, cortex, and then from its large vessels will arise small branches that will penetrate the cortex and supply the cortex. The central core has this pattern of vascularization on its insular surface, but deep inside the central core, it has a different pattern of vascularization made by penetrating arteries. These perforating arteries, they will arise from the most proximal regions of the ICA bifurcation, M1, A1, and will penetrate the anterior perforated substance, one on each side, and more posteriorly, they will penetrate the posterior perforated substance. So here, posterior to the mammillary bodies here, and the posterior perforating arteries, they are arising from the basilar bifurcation and proximal regions of P1 on both sides, and they will be called here the thalamoperforating arteries because they will supply mostly the thalamus. The anterior perforated substance, they will be perforated by the lenticulostriate arteries because these arteries here, they will supply mostly the anterior part of the central core. That's the lenticulostriate arteries. So the lentiform, the striate nuclei, and the internal capsule. So at this dissection here, I removed the central core that was on our side. Here's the lenticulostriate arteries. They will penetrate the anterior perforated substance and supply the most anterior part of the central core, while the posterior perforating arteries, they will penetrate the posterior perforating substance. They will supply mostly the thalamus, the posterior part of the central core. So in another dissection, come from lateral to medially, we can understand and remove. Here we are removing the IFOF, but I want to show how the lenticulostriate arteries, they will penetrate the anterior perforated substance and then continue superiorly to supply the lentiform. So some of fibers, some of the vascularization can come anterior to the anterior commissure. They will mostly go to the putamen, and another peduncle will be posterior to the anterior commissure. They will go mostly to the globus pallidus. This is a superior view. If we remove the lentiform, these same vessels, they will continue even more medially to supply the internal capsule. And later on, they will even more medially to supply the caudate. So they have a long way to go to the caudate. Here I am removing the putamen, and you can see how this name perforating arteries is very appropriate because they're really perforating all these nuclei. Here I am removing the putamen, removing the globus pallidus. And here is also superior view where we are detaching with my instruments here the caudate from the internal capsule, and we can see the final place of these vessels that begin medially and go even that begin laterally and go medially to terminate at the caudate nucleus. The Heubner recurrent artery, it has a more direct route to the head of the putamen, that's here, while the other lenticulostriate arteries, they will first penetrate the anterior perforated substance, supply the lentiform, then internal capsule, and then caudate nucleus. Attention to this place here. If we remove the ventral striatum, that's the gray matter here, we can see the ventral extension of the anterior commissure. The anterior commissure will have a ventral extension and a lateral extension. And if we see the anterior perforated substance from superiorly, we can see that it's formed by the ansa peduncularis. The ansa peduncularis is a set of fibers that are arising in the temporomedial region, amygdala, and you continue more medially. We have a paper about that. You can go and see it. And at our research, we suggested to understand the route of these pathways inside the central core and to divide this region into quadrants regarding the position of these fibers, but also understanding that it has a parasagittal split that cannot be crossed surgically, made by the internal capsule and the vessels that will supply it. Okay, thank you, George. This is my email if everybody, if anyone wants to get it, to get to me.
Thank you very much, Eduardo. That was a spectacular talk of insular anatomy. I wonder if Nina can join us for a sec, because you have showed very nicely the insular cortex, and Nina Dronkers have very nicely demonstrated that the posterior short gyrus of the insula is involved in the motor planning of speech. And I wonder, Nina, if you can still hear us, how did you make this assignment? How did you assign anatomically your territory on this particular cortical parcel?
Well, we looked at this in stroke patients. We looked at those who had apraxia of speech versus those who did not. And all the patients with apraxia of speech had involvement of the superior tip of the precentral gyrus of the insula, while those who did not have apraxia of speech had this area completely spared. So we looked at the CT and MRI reconstructions of all of these patients in order to make this determination. Of course, we looked at, many different atlases to be able to affirm that it was the superior tip of the precentral gyrus that was involved in the planning of complex articulatory movements. So, for example, you don't need your insula to say banana, 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 but you do when you say spaghetti, spaghetti, spaghetti. You can see I even tripped over it myself. Those consonant clusters and longer words are more complex in that they involve a lot of transitions between the different articulators.
Mm. Do you have any idea what is the basis of autocorrection? So they get it wrong a few times and then they correct themselves. Is there any reasonable explanation for that?
Well, I think they hear their errors. They definitely know that they're making mistakes, and so they try to correct. But with those more complex consonant clusters, for example, it's hard for them anyway. And so if they try again to pronounce the same word, they're still going to have trouble with those clusters. That's why we ask patients to say these complex articulatory gestures rapidly and in succession, so that we can really hear where they make those irregular errors.
Do you feel that there is also a component deeper than the insular cortex that might be affected by the stroke territory?
Yeah, I think that, there are certainly other regions, that participate in speech production and, pardon me, and I will talk about that a bit tomorrow. There are certainly lots of fiber pathways, as we just heard by this excellent talk by Dr. Ribas, that pass near that area. What's interesting is that we don't seem to see involvement of those tracks per se. It's not the disruption in the precentral gyrus of the insula is not because of an underlying fiber tract lesion. It's really in the cortex of this precentral gyrus. It's remarkably consistent. Having said that, I do see in our tractography that there are fibers that drop down into the precentral gyrus of the insula from the arcuate fasciculus. So I myself would be very curious to ask Dr. Ribas and Dr. Miranda if what they think about the fibers of the arcuate that drop down into the precentral gyrus of the insula as well. They're often ignored in discussions of the arcuate. And I think it has a lot to do with where you place your region of interest when you tease out the arcuate fasciculus in tractography. So I'd love to hear what one sees in dissection.
Mm-hmm. Juan, shall we start with you and then we'll go to Eduardo?
Yeah, that's a very interesting point. So connections from the short insular gyri to the arcuate, I think they exist in the form of U fibers, but I am not sure whether there are insular fibers that incorporate into the arcuate tract and go to a certain distance. I really am not sure. I think there are more U fibers between the insular gyri and the adjacent, frontal opercula or parietal opercula, etc. That's what I've found, but I'm not certain.
I agree. I would agree with Dr. Juan. Of course, I do believe it can have a minor contribution because everything is possible, right? But the extreme capsule, it's made of U fibers, and once you go to the more outside boundary of the insula, the short U fibers, they will go underneath the insular limit and suicide towards the opercula. So maybe these are the short U fibers of the extreme capsule. And as Dr. Dronker said, the precentral gyrus, I think she was referring to the most posterior short gyrus of the insula, right? That's really the first anterior to the central sulcus of the insula. And the central sulcus of the insula, it's almost aligned with the central sulcus of the brain. So the precentral gyrus of the insula would be almost aligned with the precentral gyrus of the brain. And the U fibers of the extreme capsule might connect them. I see that's really plausible. I think it's really possible.
So when you say, Nina, the arcuate fasciculus drops down to the insula, are you referring to the extreme capsule or insular cortex or the U fibers? Have you got anything specific in mind?
Right. Yeah. And I should have clarified. The fibers I'm thinking of actually come off the temporal lobe, particularly the middle temporal gyrus. And then as they're coursing over the ventricles toward the frontal lobe, there's a branch that drops down into the precentral gyrus of the insula. We've been looking at that pretty extensively with CSD tractography and being able to follow those tracks pretty well, but we haven't dissected it. And so that's why I was really curious if you see those. Again, they're not well advertised. Excuse me. And so I was curious to know, I know people have mentioned it periodically, but just curious whether it had been studied more. I agree about the U-shaped fibers, definitely, particularly from the operculum.
So when you're referring to the temporal fibers, it is likely that if it is superior middle temporal gyri, possibly the ventral arcuate fasciculus originates from this, and as it goes around the supramarginal gyrus and to the pars opercularis is the closest fiber tract I can imagine being in proximity to the insula. I'm not aware of any connections, but I think topographically, the ventral arcuate fasciculus would be the closest from any of the fiber tracts. Do you think that this is reasonable, Juan?
You know, I am not sure. I've seen those connections that Dr. Dronker is mentioning with fiber tracking. With fiber dissection, we actually should study that. That's a good idea. I'm going to ask Max to look into that. The thing is with fiber tracking, as you all know, it's very easy to get artifacts, and that area is a crossing point where you have the arcuate going in one direction, and then you have the fibers of the external capsule going up. So there is an overlap, and it's so easy, especially with DTI techniques, so easy to get false continuation, and you can see tracks connecting the arcuate with the insula very easily, but those we know are false. But do they exist further connections? I don't know. We need to investigate.
Well, I'm just down the road in Berkeley, and I would be delighted to come and join you sometime if you do that dissection. Thank you.
Thanks so much.
Great. Thank you very much. That was a very productive discussion. Now we're going to discuss some cases submitted by our faculty. And what I would like to do is just ask our faculty to put our heads together and see how we can plan this surgery. What we've been doing at the University College London for the last few years is that before we decide how to map every patient, we sit around a table with speech and language therapists, psychologists, and cognitive neuroscientists and functional neuroimaging. And then based on the patient's pre-op data and imaging, trying to understand what is the best way to test this function. So I would propose that we will do something similar here. So this is a case that Juan has submitted. And once Juan describes a very brief story, clinical history, then I would ask our anatomist to give us a brief definition of the cortical parcel, and then perhaps our cognitive neuroscientists and language experts can make some comments as to the function and how to best test this. So Juan, do you want to give us the clinical history first of all?
Sure. Yeah, it's very simple. This is a young guy, 22-year-old kid that presents with first-time seizure. It's a generalized seizure. And on exam, she's basically normal. On the exam we do at the bedside, he might endorse some mild word-finding difficulties, healthy otherwise. Okay,
good. So, Guilherme, would you like to tell us what you think is the cortical involvement? What are the cortical parcels? And at the same time, our delegates, please write your answers. What anatomical structures do you think are being involved in this case?
Can you hear me?
Yeah.
Okay, we can see very evidently that we are talking about the supramarginal gyrus area. We should understand this as a region. You see that the superior temporal gyrus is continuous with the supramarginal. You have all this abnormal signal. And as we know, it's very common for these tumors, even when they are high grade, to stay in a region, to stay within some boundaries. So I would definitely say that this is circumscribed to the supramarginal gyrus. We can see that it's swollen, but I would consider it.
Do you think there's any involvement of the anterior angular gyrus?
I think, you can see, I would think that it's the intermediary sulcus of Jensen is displaced posteriorly. We would have to have a cut more lateral to make sure of this, but I can see that the middle temporal gyrus is getting into the angular gyrus region. And you can see that the superior temporal gyrus is continuing with the tumor posterior and superiorly. So I would assume this tumor is only within the what would call the supramarginal gyrus region.
Thank you. So
we have always to consider that especially when we're talking about a low-grade tumor, that in order to get to the angular gyrus, it would have to destroy the intermediary sulcus, or it would have to go underneath the intermediary sulcus. And so very probably that's not the case here.
Thank you. So let's say that we would like to test the function of the supramarginal gyrus, and I think we hinted earlier that there's quite a few and diverse functions. So I might have to put some of the faculty on the spot, if that's all right. So can I start with Anthony? And Anthony, do you think you would be able to share with us some of the functions, some of the measurable function of the supramarginal gyrus?
I think in this case, I would be concerned about some phonological working memory deficits. There's this great study by Dufault, I think from 2003 or so, where he does a stimulation study of the white matter under supramarginal gyrus in the left hemisphere, and that was the main deficit he identified. So that would be a place to start.
Mm-hmm. And could I ask Edward Chang, who has published a number of maps of stimulation findings, what would be his approach to map this intraoperatively? Okay, he might have some connectivity problems. Nina, can I come back to you and ask you whether you have any views from your quite large stroke database when you have patients with stroke in the supramarginal gyrus territory? What sort of deficits do you normally see?
Yeah, we typically, I agree with Anthony that when the lesions involve supramarginal gyrus and dip down into the posterior superior temporal gyrus, what we see there are the phonological, the really auditory, verbal, short-term memory deficits. Most of these patients are characterized with a conduction aphasia, but where the key finding is that they have these echoic memory problems. So if you ask them to repeat, they can repeat simple sentences that are easily understood and repeated back just based on their meaning. But if they get a sentence that is more low frequency in terms of how it occurs in normal conversation, those patients have a lot of trouble with those sentences. So the best example we have is really a patient was asked to repeat, "The pastry cook was elated." And the patient struggled and said, "Yeah, I just don't know." And we said, well, what was the sentence and was about? What was it about? And he said, "Well, it's something about a happy baker." So you see, the meaning gets in, but they're not able to repeat the sentence verbatim. So that's consistent with an auditory verbal short-term memory deficit, the kind of phonological store that Anthony was talking about. So I was curious when I saw this lesion, since it does sort of dip into the posterior sphirotemporal gyrus, whether that patient would have had repetition deficits for these more low frequency sentences. I'm also curious as to Dr. Ribas's opinion as to whether it does sort of drop down into the posterior sphirotemporal gyrus as part of its sweeping connection to the supramarginal gyrus.
What do you think, Guilherme?
Well, let me first clarify that when we're talking anatomy, we're trying to define a compartment. And this is Professor Yasagil's idea, you know. For example, it's the different when we talk about a compartment or when we talk about a system. When we talk about corona radiata, internal capsule, well, we're talking about a system. When we talk about the central core of the brain, about the insular block that Eduardo just talked about, we're talking about a compartment. We have sometimes different functions, different fiber systems, different systems within this compartment. I think the gyro concept, it's a concept of a compartment. And of course, then you deal with nomenclature, because where it ends something and start the other structure, it's very difficult to define. I think you can have something in a compartment and you can have a function, abnormal function that is due to a system that this compartment is linked to the other compartment or to another system. But anatomically speaking, strictly speaking, I would say that this is pretty much confined into the supramarginal gyrus. It doesn't really get inside the superior temporal gyrus. It doesn't mean that it doesn't have interfere with the system from the so-called Wernicke area, but anatomically speaking, I would say that it's confined to the supramarginal gyrus.
Thank you. Juan, do you want to tell us the tractography findings described to us?
Yes, of course. First, let me say that I agree very much with Professor Ribas on this being in the supramarginal gyrus, and his concept is so important. I've been influenced by his thinking for many years, and I very much agree by that concept of the compartmentalization of these tumors. And funny enough, this tumor was categorized by the radiologists as being in the angular gyrus. So there is so much confusion about supramarginal versus angular gyrus, and I think this is relevant. In terms of the fiber tract, so we see easily, you see this actually is DTI acquisition, so it's part of the routine scan, and then is reconstructed with DSI methods as developed by Frank Ye. So it's not very good quality acquisition, but very good quality reconstruction. Now, that being said, because if it were DSI acquisition, it would look better, the fiber tracts would look much better than this. Anyway, this is in brown, you can see SLF, and we could say this is SLF, the ventral one, the one that connects with the supramarginal gyrus. And then we have a dorsal one, I meant. And then, although here maybe they're both together, that's the controversy of whether we can separate always or not. Then we have arcuate fasciculus clearly in yellow, and in blue you have the temporoparietal Aslan tract, or the so-called vertical or posterior arcuate fasciculus. And also interesting, you can see, which is one of the beauties of fiber tracking for these cases, is that you can actually get the idea that the fiber tracts are being displaced. You can see the yellow tract, the arcuate, there is some mass effect on it. And this is a beauty of low-grade gliomas. Many of those act like causing mass effect on the fiber tracts.
Thank you. And do you want to tell us what happened next?
Well, these are the cortical terminations. Well, we took the patient. I'm going to actually show the case later, but so we can actually see the operation itself. But we did him with him awake. He did tolerate the operation really well. This was a young kid, as I'll show you later, and very motivated and well ready for the operation. And we were able to do mapping. There were, I don't want to disclose the whole story because I'm going to tell it later, but we did have some paraphasias, especially semantic paraphasias, on the deepest portion of the cavity, and what I think correlate with the arcuate fasciculus. And that's when we stopped the resection. But also some other at the posterior aspect of the resection, as I'll show you later, he had some out-of-body experience that I'm going to explain later in more detail, something very interesting. But in terms of speech, he has some semantic paraphasias. But as I decreased the power of the stimulation, he was tolerated and doing less and less mistakes. So I continued resecting. And this is also an interesting thing. You see the post-op fiber tracking, which is not very often done, but I think it's very important to see how it looks after the operation. The arcuate fasciculus were missing part of it. Part of it was infiltrated. But his speech is grossly intact, although I would love to have Dr. Dronkers doing an accurate evaluation of his speech. I'm sure he will find deficits there, but grossly intact.
How did you test the arcuate fasciculus one? What was your paradigm?
It was picture naming.
Do you ever use repetition of sentences or words?
Yes, also repetition of sentences or words. Yes.
Great. Okay, well, that's a great case. And we might just do one more quick case, and then we might have a very fast, quick break for a few minutes. So let me find one more case that Professor Ribas has kindly sent. So this is Guilherme Ribas' case. So Guilherme, do you want to present the case briefly?
Okay, this was an orthopedics, 56 years old, who November 2019 came to us with a difficulty to speak. And he had this tumor that, if we can see here in the lower one at the left side, the axial cut, we can see that it's a cystic lesion with some enhancing areas, pretty much inside the precentral gyrus. But unfortunately, other than that, he had some abnormal signs also in the MRI, in the oncus, in the amygdala, as you can see here in the bottom of the right side. The next slide. Well, at this point, we discussed with him that we thought about doing awake surgery and trying to remove as possible this lesion. But since he had other abnormal signs, he had a multicenter lesion, and it was sort of deep lesion here within the precentral gyrus. We talked to him, and both together we decided to go ahead and do a biopsy. We did a stereotactic biopsy that showed a high-grade glioma. It was indeed a wild IDH GBM. And then he was treated with the STUB protocol. And when he was at the 10th cycle, more or less, he was worse. He came back worsening of, again, difficulty to speak. It was more dysarthria than dysphasia, but he was both of this. And then we did another MRI. Indeed, this previous MRI already had a small cyst that was developing, and he had now this huge cyst, as you can see, within the precentral gyrus as well. So next slide, please. He was very symptomatic. He didn't get better with the, yeah, please come back to the previous one, George. Just leave this one. That's a very interesting slide anatomically. So we decided to go ahead operating him awake. So you can see here the 3D reconstruction. And let me emphasize again this beautiful U that is the opercular part that you can see just in the anterior aspect superior to the Sylvian fissure, and with the precentral gyrus coming in there. So just coming from this recognition of the U-shape opercular, you can see that the abnormality of signal is exactly within the precentral gyrus. And then again, the more you know, the more you see, and the more you get accustomed, the more you identify. If you look here at the left side, in the right side, at the surgical appearance, you can see the beautiful U. You can recognize exactly there, just above the Sylvian fissure. So you know anatomically where is the precentral sulcus, you know where is the central sulcus, and of course you know where the tumor is. In this case, next slide, please.
So, Guilherme, would you mind if we make this as a discussion? So if you wouldn't mind, let's go back for a second. So we know that this is involves the precentral gyrus, but also looks it goes more anteriorly, and anatomically looks like it could be the dorsal premotor cortex, is possibly middle posterior part of the middle frontal gyrus. So I wonder if Roger Lemon would have any, who's a world-class expert on the motor system, would have any thoughts on the function of the ventral, I beg your pardon, of the dorsal premotor cortex.
Yeah, a good question. Yeah, I wonder what the motor problems were for this patient. Did he have any motor problems? Somebody's mentioned this on the chat as well. The sort of thing you might expect from experimental studies here is that there would be deficits in doing fast reaching towards visual targets.
Do you know this, Guilherme?
Pardon, I didn't understand, George. Does he have what?
Does he have any motor, upper limb motor deficits?
Upper limb.
Yeah.
Oh yes, he had a difficulty to write. He had mostly a hand deficit.
Yeah.
This upper limb deficit, definitely. Yeah.
Because it's always
a lower limb deficit. He didn't have. He could walk. It was much more a brachial hemiparesis.
It's usually the case that if the primary motor areas are affected, as they probably are in this case, then trying to sort out what the premotor ones are doing is quite difficult. If there's a basic motor problem in the upper limb, then looking for things like apraxia, reaching problems, grasp and manipulation problems, over and above the motor deficit, which is resulting from his primary motor cortex involvement, is probably going to be quite tricky.
You are completely right. He had just this upper limb deficit and some dysarthria. Now, let me do a comment about what George said. We are very accustomed to see tumors that, infiltrating tumors that go from the precentral to the middle frontal and vice versa. But you can see here, this is a cystic lesion. The lesion is started only within the precentral gyrus. So it was really inside this compartment. I think that the precentral sulcus is displaced anteriorly, and we don't have at least neuroradiology, a real infiltration of the middle frontal gyrus in this case, I guess. So it's really confined to this, to the precentral gyrus compartment.
Thank you. So for the sake of argument, if I go back to Roger, if we say that the precentral gyrus was not affected, would you test a dorsal premotor cortex with reaching and grasping, which some people like Bello have used a screwdriver test? We tend to use the door handle test, the American door handle, when you have to grab and twist. And will this involve some parieto-frontal connections, Roger?
Yes, certainly. Yes, I think, particularly if it's if you're able to test that under patient vision in some way, either directly or through some sort of video arrangement, I think that's that's certainly shown up some very interesting features of these premotor areas that are quite separate to the sort of weakness that you would expect from a primary motor cortex involvement.
Thank you. Okay, so for the last minute, Guilherme, would you mind tell us the conclusion of this case?
Okay, so it's very interesting that you can see here during surgery, we just found a spot with him awake that we could get inside the precentral gyrus. You can see the beautiful U again, just anterior to this at the level of the Sylvian fissure. And if you go to next slides, you can see that we achieved a pretty good decrease of the cyst. We did many biopsies of the cystic wall, and very interesting that they didn't show any tumor at this point. And this patient got much better. Of course, it was just a pressure effect here mostly. Next slide, please. And but unfortunately, after a few months, he got worse again, despite steroids. There was an increase of a small nodular portion, and then he was sent to do radiosurgery with bevacizumab as a radioprotector, as the UVA people, University of Virginia, had published about.
Thank you.
He's doing fine so far.
And also a good result, Guilherme. Thank you very much for sharing this with us. Now we'll have a very quick break, only 5 minutes. So you can just grab a cup of coffee, stretch your legs, and I come back in exactly 5 minutes. Okay, we are back. So we're now moving to the second session of the day, and this is around mainly image-guided surgery, functional neuroimaging, tractography, fMRI and DTI, and language aspects of cognition. And for the first talk, I would like to introduce Alexandra Golby, who's professor of neurosurgery at Brigham and Women in Boston and professor of neurosurgery at Harvard Medical School. And also she's director of the image-guided neurosurgery. And among the neurosurgeons in the neurosurgical community has contributed as very few in the field of image-guided neurosurgery. She has published extensively on integration of functional data in tumor surgery. And what I ask Alex to do is to present to us how reliable language testing is in neurosurgical patients. We know fMRI could be has no more sensitivity than 65 or 70%, but we employ this routinely, and therefore we would like to see the pros and cons, the advantages and disadvantages of image-guided neurosurgery. So Alex, thank you very much for joining us today. We appreciate your contribution.
Thank you, George. And I'm really, really happy to be here. I wish we were all meeting in person, but this is as good as we can get this year. And I really enjoyed the session this morning. So I'm going to transition really now to some practical aspects of how we do this language mapping for our patients using non-invasive methods. So George, I cannot share my screen.
Your pardon.
It's disabled.
Just give me a second, Alex. I'm sorry about that.
Okay. Is it being shared now?
Yes.
Yeah, okay, great. So as I said, I'm going to transition now to some practical aspects of brain mapping as it can be applied to pre-surgical mapping for our patients. And building on what you heard this morning, which was just beautiful functional anatomy. Translating such complex issues and questions into practical paradigms is really a challenge. And we're all working to improve that over time. So this is definitely a work in progress, but also one which has tremendous practical implications as we plan surgery for our patients. So specifically, when we're doing this pre-surgical mapping, we are interested in the individual variations in patient anatomy. And that's critically important because we know that not only is there intra-person variation in the normal situation, but that is particularly accentuated in patients with lesions who may have distortion or reorganization of their language areas, and may also have lesion-induced deficits, which make mapping more complicated. So we'd like to really define pre-surgically the relationship between that particular patient's functional critical areas and the lesion to be removed, and to do so both for cortical areas and for white matter tracts. I'm going to focus on cortical areas, and I'm going to focus even more on functional MRI today, because you're going to have some talks coming up about both TMS and also diffusion MRI for white matter mapping. So to recap, as we approach our patient, such as the two cases that you saw just now, it's critical that we preserve the language function, and not only for language function per se, but also because of the impacts of language deficits on our quality of life, our social interactions, and even with mood. Moreover, we know that new deficits are actually not only associated with quality of life, but also with quantity of life. So patients who have new deficits tend to have shorter overall survival. And as you've heard again so eloquently today, there are so many language functions: speaking, understanding, reading, writing, internal dialogue. And these depend on this complex interplay of so many different functions. And we have to somehow implement this and operationalize it into some tasks for the mapping. And that really betrays the fact that language is not a monolithic thing. It's a multifactorial, multi-dimensional interplay of these different functions in a complex network. So what we'd like to do is speak about the different goals from the simplest to the most complex. So the simplest is language lateralization, and then for surgical planning in detail, language localization, and ideally, eventually towards this definition of the complete language network, its nodes, and to try to differentiate nodes which are essential for different functions versus nodes which are redundant or could be less essential and more easily compensated for. We use functional MRI as a non-invasive method, and this has several distinct advantages. One is, of course, it can be done preoperatively. The second is that it maps the entire brain, and so not just the area around the lesion, but other areas adjacent to it, or even in the other hemisphere, thereby allowing a more complete investigation into the networks, including cortical areas which may be recruited because of plasticity. fMRI also is very helpful because it's widely available and non-invasive. And there are a number of tasks which can be used. The tasks include receptive, expressive, and semantic tasks. The stimulation can be done auditorially or visually with things such as sentence completion or picture naming, as you heard earlier. And the tasks can also be active or passive. Vocalization of answers can be complicated because it can introduce movement or air artifacts. And that's one of the constraints of language mapping. And most tasks are done with a subvocal response. And then we'll talk a little bit at the end about resting state as a viable alternative for language mapping, which may be especially beneficial in patients who have existing deficits. So as I've mentioned, it's critical that the patient be able to perform the task. If the patient can't perform the task or doesn't understand the task or can't perform it with optimal timing, then the results won't be valid at all. And when considering how valid preoperative mapping results are, one has to consider the situation as it relates to how the patient is able to perform, how the acquisition was done, and how the analysis was done. And in particular, all of this has to get folded into an interpretation of the functional maps which are generated. I think it's very important to view the maps as sort of the start of the functional assessment of the patient rather than the end, because the activations that we see with fMRI have to be interpreted in the context of the patient's clinical situation, the quality of the imaging, quality of task performance, and the surgical goals. Other considerations include adapting the task potentially for patients who have deficits. That includes alterations for people with visual deficits. So we can do correction for patients who have nearsightedness or farsightedness, choice of auditory or visual stimuli, and even different language mapping, which I'll show you later. And then the selection of the baseline task is very important because fMRI looks for a difference in activation, and the higher the baseline, the less the cognitive subtraction will highlight, and therefore it will be more specific to a particular task, but less sensitive to the broader network. And these are some tasks that have been used. And in particular, I'll note that the Association for Functional Neuroradiology has some recommended tasks and some batteries available. In terms of language lateralization, this is a study recently that shows the difference between task-based and functional using a resting state, and demonstrates the overall finding, which is that the lateralization is more distinct with a task-based. So if you look in each of these little grids, the separation between left and right is greater along the horizontal axis, which is the task-based study, compared to in the resting state. And that's not a big surprise because the resting state is going to be demonstrating the entire network that's activated, including non-dominant aspects of language and understanding, and other aspects which are less lateralized. So at this point, I think it's fair to say that it's probably advantageous to include task-based fMRI, especially for lateralization. This is another recent study that was done in patients undergoing, I'm sorry, not in patients, at 82 centers which do epilepsy surgery. This was a survey, and I thought this is very, very interesting in terms of the adoption of these techniques for epilepsy surgery planning. So of 82 epilepsy sites, and these are high-level sites which have been accredited for epilepsy surgery, all of them use fMRI for surgical planning. And in terms of lateralization, about half have found that the fMRI never disagrees with other measures of laterality of language, but about a third reported at least one instance of disagreement with the WADA test in this regards. And usually these disagreements occur in patients who have atypical language lateralization, either non-dominant or right dominant or mixed dominance. And so the authors have proposed the following rubric for deciding whether fMRI can be used as the sole language lateralization or whether a WADA test can be required in some patients. And they note also that expertise in designing, administering, analyzing, and interpreting the data from the fMRI is critical. Moving on to localization, the reliability has been investigated from test to test in the same subject. So both short-term and long-term reliability, meaning in terms of a few weeks to a few months, has been demonstrated across multiple tasks and multiple areas. The the localization of the activity in terms of the centers of the activation is very reliable. But as you might anticipate, the extent of activation is more variable from study to study. And that's due, of course, to a number of things, both cognitive changes that the patients may have, as well as differences in the scanner and the acquisition, and of course, differences in how the task is thresholded. This is an example of how we can use the technology to address our individual patient needs. So this is a patient of mine who is a professor and trilingual, and we wanted to be able to map all of the separate languages which he spoke, German, Portuguese, and English. I think of note here is the finding that there is significant overlap in some areas, particularly in the inferior frontal areas, and in the superior temporal gyrus have been pushed down a little bit by the lesion, but there are also areas which are distinct for the different languages. Another interesting study is about task and threshold effects. So as I mentioned, the extent of activation varies significantly on the basis of threshold. That's the nature of the statistical threshold. In terms of reliability, that can be altered based on the threshold that's selected. Another conclusion from the study is to consider repeating the tasks and comparisons across tasks. In general, we have a finding across many studies, which is that reliability tends to be worse in patients. And this is not surprising because of the previously mentioned issues about task performance in patients. In addition to that, patients can have edema or other changes in perilesional areas, which can interfere with mapping. And also in general, motor mapping is more reliable than language mapping. Again, this is not a big surprise. The motor system is fundamentally simpler. The task tends to be more robust in terms of its activation and performance more easily verified. Moving on to quality control, as I mentioned, it's very important that the studies be interpreted with the clinical context in mind. This is an example from our own work where we see poor activations when we use a stringent threshold. This turns out to be related to poor task compliance. And if the threshold is lowered in an effort to be able to visualize any activations, that's when we tend to see false activations, false positives, such as this activation in the tumor itself. Another example of poor results comes from motion and potentially decoupling. So fMRI as a method which investigates localization on the basis of blood flow changes. And we know that around lesions, there can be neurovascular uncoupling, particularly around high-grade lesions. So all of that has to be considered, and a caveat put in the event of negative activations in areas where they would be expected, that it could be false positive due to such decoupling. Here's another example of a false positive in the region of the lesion. This false positive, you can see it extends across both tasks that were done, suggesting that there is some sort of vascular response in this contrast enhancing tissue, which is causing a false positive. And another type of false activation, false positive that we see is due to head motion artifact. Patients tend to have more head motion than healthy subjects, and you can see this edge effect where the head has moved in and out of or the brain has moved in and out of the region of interest in a way that correlates with task timing and results in these false activations. Putting this together, there is a number of studies that have demonstrated that fMRI can be used to guide surgical intraoperative mapping to help validate negative findings with intraoperative mapping, and in this case, also to increase the extent of resection overall compared to DCS alone. New approaches that are being done include resting state, and this is an example of comparison between cortical mapping, resting state, and task-based fMRI. This is motor mapping, and you can compare the areas that were found to have the responses on ECS, which have been translated into the imaging space with both the resting state and the task-based fMRI. And we have also been interested, as have others, in developing resting state for presurgical language mapping. This is an example of intra-run reliability between 2 runs of resting state, as well as compared to a task-based. And you can see the DICE scores, which are fairly good for the whole brain and considerably better within the language ROIs per se. Another approach which we are working on is movie watching. This is a type of task where the task performance is easier for patients who may have deficits. Task timing becomes less important, and patient cooperation seems to be improved. And one of the ways we see that is in terms of head motion. So this plot on the left demonstrates that we have significantly less head motion, especially on the outliers of who have more motion when they're doing the movie watching task compared to the antonym generation task. And we also have reports from the subjects that they have better attention and recollection for the movie compared to the conventional task-based fMRI. We've done this in healthy subjects, and then also in patients for individual subject mapping with good success. This is based on temporal correlations rather than spatial correlations. I'm not going to talk about TMS because you'll have a wonderful talk coming up on that, I believe tomorrow, but only to say that this is an alternative cortical mapping method with good sensitivity and specificity, and which can be combined with fMRI in order to improve individual subject mapping. So we find in general that the fMRI mapping has higher specificity and the TMS has higher sensitivity. And when these are combined, it improves the receiver operating curve performance versus DCS as the ground truth. Some future directions include development of novel acquisition, including ultra high field, so 70 acquisitions. Optimization and standardization of tasks and analysis, I think, is very important for us as a community to really try to be able to do multi-site studies, which will help us to further validate this approach. This is an example of our preliminary work using 7T in the same subject at different scanning sessions, comparing 3T and 7T. You can see that the 7T activations are more spatially constrained, tighter, with fewer extraneous activations. And this is due to the higher signal to noise, which allows us to threshold the maps more stringently, and also the smaller voxels and less spatial smoothing required for the analysis. And one of the most exciting areas is this approach to trying to map the entire language network. As I stated at the beginning, we know that language function is not a monolithic function. It's many functions which are interdependent. And I thought this was a particularly elegant approach from last year from the group at WashU, where they developed a language network model from 2,700 normal subjects, creating a 3D neural network analysis, and then trained the model and then applied it in patients with brain tumors, and were able to accurately and specifically localize the language network in these patients. And you can see the ROCs for both Broca's and Wernicke's area shown below. So that's really what I wanted to talk about with you today, to give a flavor of what we can do with non-invasive preoperative mapping. It's broadly used in clinical decision-making. There is still wide variation in implementation and use of this. And as a group, if we can come together with some standardized approaches and paradigms, I think that will be very beneficial, especially for more multi-center studies. I think one of the fundamental take-home messages for people who want to apply these techniques is that it requires great expertise in design, administration, and analysis, as well as especially in interpretation, taking into account the clinical context. And so having experts in the MR physics, experts in the cognitive science and design of the tasks, and then having the clinical radiologist and the neurosurgeon working together is really key. And I would say that in terms of George's first question about reliability and validity, it's like anything that we do. It's very dependent on knowledge and technique, and that hopefully with novel approaches and a lot of collaboration between these different areas of expertise, we will continually evolve and get better and better for our patients. So thank you very much for your attention. Again, I really appreciate the chance to be able to kind of open this translation to the clinical application of the beautiful work that you saw this morning. And I'm happy to take any questions.
Thank you very much, Alex. That was a wonderful presentation of fMRI in clinical neurosurgery. I also echo your invitation for collaboration, and one of the aims of this newly founded association is exactly this: to foster collaboration, to create a wider collaborations between specialists of different disciplines. And in the coming months, the association will try and set a framework where individual members and also faculty will be able to exchange ideas and set up networks and collaborations. So thanks very much for also introducing this concept. Clearly, you use oftentimes fMRI and awake craniotomies. Would you be confident enough to rely resection on fMRI for language without having the patient awake?
So I think it depends on a number of things. So really, like everything, it's about tailoring it to the patient and what their baseline is, what their disease has affected, and what they're able to do, what their performance is like, and then very much also what the lesion is like. So I think there's a huge difference between, say, a metastatic lesion that's in the immediate subcortical area versus an infiltrating glioma with diffuse margins. And I also appreciate very much that there are differences in people's trust in this type of non-invasive mapping. So I do all the fMRI for all of my colleagues at the Brigham, which is about between 120 and 150 exams per year. So it's a lot of exams. We look very carefully at whether the patient was able to perform the tasks. If the patient has poor performance, then I would say the maps have to be interpreted very with a lot of skepticism. But similarly, those patients who have poor performance on fMRI are the same ones who are likely to have trouble during the awake craniotomy testing. They're the ones who sort of very quickly tend to fall off on performance, even perhaps before starting surgery, just from the sedation. So it's a balance, I think. I am personally much more confident to proceed on the basis of the non-invasive maps alone. And I didn't even talk about the diffusion imaging tractography, which is the other side of it. I think another area for discussion is the integration or joint modeling of the white matter with the functional cortical anatomy. But I also have colleagues who are much less confident and who decide that they would prefer to have the awake mapping as a check or as a backup, or just to feel more confident in their decision making. So I hope that answers your question, George.
It's interesting that you are doing the fMRI as a neurosurgeon, which is a rather unusual paradigm, at least in the UK. But I suspect you have a better insight than the rest of us.
Yeah, so I'll tell you, for me, it was very much an evolution. And in fact, I really was brought on at the Brigham to do awake surgery, and I loved doing awake surgery. And then we started doing the fMRI, and we really were most interested in the results of the cortical and subcortical mapping to validate the non-invasive mapping. But what I found was that I was increasingly skeptical of some of the findings during surgery and how well they predicted eventual post-op recovery, which of course is really what we mean when we say what is the reliability. I don't think it's strictly proper to say that the reliability is whether it agrees with ECS or subcortical mapping, because we don't know how much those maps are in any given subject, in any given cortical or subcortical location, predictive of eventual outcome. And we also know that subjects can have temporary deficits, and we're very, very bad at predicting whether a deficit will be temporary in a matter of hours or days, sort of mid-level in the matter of 6 to 12 weeks, or permanent. And that's really one of the questions I'm hopeful that this group can start to answer, is how do we predict eventual clinical outcomes on the basis of all of these tests, both invasive and non-invasive mapping.
Thank you. Another task which is quite difficult to assess is reading. And Greg Hickok would like to make a comment on this. Craig.
Hi there. Yeah, this was just a comment about the last talk where the question was, how do you evaluate phonological level abilities with a tumor in and around the posterior sylvian fissure? I suggested, following Nina's suggestion about complex repetition, that multisyllabic non-words would work quite well. And then the point was raised in the chat that might be difficult in a noisy surgical suite. So I pointed out that we've done some work on the Philadelphia naming test, where we've analyzed it in the context of a large sample of people with aphasia, and we've been able to identify, using a multinomial processing tree model, the items which tend to tax phonological level abilities more than semantic, and vice versa, kind of like what we heard about using the Dell model, P weight versus S weight in a previous talk. So I'm happy to share that. So we've got some data, basically using item response theory, and we haven't formalized it yet. I'll put a couple of links to the foundational papers in the chat box, but it'll take a little extra work to pull out those items that might be used clinically. But I'm happy to do it.
If you want to share quickly your screen, Greg, you're very welcome.
Oh, it's okay. I don't have anything ready to go right now.
No worries.
I'll talk briefly about it tomorrow.
Thank you. So Alex,
can I finish by asking you one question, which is how you would use fMRI to inform your choice of intraoperative mapping technique and tasks?
Is that for me or for
whoever?
Greg, please go ahead.
Well, I think Alex, what you're doing is brilliant and probably more relevant to the kinds of stuff that we do with fMRI for that. I mean, there are some tasks like the listen and repeat tasks that we use to localize auditory motor networks, which work pretty well in individual subjects. So there are things like that might be added to a protocol, and they're relatively simple to do. And they're pretty good at identifying auditory motor networks, including this area we've identified called SPT, which seems to be underlying some of the problems in conduction aphasia and so on. So Yeah, but I mean, the methods that you're working with, Alex, seem exactly the right thing to do. I don't have much to add to that.
Mm-hmm. Good. And I would also agree with you, Alex, that the fMRI is very useful. And in my practice, I request this constantly. But as you may appreciate, the paradigms are rather limited, at least the ones that have been routinely used, like verb generation and verbal fluency. There are a lot of more cortical functions, like on SMA, where you want to test speech initiation, sentence completion, and many paradigms haven't been standardized. It would be ideal, in an ideal world, to use the same paradigms on fMRI and during mapping and surgery, but it turns like they're completely disjointed. So you have an MRI physicist or a neuroradiologist doing one set of tests, and then you take the patient to theater and you do a completely different set of tests. But it does provide us with a density map of language-rich territories, and therefore we can inform the patients that the chances of language damage are high or low, and we can plan the trajectory. But I would echo your view that it's a very useful tool, but personally I would rely more on direct electrical stimulation for the time being. Clearly, these techniques of probabilistic atlases of function are going to be amazing. So I look forward to more technology in neurosurgery.
So I think that the point about trying to standardize tasks across methods is really important. And we actually tried to implement the identical tasks in the OR as we were doing in the scanner. And really those should even be broadened to a more consistent pre and post-operative assessment of function. So I think we're also, as a group, we're at the cusp of being able to administer automated tests using technology like iPads, which we could give to our patients in a standard way, which would bring in the same sub-language domains that we want to test using whatever methods.
That's very interesting. So I hope to pick this up within the group in the coming weeks and months and all contribute to this wonderful effort. So thank you again so much for your contribution. We are moving on now to the next speaker, which is Juan Carlos. I introduced a little earlier today. And in addition to his amazing work on the anatomy, Juan has also established a virtual training laboratory, which becomes increasingly important, utilizing virtual reality to train future surgeons. And I see no reason why this could not be extrapolated to the wider neuroscience community to understand better anatomy and connectivity. Over to you, Juan.
Thanks, George. Yeah, first, let me say before I start that I have no conflict of interest relevant to this presentation, because I'm going to show work that is related to a certain company, but I do not have any conflict of interest. And I think that's important to state at the beginning. And I'm going to introduce today this concept that I'm excited about, although, of course, it's in its infancy and the accuracy is still, of course, an issue. But the concept of augmented reality fiber tractography. But first, I wanna show you how we get there. I showed you before my team in the laboratory that has really helped me putting many things together. And George asked me about talking about surgical and anatomical simulation. And this starts here. This is the laboratory we have at Stanford. We are grateful to have an excellent microscope and great equipment to simulate what we do in the operating room. And we have the same equipment as in the operating room. And this is the way to train and to prepare for surgery. And like the case we just discussed before, the best way to have the anatomy in your mind when you do this case is to have it done in the laboratory before, understand the fiber tracks, the vascular anatomy, the cortical anatomy, all the details you need to know for a case such as this one. And as we said, we do anatomical fiber dissection, but we also do computer-based fiber dissection. And one of the things we've been doing in the lab, thanks to Vera, is doing this photogrammetry. So we are doing the dissections, but also we are scanning this, or we are actually taking photography all around this specimen. And then through complex software reconstruction, you can create this 3D model that you can actually manipulate. You can zoom in, zoom out, you can label the structures. And I believe this is an important tool for teaching that can, deal with the low supply of anatomical specimens in different laboratory settings, and you can do this in your cell phone. So you can study anatomy now in an easier way with models such as these ones. It makes the specimens much more accessible for students and those interested in this field. We can even incorporate fiber tracks into these anatomical dissections. This fiber track is not from this brain, it's from a different brain, but you can actually overlap it. And this helps you understand, and maybe helps understand radiologists or people only doing fiber tracking, helps to understand how this matches within the brain and vice versa. And here we can do it bilaterally. You can see the arcuate tracks superimposed on both sides of this dissection, this brain that has been dissected. So this again is photogrammetry, and I believe it provides a unique tool for teaching and for education. But for computer-based simulation, as we said, and some people were asking in the chat, this is DSI Studio. And from my perspective, this is the best software there is for fiber tracking. I've been using it for over a decade, thanks to Frank for developing it. Frank and I, we worked together at the University of Pittsburgh. I'm hoping, Frank, I saw you were around before, so I hope you come to Stanford so we can keep working together. This is a phenomenal software because you can do your dissections for your case at your desktop. You could even do it in a stereoscopic mode. You can put your 3D goggles and your 3D TV and see it in 3D and be playing with this software. And to me, this is so important because it's a way for me to understand the regional anatomy of the case I'm going to operate. So for every case, I try to spend some time understanding where the fiber tracks around, or sometimes where the fiber tracks within. Sometimes you can do with this something you can do with this software. You can study, are there any fibers within the tumor, or all the relevant fibers have been displaced around the tumor, although they may be infiltrated, but for the most part, they have been displaced. So this you can answer with this great software. And we published about this, and we studied this extensively on how fiber tracking can really help you plan your surgery. This is a very large tumor in the supplementary motor area for the most part, but you can see all the fiber tracks that are around. And we like to do this before and after surgery to see the signature of our operation afterwards. And another example here, you can see how accurate these reconstructions can be. And you can do it with the FLAIR also, and you can contour the tumor and see the fiber tracks around. And this also helps defining the type of tasks you're going to do in intraop for these patients based on the tracks that you think that are being affected by the tumor. And for years, we were also thinking, can we translate this into the OR? Because all this is pre-surgical planning, which I think is very important. I believe is the most important part. But you also want to be able to have this information during the operation, and translate it into the navigation system. It has a number of limitations because most image guidance systems are closed. They are not open platforms. And this makes difficult integration of different modalities. This was an attempt, but you see the fiber tracks are not really, they're more like regions of interest. And this is very primitive. This is what we did, about a decade ago. But since I moved to Stanford, thanks to the efforts of Gary Steinberg, our previous chairman, he established this virtual reality laboratory, which we use for surgical planning, patient education, teaching of residents and fellows. And it is a phenomenal laboratory that is, equipped with these virtual reality platforms. And we use it for all types of surgery, vascular surgery, skull-based, but also for brain tumor. And I want to acknowledge also Diana, who prepares many of this work for us. So I'm going to highlight some of the things we can do for brain tumor surgery in the virtual reality laboratory. This is a recent case, 57-year-old patient with seizures. He has this, looks like a low-grade glioma in the supplementary motor pre-SMA area. We do fiber tracking. This is just DTI, not very good quality, but regardless, we can see the arcuate, SLF, and frontal aslant tract. You can see the frontal aslant tract, how is different, is displaced on the side of the tumor posteriorly. But we can do this in a more advanced reconstruction model. This is the tumor has been segmented in green, and then in blue you have the frontal ascent tract. And you can see the relationship of the tumor with the frontal ascent tract. You can see also the supralongitudinal fasciculus in orange. And then you can see in red the cingulum at the depth of the tumor. The beauty of these models is that this platform is open and allows integration of multiple modalities. You can put vessels in here. If you're interested in vascular anatomy, you can, of course, push the skull. If you're interested in the skull or skull-based anatomy, the gyri and the sulci can be seen very nicely. And I believe this helps preparing the operation better. And of course, you use a VR headset, so you are fully immersed. And suddenly, this becomes a very large field for studying, to the point that the quality is decreased because you can be fully immersed. You can actually walk into the brain and into the tumor. But I want to show you now how we can integrate this into the surgery. This is augmented reality. And this, I found this very, very interesting and fascinating, because I can now superimpose the fiber tracks into the operating field. You can see in blue, you see the frontal ascent tract. And I can stimulate, and when I'm stimulating with the Ogyman stimulator in the cortex, I can at the same time identify which fiber tract I'm stimulating. Before I had to bring the image guidance probe, and it was not so accurate. And in combining this, I have instant correlation of my mapping with the fiber tractography. And that's doing subcortical mapping. And as we do the subcortical mapping, this was the first case I did with this, so I was still learning. You can actually bring the fiber tractography, and you can identify the fiber tract that you're stimulating. Again, there's always been an issue on the correlation between electrical stimulation and the fiber tract that you're stimulating. And I believe this is going to be one way that we can solve this problem. Of course, we have to deal still with the accuracy of or image guidance and overlap. I'll cover that in a second. But you can see here at the end of the resection, and this patient got, interestingly, speech arrest or speech hesitation, I would say, where you see the blue track, which is the frontal ascent tract. You can see red is the depth where the cingulum is, and the cingulum has been preserved, and you see the blue track at the end of the resection in the posterior margin. So bringing this into the operating room, it really adds additional piece of information. And we are starting to learn how to use this efficiently. This is the patient post-op with a good resection. This other case, yes, I'm going to show this case just as a proof of concept. This patient has this temporal lobe tumor. It looks high grade, presents with seizures. This is our video of the reconstruction and the different tracks we're interested about. But here you see in green, this is the arcuate fasciculus, and the arcuate terminations on mostly in the ventral premotor cortex. And I can stimulate directly into the ventral premotor cortex. So I can see the positive response directly while I'm stimulating on the cortical termination of the arcuate fasciculus fibers at the frontal area, in particular the ventral premotor cortex. So I can tell, okay, this stimulates the arcuate fasciculus. It's a direct correlation of the mapping with the fiber tractography. Again, you need to rely on good alignment and registration and lack of shifting, as we know. But if you solve those problems, you can do direct structural correlation of the fiber tracks with your stimulation. And I found this fascinating myself, many years working on fiber tracking on one side, stimulation on the other, and now we can, merge both together during surgery. And I will finish with this case that we showed before. And this case is this supramarginal gyrus area tumor. We can overlap the tumor on the bone, on the cortical, on the bone, and then on the cortex too. And look at this, the alignment here. I'm going to stop this here for a second. The alignment here is Very nicely done, because you see, we learned that if we align the cortex based on the vasculature, we get a very nice alignment. You see how all the vessels really line up well with the sulci and gyri. To the point that, for example, I see this vessel right here that you see crossing here, that doesn't correlate with a sulci, with any sulci. And at this point, I was struggling to find out why this is happening, and is this a mistake? But what I found is actually this is a vessel coming off the sylvian fissure, so it's about 2 centimeters deep, but you're still seeing it. So you can see how this provides very valuable information for surgery. But at the same time, we have here the cortical termination of the fiber tracts. You know, this in green, I believe, is the arcuate, and this in sort of pink is the SLF, and this in yellow is the frontal ascending tract. So I have the area of those tracts in mind, and I can directly correlate my mapping with their location. You can slide down with your microscope. You zoom with the microscope. This is incorporating the eyepiece of the microscope. I should have said before, but I'm seeing this through the viewer of the microscope. And again, I can stimulate the cortex in the area where the tumor is, or in the area where the fibers are terminating. And I can say, well, the SLF termination has this deficit or has no deficit at all, etc. And then we proceed with our resection. When I'm doing these surgical resections, I always try to do this sort of anatomical resection. Is functionally based in the terms that you have functional boundaries, but the resection is also made anatomically based on anatomical boundaries on sulci and gyri, like Professor Ribas was explaining before. And of course, trying to preserve the vessels around. And again, here in this particular case, we had semantic paraphasia stimulating here at the supratemporal gyrus, but we don't have to go there. We had sensory stimulation anteriorly, where this patty is located. And now I'm working in the depth of the tumor, trying to reach the deep portion of the tumor. And this is always the most difficult part, because this is the part where we don't know if there is tumor left or not, but I can see as I'm removing this, which tracts I'm going to be affecting. And I keep resecting because I'm stimulating and the patient is still doing well and tolerating. And it's at this point when I stimulated, the patient started describing he was having an out-of-body experience, stimulated at the area of what I believe is the frontal ascending tract posteriorly. And I'm gonna show you a bit of the patient in a second, but this is just finishing our resection and looking at the overlap of the fiber tracts as we continue this resection. You see, there's a very good resection with no obvious flare signal left. And this is the pre and post of the fiber tracts. And this also brings another interesting point of debate. You see, this patient is missing part of the arcuate fascicle. Of course, it would be ideal to repeat the MRI maybe in 3 months, fiber tracking, to see if this is real or there is some artifact. But the arcuate fasciculus was likely involved by tumor. And the mapping suggests I could remove it because he has having no deficit at that level. So is that the right thing to do, or should we preserve tracts structurally, even there is no function, or should we do the opposite, remove them if there is no eloquent function? In this case, we did remove them because there was no obvious deficit when stimulating, but there are so many things we cannot map. So that's, of course, an open debate for many of us. So I'm going to show you the patients. This is with his authorization.
Was salvia.
Salvia divinorum?
Yeah, I dabbled with a lot of that last year.
So what he's saying is that he's comparing his experience during surgery with Salvia divinorum, which is a type of is a plant that has psychoactive properties and apparently has a very fast acting into the brain functioning, and creates this dissociation or out of body experiences that this patient was feeling. And there are papers describing how the temporal junction, we have this out of body experience when it's possible to have an out of body experience when we stimulate, given the multisensory integration of this location. I'm going to have the patient continue describing this feeling.
And what I'm comparing it to was salvia.
Salvia divinorum?
Yeah, I dabbled with a lot of that last year. And it felt a lot like that. I mentioned that I practiced out-of-body experiences, and it felt like it was taking me, like the reason why I take salvia is to induce, astral projection. So,
is that what you felt today when we were stimulating?
Yeah, it felt like salvia, definitely.
like an out-of-body experience?
Or very likely, yeah, it kind of did.
Like you were seeing yourself from the top,
No, I didn't see myself. I felt, no, I didn't see myself. I didn't see any like entities or anything. Nothing like, no hallucinations. Kind of felt, and I may have felt, you know, a presence, but like something I probably like, you know, something that's connected to me, and you know, an angel in a sense, you know, like probably on your guys' side, you know, I didn't, you know, like on your guys', probably watching you guys for me. So,
was it a pleasant experience or a horrific experience or neutral?
It was actually, it was pleasant. It was pleasant. Okay. It was pleasant. It felt, I mean, I know I was having trouble with speech, you know, trouble trying to like come up with like certain words that were on the pictures.
Mm-hmm. Good, no, you did fantastic.
Yeah,
you did very well. Good. So I shared this with you with his permission, because I think this was such an interesting experience of operating in this area, and the amazing things we can learn from our patients when we are in the operating room, and how we use those for our own learning and apply for other patients. So in summary, you can see how we can go from fiber dissection that we can transform into 3D models for better learning. We can integrate fiber tracking. We can incorporate virtual reality for surgical planning, but also augmented reality into the operating room to improve our surgical technique and performance. Thank you very much.
Thank you, Juan. That was a wonderful overview of technological advances. I have to agree with you that the modern neuronavigation system are very old-fashioned, and although we've been using them for a long time now, the evolution has been very minimal. And there's no doubt that the virtual reality, possibly in the form of HoloLens, that also Microsoft is using commercially, is going to be the future of navigated surgery. Looking at the screen, it's hard to tell. Do you get a depth perception of the fiber tracts?
Yeah, great question. No, you do not. That they have the problem is that the overlay is 2D still. Mm-hmm. And I already told them that I need to make it 3D because and also it's because of obviously the brain is a sphere, right? And it's convex, but this is flat. That's one problem. And then you can go deep in planes, but it's a 2D plane.
Mm-hmm. With regards to your question, if you can preserve the structure of functionally silent track, I think is a great topic, and I will leave this for the discussion at the end of the session. And I'm intrigued about the out-of-body experience, which I have no experience in terms of neuroscience. Does any of our faculty are aware of the neural basis of out-of-body experiences?
Can I just mention something, George?
Sure.
This was very interesting because I had read this paper in Nature of 2002, and I got the reference here, is the same author, blank, but are different other authors. I don't know if this is I think this is not the paper that Juan showed, but it's very interesting because the first time I read this was in Nature 2002, about the out-of-body experience exactly when stimulating the supramarginal gyrus. Mm-hmm. That's the paper you mentioned. It's the same author, but I think you mentioned another paper of this group.
It is a different one.
Nature?
No, it's a different one.
So probably they did update what they did in Nature 2002. Yes. And I was very impressed with this, and just want to ask you: was this a deep stimulation? Did you get in a single spot? Did you repeat this? You came back and forth, and you could do it again. And did you see any other mentions in this second paper? Does it refer to any other findings in this issue?
So for this patient, we repeated. We were able to elicit it twice, and it was at the depth. And depth posterior marginal resection. So I would correlate with the parietal ascent tract, but you cannot be certainly 100% sure. But it was posterior aspect of resection in the depth.
Very interesting. Very interesting.
So is this part of body perception? So do you think that this is related on how we perceive our bodies? Is this like the same network? Anybody has any experiences, any knowledge rather than experience?
Well, what I found fascinating is how the patient correlated this with a psychoactive experience he had before. You know, of course, I haven't tried that solvent, myself, but he said it's the same experience. And I know some of the researchers are doing investigations with psychoactive drugs to investigate brain function, and this is an open door for that too.
Great. Well, thank you very much, Juan. Let's move on to the next talk. And I would like to welcome Michelle Thibault-Deschautins, who I know for the last couple of years. Mish came to London and worked extensively with Catani and co-authored the book Atlas of Brain-Human Connections, which I have in my desk for many years now. And every time I have a case, I study this again and again. I try to burn these images into my brain. Michel is also the editor-in-chief of the journal Brain, Structure and Function. And after London, he moved to Sorbonne, and now he moved to Bordeaux, where he heads a large research laboratory. Mish, thank you very much for joining us.
Hello, thank you. So we're going to talk about interhemispheric compensation of functional deficits after brain damage. And what I'm going to try to convince you with is that this compensation comes from more or less lateralization of this function in the brain. If we speak about functional lateralization, often we think about the lateralization of language in the left hemisphere. And if we use fMRI to assess this lateralization, we can find a nice network that you can see in the brain on the top right that involve classical areas of language. If you compare the left with the right hemisphere in right-handed population, you'll see on the chart, which is with green bars, that most of the participants have more activation in the right hemisphere compared to the left hemisphere. And if you model it, you can see that you have a bimodal Gaussian with most of the people being extremely lateralized, some people being less lateralized, and then you have a smooth distribution of rare cases of people that are not lateralized. In the case of left-handed, you have the same distribution. There will be the red bars that you can see here. You have the same kind of bimodal Gaussian distribution of the lateralization of language, but you also have mirror organization of the distribution of language. So in sum, what I'm trying to show here is that there is a variability that exists in the lateralization of language areas. And according to this lateralization of language areas, a recovery after a brain damage or surgery might be different. Now, this is the surface of the brain, and we all know that this is not all the story. In order for this region to communicate, they need to use long wires of connections that are like the white matter connections. And typically, the language areas are connected by the arcuate fasciculus and adjacent short fibers that you can see here in green and yellow that we classically call anterior segment of the arcuate fasciculus and posterior segment of the arcuate fasciculus. If you start looking at the inter-individual variability in the lateralization of those connections, typically the red connection, which is the classical arcuate fasciculus, which is the one we call long segments that connect the Broca territory with the Wernicke territory, is in most of the population strongly left lateralized. There is barely any fibers of the arcuate fasciculus in the right hemisphere in these people. And the rest of the population, you have people that are modestly lateralized, 20%, and then you have 20% that are strongly symmetrical for this connection. And then you might ask yourself, if you were to have a surgery or brain damage in the left hemisphere that interrupts the arcuate fasciculus, which group would you like to be? Would you like to be the group that doesn't have an arcuate fasciculus in the right hemisphere, the group that has a little bit of fibers of the arcuate fasciculus in the right hemisphere, or the group that will have a symmetrical arcuate fasciculus? I think, like, honestly, if I had to cut my left arcuate fasciculus, I'd like to be the third group with asymmetrical organization. And this is a research I've been carried on by Stephanie Fockel in London. She's been studying painstakingly many stroke patients at the acute and the chronic stage of their stroke, and she selected the ones that were showing a strong aphasia after their stroke. She's been looking at the anatomy in the right healthy hemisphere that was not damaged by the stroke to look at the arcuate fasciculus. And she saw that according to the size of the arcuate fasciculus in the right hemisphere, the recovery after 6 months of language was better. In other words, the bigger your arcuate fasciculus in the right hemisphere, the more likely you will recover after an injury in the left hemisphere. The same concept kind of extend to spatial attention. You have this work from Gordon Schulman that is showing nice activation in the right hemisphere that are symmetrical for a detection paradigm. If I run you through the chart, typically empty square correspond to activation in the right hemisphere and full square activation in the left hemisphere. You can see that the empty square are always above the full square, showing this asymmetry in all the attention network. And we've been able to show that there is an asymmetry in the attention network, or the connection between these regions, which is a gradient going from the SLF1 to the SLF3, with the SLF2 in the middle, which is slightly lateralized. And if you start doing tests of attention, such as line bisection, you will find that there is a strong correlation between the rightward lateralization of the SLF2 and the performance in attention, the asymmetry in the performance in attention test. I've heard that earlier today, there was some confusion between the SLF1 and the cingulum fasciculus. So I just prepared a little video of dissection to clarify this point. So you can see here, this is a dissection based on open data that I put on our website. You can download up to 200 different data sets already processed that you can dissect if you like to learn about neuroanatomy. And here, so in TrackViz, I've been doing a dissection of the cingulum tract first. You can see it because it's below the cingulate sulcus here, changing a little bit the opacity. It's a one region of interest. And then you can see here appearing the cingulate tract with some of the some of the corpus callosum fibers, because that's just a one region of interest approach. And then above that, above the cingulate sulcus in purple, I draw the region of interest that correspond to the SLF1, so they don't contaminate each other. And you can see those wonderful frontoparietal fibers right above the cingulate tract, the cingulum. You can see that they are nicely separated. Again, those data are available if you want to play with it or disprove this point. I encourage you to download it and play with it. But the point being, is the lateralization related to recovery or not? And is this wonderful study, which is made a mistake from Uramova in neurology, has been able to show that the level of mean diffusivity and radial diffusivity in the healthy hemisphere in the frontoparietal connection was predictive of recovery of vision neglect in patients after a stroke, kind of like a similar study to the one of Stephanie Fockel, but for stroke recovery. So in other words, it seems like we are recovering better according to our degree of functional lateralization. So based on this observation, I was thinking, wait a minute, so, how many functions are functionally lateralized then? And can we model this systematically? And so I kind of ran into a difficulty into assessing which functions are really lateralized because we know a bit about language. We have made some progress in visuospatial. There is some theory about emotions, but then, it's kind of difficult to have a clear mapping of idea of the lateralization of functions. So I tried to take like a global perspective on this. And I used a website which is called Neurosynth that capitalize onto decades of functional neuroimaging. So they've been using all the coordinates that were reported in these studies to produce maps for several different functions. And by adjusting for structural asymmetries, we've been studying the asymmetry of every single function that were on this website, which is, after cleaning up, about more than 500. We checked whether this was legit by comparing this asymmetry of meta-analytic fMRI results with true assessment of functional MRI asymmetry during a task of finger tapping. And you can see here on the top that the regions that we report as asymmetric in meta-analytic fMRI analysis are the same regions that are reported as asymmetric by doing real fMRI with a participant doing finger tapping. So it is not stupid, it just, it works. It is not as rich as the true fMRI measurement, but at least the regions that we report are true asymmetrical regions. Then in order to understand the structure, we can plot all these different map of asymmetry into what we call a morphospace, where map of asymmetries that are similar to each other are close to each other, and maps of asymmetries that are different from each other are far apart. And this build several triangles that build that this 3D structure that is a tetrahedron. This tetrahedron that you can see simplified for you to be able to read it is mainly driven by 4 axes, an axis where you will plot all the functions that are related to symbolic communication in green, another axis of asymmetry where you plot all the functions that are related to emotion in pink, another axis which is related to perception action where like the function are symmetric there, and another axis in yellow which is related to decision making. Well, this is interesting to map it into a morphospace, but since we have the coordinate, we can literally simulate any of the maps that will correspond to any location in this morphospace. And this is what we attempted to do for the extreme of each of those axes, which we call the apices. Those are the archetype map of asymmetry for the symbolic communication. You can see on the left where you have classical asymmetry in favor to the left hemisphere in the language network. You have a perception action where you have the classical asymmetry in favor to the right hemisphere in the frontoparietal network, emotion where you have, of course, the mirror neuron, the face area, and in the medial part, the amygdala, and decision making, which is rightward asymmetry, functional asymmetry in favor of the frontal lobe mainly. These areas lateralize, and why don't they recover so well? But that's mainly because they isolate themselves when they lateralize. And we can see it here where we plotted together the level of functional lateralization of a different part of the brain with its likeliness to be connected by the corpus callosum. And you can see in red the connection of the lateralized area in the right hemisphere, and in blue, the connection of the lateralized area in the left hemisphere, which shows this very nice replication, whereby the least connected you are with the corpus callosum, the more likely you will be lateralized. Of course, this is based on all the functions that have been studied with fMRI, and we only have a limited knowledge of all the different functions we can study in the brain. And this is something we've been realizing when we started mapping systematically function on the white matter. You can see here on the top of the slide an attempt of a first atlas of the function of white matter connections. And at the bottom right in the C panel, you will see this difference of color between the left and the right hemisphere, because in the atlas, we show always functions that load the most onto the white matter, but this is just the top of the iceberg. Many functions load onto those same tracks. You can see that there are way more functions that can be decoded and explored in the left hemisphere, involving the left hemisphere when compared to the right hemisphere. So for instance, in the left, the red corresponds to 70 functions for those given tracks, while blue is barely 1. And this is not because, you know, the left hemisphere is the dominant hemisphere and does everything, and the right hemisphere is useless. It's just because we don't know and the right hemisphere doesn't speak. So my take-home message for this would be that the more a function is lateralized, the less it will recover after an injury. After all, it makes sense. If you destroy all the areas that are dedicated to this function, it is very much less likely to recover. This function mainly concerns the domain of symbolic communication, perception, action, emotion, and decision making. It will be interesting to discover a new function, but it's still a work that is to come. And this is probably, and this is just an hypothesis, because with evolution, functions that lateralize became isolated from the other hemisphere to speed up process. So they don't go through the corpus callosum to recruit the other hemisphere, because they'll have to go back and forth into this big brain, and then will be so slow to react. But this is at the expense of recovery after brain damage. And that's why when we try to model functional impairment in animals with smaller brains, they actually tend to recover better. And so we know the left hemisphere disproportionately more than the right hemisphere. So I can encourage you more to study the function of the right hemisphere, especially when you have the chance to be a neurosurgeon and look at it every day. Thank you very much. And I'd like also to thank the student that you can see here, Stephanie Fockel, Chris Foulon, and Slava Karolis, who have highlighted the work today. Thank you.
Thank you very much, Mish. That was a great contribution to our discussion. You managed to achieve a great mastery of the tractographic methods in a rather short period of time. And there are quite a few different software packages, and I think different people today and tomorrow you'll be using their own software. Do you have any advice on the students who wish to understand better tractography? Are there any initial steps they have to undertake, or any specific software you would recommend?
I'd say, like, all the softwares are good. The main issue that we're having now is the way we process the data. And so it's a school of belief. Some people will try to have as many streamlined as possible, to have everything connected with everything. Some other will be more conservative. I come from a school where we always compared our tractography to postmortem dissections, and every single track I've been reporting, I always saw it in postmortem dissections. And that's how we adjusted progressively our tractography. I don't want to really take part on is there a software better than another, because it's not the problem here. It's just like people need to adjust their algorithm and really painstakingly go through anatomical books to validate their tractography and their approach in the dissection. There is no other way than learning anatomy to do anatomy.
Thank you. Well, thank you very much for joining, and I hope to see you later.
Thank you.
So we are now moving to the next speaker, which is in the same theme of tractography. This is Anastasia Yendicki, who is an associate professor at Harvard, and also she's associated with the Athenula Martinos Center, a very advanced neuroimaging laboratory in Boston, and also she's an investigator at the Mass General Hospital. She has also developed her own software called Tracula, and I hope she can tell us a little bit more about this. But more importantly, one of the reasons I wanted Anastasia to talk to us was to understand better how these tracts appear on software packages, And what does this mean for us neurosurgeons? For example, I've heard many neurosurgeons say that the frontal tract does not exist, or other people say the SLF1 does not exist. And of course, we know that these tracts do exist. But I was hoping that Anastasia will explain to us what are the methods that can guarantee or at least maximize the possibility that what we see is actually a real anatomical tract and not some strange artifact. So thank you very much, Anastasia, for joining us.
Thank you very much for having me. I don't know if I will be able to give definitive answers to all of these burning questions in the next 20 minutes, but I can talk a little bit about our efforts in postmortem validation of tractography. And actually, I will not talk about the other part of our work with tracula, which is in developing methods for analyzing in vivo data. There's just not enough time in 20 minutes to go through all of these things. But I hope that you will find some of our early findings with these postmortem studies interesting. Okay. So as we now know very well, the method that we use to map fiber pathways in the brain in vivo is diffusion tractography. So first we collect a series of diffusion-weighted images. Each one of these scans tells us how much diffusion of water molecules there is in every location in the brain in a particular direction. And we collect this information from many different directions, and then we analyze these images to detect the preferential direction of diffusion at every voxel in the brain, which is understood to coincide with the main orientation of fibers in the same voxel. A tractography method then follows these vectors around the brain, connects the dots to reconstruct the white matter tracts. And the initial excitement about the diffusion tractography technique when it was first introduced had to do with these early studies that showed that tractography allowed us to delineate the main highways of the brain. Just two examples shown here are the fornix and the thalamic fibers, which up until then was only possible post-mortem. And it was understood early on that tractography did a decent job at delineating the core part of these big highways of the brain, as we expected to see them based on chemical tracing studies in non-human primates shown up top, or gross dissections in humans at the bottom. But it was also understood that in order to get that correct core part, we had to do a lot of manual editing to remove the parts that tractography got wrong. And so many of the early efforts in the field in reducing those errors in tractography focused on solving the so-called crossing fiber problem. So when we have just a single bundle of highly coherent fibers in a voxel, this is going to lead to a fairly simple diffusion pattern, which the first methods that were used for tractography fit a tensor to. So the tensor has a main axis, which is well aligned with the orientation of the fibers in this case. And so the early tractography methods simply followed that principal axis of the tensor. And when there's just a single bundle, like in this example, this works really well. But when there are two or more bundles in a voxel, then there is a more complicated diffusion pattern. And if we try to fit a tensor to that, the tensor is going to fit to the average signal, diffusion signal from all these bundles. And so it's not going to give us the correct orientation for any of them. So when a tractography method reaches this weird looking tensor, it will not know which way to proceed. So in the intervening 20 years or so, many methods were introduced for solving this crossing fiber problem. And these methods in general, instead of fitting a simple tensor, they try to fit what we'll generically refer to as an orientation distribution function, or ODF. And ODF has multiple peaks, and so it can fit more than one fiber population per voxel. And in parallel with the development of these analytical tools, something else that happened over the last 10 years is that the quality of the diffusion MRI data that we can collect routinely in vivo has greatly improved. And this is in big part thanks to efforts spearheaded by the Human Connectome Project. And this involved both building scanners with stronger gradients to allow a higher diffusion contrast to noise ratio, as well as accelerated sequences for acquiring the data with higher angular and spatial resolution in a shorter amount of scan time. And these new acquisition techniques were deployed for large-scale data collection, initially by the Human Connectome project and then by several other projects like the ABCD and the UK Biobank and then others. And so now we have a large amount of high quality data for both normative as well as disease populations. So has this helped tractography? It was shown early on that these newer acquisition techniques can sharpen the orientation distribution functions, and this is good for resolving crossing fibers. So we have much better quality data than we had 10 years ago, and we have great methods for resolving crossing fibers. Has this eliminated all the errors in tractography? Unfortunately, no. And let's see why. So as we get better signal to noise ratio and higher resolution in diffusion MRI, we can get very accurate orientation distribution functions. But the spatial resolution is still going to be in the order of a millimeter, a little bit bigger than that. And so there's going to be very frequently more than one axon population in each voxel. And there are going to be different configurations of multiple axon populations in a voxel that can give rise to very similar orientation distribution functions. This is an example of two fiber bundles, a blue and an orange one in a single voxel, 3 different configurations that can give a very similar looking ODF. So when a tractography method reaches this voxel, it can see that there are multiple bundles there, but it still doesn't know which way to turn. Typically, these algorithms are tuned to go as straight as possible, and this is tailored to solving the first kind of configuration, which is the crossing. However, that arbitrary choice is not always going to be the correct one. And this is a problem that will persist even if we can have perfect SNR and infinitesimal angular resolution. So this suggests that this is not a problem that will be solved by better data alone. The result of having to make this arbitrary decision about which ODF peak to follow, which turn to take at every voxel, is that sometimes we will get anatomically incorrect connections, even with very high quality data. This example is from a scan from our Connectome scanner collected with a maximum B-value of 10,000. So what happened here is that because of choosing the wrong ODF peak somewhere along the line, the tractography fibers that were following the corticospinal tract then jumped onto the corpus callosum and then jumped onto the CST on the other side. This is a very obvious example that you can simply eliminate. But then there are other examples where you cannot be so sure if this is a true or false connection. So just because we have gotten better data and just because we have methods to solve the crossing fiber problem, that hasn't solved all issues of tractography. In addition, by focusing on the crossing fiber problem, we are ignoring other fiber configurations that are also quite frequent in the brain. And this is where we can glean important insights from tracer injection studies in non-human primates. And we've been working on this with Suzanne Haber, whose lab has been doing these kinds of tracer studies in non-human primates for decades now. The example that I show here is from a tracer injection in the ventrolateral prefrontal cortex. The injection site is shown as a small white sphere. And we see that as the fibers leave the injection site, then they start branching into multiple sub-bundles. And Suzanne's lab has done many of these injections and has mapped how the fibers that leave different areas of the prefrontal cortex branch into multiple sub-bundles, and then each of these sub-bundles merges onto a different one of the big highways of the brain, like the corpus callosum, the internal external capsule, the cingulum bundle, etc. So these branching and merging configurations that are nothing like a crossing are actually quite frequent in the brain. So it would be interesting to compare tracing and diffusion MRI for this and for many other reasons. There have been a few studies that have done so in the past. And I'm showing them here arranged in terms of the quality of the diffusion data that they looked at in terms of spatial resolution as well as the maximum B value. So the vast majority of these studies had access to information only about the terminals of the injections and not the full trajectory of the axons. And as a result, when you have only the terminals, you can tell if tractography reached the wrong destination. But if it did, you can't tell where exactly along the path the error occurred and why. And in addition, many of these studies used injections from an existing database. And as a result, the injections and the diffusion MRI data came from different animals. In the small set of studies that used injections and diffusion MRI data from the same animals, they focused in injections in a well-characterized system of motor fibers. And so with Suzanne, we have been working over the last few years to collect a set of data where we have chemical tracer injections from Suzanne's lab and high-resolution post-mortem diffusion MRI scans collected at the Martino Center in the same brains. These include injections in many challenging areas of frontal cortex, and we have both high angular and spatial resolution by scanning the post-mortem brains over an entire weekend in a preclinical 4.7 Tesla scanner at the Martino Center. We've used a set of these data in the Iron Track Challenge, where we've had 15 teams of researchers from over 20 different institutions around the world compare their diffusion pipelines to see which one attained the best accuracy when compared to the ground truth tracer injection data. The second and last round of the challenge was completed in this past fall, and so we will be presenting the results in the upcoming ISMRM meeting. The main finding of the first round is that when you optimize your tractography pipeline for one seed area, that does not guarantee that you will also get high accuracy for another seed area. But there were a couple of teams that achieved good robustness in terms of accuracy when optimizing on one seed area and then testing on another seed area. In the first round, we followed up on this, and we found that there were some simple post-processing steps that these two teams did that anyone can adopt and improve both overall accuracy as well as robustness in terms of good accuracy between different seed areas. We found that there were several ODF reconstruction methods that people used and that exhibited similarly good accuracy. So there wasn't a super clear winner there. But where there was a clear winner was that probabilistic tractography methods outperformed deterministic tractography methods, regardless of the specifics of how the ODF reconstruction was done. So going back to the question of where do the errors occur, if the errors were mostly due to crossing fibers, because we expect to see crossing everywhere, crossing fibers everywhere along the trajectory of these fiber bundles, then the errors would be pretty much uniformly or kind of randomly distributed. But what we saw, and what I'm showing here on the right, is a histogram of the true positives among all the teams that competed in the first round of the Iron Track Challenge. We see that there are certain points along the trajectory of the bundles where we see a sharp drop in the number of teams that got a certain leg of the route correctly. And these areas where these sharp drops occur are exactly those areas of the branching points that we see in the tracer injection on the left. Suggesting that exactly because all of these methods are ODF-based methods that have been tailored to solving the crossing fiber problem, they do not necessarily do well in configurations of fibers that are not crossing, like branching or merging. So what can we do in humans where we cannot do tracer injections to further investigate these failure modes of tractography? We can use, not in the whole brain, unfortunately yet, but at least in small human samples, optical imaging techniques that rely on the birefringence, which is an optical property of anisotropic tissues. And in the brain, the birefringence is due to the regular pattern of arrays of lipids and proteins in myelin. We use a technique called polarization-sensitive optical coherence tomography, or PSOCT, which exploits polarization to probe birefringence. And it gives us direct measurements at microscopic resolution. These examples were collected at 3 micron in-plane resolution of the optic axis, which corresponds to the orientation angle of the fibers, as well as a map that's called retardance, which has a similar contrast to fractional anisotropy. And so we can use it for cross-model registration to diffusion MRI data collected in the same samples. A benefit of using PSOCT as opposed to histology or PLI, which is another optical technique that uses birefringence, is that with PSOCT, we do not first slice the tissue and then image it. PSOCT images the superficial layer of tissue, then cuts it, gets rid of it, images the next one, and so on. And so the slices are aligned to each other, and we do not have a very challenging registration problem to solve. We do not have to do any nonlinear registration. This shows an example of two samples from two adjacent samples from a human SLF that we imaged separately. The image of the hemisphere that they were taken from is shown in the center. And what you see on the right is the orientation maps from the PSOCT scans of these two samples. So the slices are well aligned, even not just within a sample, but even across samples that were imaged separately. We've done some extensive validation studies. You can see many of the results in this paper by Robert Jones that was published in Neuroimage last year, comparing the PSOCT to 9.4 Tesla postmortem diffusion MRI and looking at how various aspects of the acquisition and the processing affected the accuracy of the orientation angles. A couple of interesting findings, just to pick two, is that the very high B values do not seem to contribute substantially to the accuracy of diffusion MRI orientation estimates, suggesting that these ODF-based methods do not necessarily exploit all of the information that's included in the high B value data that we can collect either in a preclinical system or with something like the connectome scanner in humans. Another interesting finding was that the spatial resolution does play a big role. We saw a deterioration in accuracy of the orientation estimates as we went from 1 millimeter to 2 millimeter resolution. And this suggests that if you have a little bit of extra time to burn scanning your subjects, you should go for collecting smaller voxels. So going back to the issue that I talked about earlier with the tracer studies in the non-human primates, now looking at it with this different modality in human samples, how often and where do the errors of tractography occur? And specifically, as the errors often have to do with the tractography choosing the wrong turn by choosing the wrong ODF peak, we looked at the ground truth fiber trajectories that we can map at microscopic resolution from the PSOCT and compared to the trajectories that we get from diffusion MRI. So how often did tractography choose the wrong ODF peak to follow, leading to a different trajectory than what we see from the ground truth from the optical imaging? In this first set of preliminary results from 4 different samples, we found that that occurred about 20 to 40% of the time. And it was less frequent in samples that mainly contained the conventional sort of crossing fiber configuration, and more frequent in samples that also contained other kinds of configurations like branching or merging fiber bundles. So to conclude, we see that solving the crossing fiber problem will not solve all errors of tractography, and we need to start thinking beyond the crossing. There are very important insights to be gained on this issue from postmortem studies, both in human samples as well as in non-human primates. And it's important to get converging evidence from multiple different modalities because no postmortem imaging method is perfect. And in our next steps, we hope to use these postmortem data not only to evaluate the accuracy of existing tractography methods, but to engineer the next generation of methods. And I would like to finish by thanking my collaborators and thanking you for your attention.
Thank you very much, Anastasia. Great overview. Clearly, there's some difficulties with tractography like any other technique, and I'm sure that in the coming years, they will improve.
I am hopeful that the answer is yes.
Do you have any solution or when a track is close, so a white matter, a fiber is close to CSF or gray matter?
So that's another situation where the spatial resolution is key. And I know that the field has been focused very much on collecting more directions or collecting higher B values. And we have maybe neglected the spatial resolution, which sounds a little bit more boring. But actually, I think that the spatial resolution, based on what we're seeing from our validation studies, is very important. And for the kinds of problems that you describe of partial voluming with CSF is a great example of that.
Thank you. I think we have one question from Guilherme and one from Anthony.
Well, indeed, George, I would like to do this question originally to Michelle, but I think if Michelle is not around, Anastasia would be a nice person to ask the same thing. We read in some neuroanatomy textbooks like Gray's Anatomy, 36th edition, 1980, that regarding the sensory cortical areas, only the midline, the trunk cortical areas are connected by callosal fibers, and that the more peripheric, the limbic areas are not connected through callosal fibers. I would like to know if there was, if this was be seen also in tractography, and if Michel is still around, I'd like to know his opinion if this possibility has anything to do with more difficulty to recover from lesions.
So the validation studies that that we have done have been focused on frontal and prefrontal cortex. And there, to be honest, I do not know of examples of areas that are not connected through the corpus callosum. I'm not in the best position to answer questions about other areas of the brain, but we do have a new project with Suzanne Haber now to extend the tracer injection studies in other areas of the brain. So I'm happy to take requests for interesting areas with interesting problems like that.
Mm-hmm. Thank you. Anthony, you want to ask a question about the high B values?
Mm-hmm.
Oh yeah, this is just really quick. So you said high B values don't really contribute to improvement in tractography. So when people are making decisions about acquisitions, what do you find as a high B value?
So we were looking at, so the highest thing that can be done, at least presently in humans, is on the connectome scanner. So high would be 10,000. So we found that even when we used a single shell with fewer directions and a B value around, I think, like 2,000 or 3,000, we got very similar results. So this is what I mean by very high B values not contributing. I think that it depends very much on the task. So if you are doing a study where you're interested in looking at microstructural properties of known white matter bundles, and you're going to use a software that is going to constrain the tractography in some way, then you actually can get away with, just using 64 directions and a B value of 1,000, I believe. But when you're going to do seed-based tractography, where you are not going to constrain the solutions to, some known highways, that's when, doing something like the fancier with a 2,000 B value or higher would be useful. In addition, the higher B values and, having 2 or more B values are really critical for extracting different microstructural properties. Like doing things modeling that goes beyond the tensor, DKI or NAUTI or what have you.
Right.
So I would suggest that you choose the B values more based on the kind of microstructural properties that you want to extract than based on tractography.
I think it's saying up to 3,000 is still low is I think an important thing for especially clinically relevant acquisitions.
So no, I would not say that it's still low. So
well, you
like
were
I
saying
said,
extremely high would be like your 10,000 B value.
Right.
Yeah.
But again, if you just want to extract, the unsymmetric fasciculus, the cingulum, et cetera, because you will be constraining the tractography in some way, it will not be purely exploratory, then you'll be fine with just a B value of 1,000.
Great, thank you.
Jeremy, do you have a comment?
Yeah, I think that the lack of connections from sensory cortex was based on older studies, maybe using degeneration. Every cortical area has causal connections, including the motor cortex, distal extremity. So that's an older piece of information that's no longer accurate.
Right. So this.
What about the limbic areas, Dr. Jeremy? There's a paper on monkeys with Dr. Pandya that he does not point to limbic areas like cingulum and parahippocampal.
This lands up being a principle of connection to the cerebral cortex, that every cortical area has causal connections with almost, I don't know of no exception. Even the hippocampal commissures, every cortical area has a cord of information, a cord of fibers coming out of the cortex, that part of which goes to the opposite hemisphere and the other part goes down to the brainstem or diencephalon. So that's a principle of connectivity.
Thank you. We can discuss this a little bit later at the end of this session. Now we're moving on to another very interesting topic, which is how all these white matter tracts and cortical areas related to language are being developed. And language initiation has been found in pretty much every civilization by anthropologists and every distant tribe. So the inclination to speak is innate, and this is in contrast, for example, with the inclination to write. People don't intuitively want to write. They have to be taught, they have to be instructed, but that's not the case with language. So I'm very excited to introduce our next speaker, who has done very extensive work on how language develops in children. Patricia Kulf is a professor of speech and hearing services. He's a co-director at the Institute of Learning and Brain Sciences, and she's endowed chair of the Bezos Family Foundation. She wrote the chapter on language on the principles of neural science, which is a Bible for all neuroscientists, and she's given a number of TED Talks. So we're very delighted to welcome you, Pat. Thank you very much.
Well, thank you, George, and thank everyone who's preceded me this morning, because the images and the new technologies that are being applied are simply stunning. So I'm here today to share some information on the very young babies, typically developing babies, and we're interested in the entire phenomenon of language and trying to use the tools of modern neuroscience to functionally image the babies with MEG and to structurally analyze the brain to compare functional activation and structural information from infants, and then relate these multimodal imaging methods to an extensive array of very sophisticated behavioral measures that we have that project quite nicely how the next phases, when you look at these very early measures, they're quite sensitive to the next changes that are going to happen in the baby's development of language. So there's a particular assay that has been used behaviorally. I've used it over 30 years in many, many countries to examine a change that happens in phonetic perception. So phonetics are the consonants and vowels that make up words. And it turns out that all over the planet, no matter what country you're in, no matter what sounds you use, there's a very distinct change that happens in perception between 6 months and 12 months. And so here I'm showing a study in which we were testing Japanese infants in Tokyo and American infants in Seattle. And this is a behavioral head turn task. It's operant conditioning. The baby's trained to turn their head when they hear the sound change. And we're testing sounds from English that are not relevant to Japanese, the consonant syllables ra and la. So what we see here is a developmental change between when you test 6 to 8 month old babies and then test them again at 10 to 12 months. What you see is that the babies for whom the contrast between R and L is important to the language, those kids are going way up in their ability to distinguish those two sounds. Whereas babies coming from a country in which that is not a relevant sound are changing in the opposite direction. Their decrease in what for them is a non-native contrast, not important to the language. And we see this change all over the world. No matter what country the baby is growing up in, all the children are making this change in perception between 8 and 10 months of age. And it's rather surprising because it's before they're really producing much speech. If you've been around babies, you know that at 3 months they're cooing with vowel sounds, and at 7 months or so they start canonically babbling, ba 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 ba, that kind of thing. But they're not really producing words yet. This perceptual change happens before real speech production begins. So we can do this kind of assessment, this assay, either behaviorally or, and you can see that blue indicates this sort of sensitive period. You can also do this assay using neuroscience tools. Here's event-related potential studies that we published quite early, looking at the child's brain waves change, the mismatch negativity, or the MMN, change from a standard sound repeated as the background and a deviant. And what you see here is a big change, a big negativity when a new sound, the deviant, is presented. So if the baby's listening to ra, 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 and then you present la occasionally on 10 or 15% of the trials, you'll get this prominent deviant waveform, this negativity. So that's considered the MMN. So what you see in babies, again, all over the world, is that at 6 months of age, you'll see the deviant and standard looking identical for native sounds and non-native sounds. But then by 11 months, you will see that only the native sounds evoke this kind of response, and that the baby's auditory system is not responding to the non-native sounds. So what's interesting about this change that occurs between 8 and 10 months of age is that it predicts future language out to, we've had many studies show it predicts out to 30 months, vocabulary acquisition, sentence production, mean length of utterance. And more recently, we can trace the effects of this initial measure of learning all the way to 5 years of age, and in a study just about to be published to 6 years of age, predicting syntax at 6 years of age. So, and interestingly, we can also show that the skill at native language perception predicts a positive trajectory of language growth, but a child who remains really good at non-native perception in the absence of being exposed to that language, it's a negative predictor. So the stronger a baby at 11 months responds to a non-native sound that's not occurring in the environment, the slower that child's language develops. So there's attentional mechanisms at work here in which babies are attending, babies who are listening to ambient language speakers and attending to the relevant changes are going faster towards language. So of course, as a cognitive neuroscience, we ask what's happening during that sensitive period. And there's two things that are happening that I want to tell you before I get to the brain science. One is statistical, and the second is social. We've known for a long time that babies are kind of what I like to say, taking statistics as they listen to us talk. They're quite sensitive to distributional frequency. They're sensitive to adjacent probabilities between syllables that help them detect words. They're quite good at this, even in very, very early in development, picking up the statistical properties of ambient speech. But a more exciting development, more recent, is to show that babies are taking statistics in the early period only in the presence of a social interacting human being. And I know this because we have done studies in which we, here's the background to we expose babies to new languages at the critical period and see what they can learn in 6 to 12 sessions over a month's period. So here's the backdrop. When we were in Taipei, and comparing the Taipei babies to American kids growing up in Seattle. In this study, we're using a Chinese contrast that Americans do not respond to. We don't find it easy to detect that contrast. And the Taiwanese babies, we see the same change. The Taiwanese babies are getting better. The American babies are getting worse. And what we did is expose the American kids to an intervention, 12 sessions, 25 minutes, 4 different speakers. It was as though they had Mandarin relatives come visit and speak to them for 12 different sessions over a month's period of time. And here's what these sessions looked like during that period.
Jasper, 早安 Finn。嗨，我们要来讲一个故事哦。今天要讲的故事是《小熊七七探险记》。来看看发生了什么事情哦。小熊七七在这里，一个绿色的圈圈，绿色的圈圈，绿色的圈圈，再转一下，转一圈，嘿，一圈，再一次哦，转一二三。
Okay, so what did we do to those children's brains as we exposed them to this new language? Here's that background study result again with the Taiwanese babies and the American babies that are just growing up monolingually. And a control group exposed to English shows no change in the Mandarin detection, but those kids who had the 12 sessions of social exposure to Mandarin were statistically equivalent to the Taiwanese kids. So the next study relevant to our discussion here is whether or not that same learning over this 12-session period during the sensitive learning interval can be achieved with a videotape. And the answer to that is no. When we exposed babies to audio alone, that was another control condition, no learning whatsoever. When we exposed them to a TV video version of the very same materials, same dosage, same everything, absolutely no learning took place. So we have a case here in which the social exposure seems to be critical. The other thing our studies were revealing at the same time is that when you put a person in front of a baby talking, what you tend to evoke are these conversational turns. It seems the presence of another human being, as opposed to a television set, encourages the rate of these conversational turns, not as though the kids are coming back at you with sentences, but they're verbalizing in ways that can be described as speech-like. They're not creating other non-speech sounds, but their face-to-face produces a kind of impetus for conversational turns. And so we started imaging using MEG because we wanted to see what areas are activated as children listen to speech versus non-speech. We were the first in the world to put little ones in an MEG machine. The nice thing about it is that it's extremely quiet. The machine is on, babies listening through insert earphones to the sounds of different languages. We're keeping the baby entertained and simply trying to record what's happening in the brain and which areas are activated for native and non-native at these different times in development. And what we see is a very interesting interaction with 2 areas of the brain, superior temporal and inferior frontal. In the youngest babies at 6 months, it doesn't matter if they're listening to English or Spanish, you get equivalent activation in ST and IF for both languages. But at 12 months, when kids have made this change towards their native language, they look just like adults in that auditory areas begin to peak for native language sounds, and IF areas, inferior frontal motor planning areas, begin to peak for non-native signals. And we're arguing that right from the beginning, babies' motor planning areas are contributing to auditory encoding. And perhaps as they're learning to produce speech by 12 months, it becomes difficult to kind of simulate what in the world you would do with your motor system to produce that sound that you're hearing. These are kind of speculative interpretations, but there's a lot of data now indicating that the motor system is highly active as we listen to speech and not to non-speech. So we have our own studies, an early one when we were first testing the feasibility of MEG that showed this activation in prefrontal to anything speech-like and not showing it, inferior frontal, not showing it to non-speech. We now have unpublished data by doctoral student Maggie Clark showing that this activation of the motor planning areas, inferior frontal, is there as early as 2 months and does not occur for non-speech. So the interesting thing about 2 months is it's prior to baby's actual ability to produce vowel-like speech utterances. That is a very specific change that happens at around 12 weeks of age. So these data from MEG brought us to think very closely about the interaction between the sensory and motor system, the sensory motor loop, and its contribution to what was originally considered a purely sensory change in perception. And these studies, of course, all brought us to MRI and structural imaging. We wanted to look at specifically at the arcuate, but in general, we wanted to look at the change from 7 to 11 months. We were working with, our group has an MRI physicist, Vasily Yarnik, and he has produced this MPF measure, and he has used it to identify and estimate myelin. And I'm not going to go into detail, but point you to a publication in which the histology has shown pretty clearly a strong correlation between these MPF methods and actual myelin in mouse models. So we're using this MPF method both in adolescents and in babies. And again, I won't spend time on it, but here's a publication that occurred just this year where we're looking at children in late childhood and adolescence between the ages of 9 and 17. And you see the MPF mapping templates on the left, and then the right side shows pretty much our significant findings that when you look at both MPF in gray and white matter, it's the gray matter in red that's making the change in this transition from 7 to 17. And the white matter, myelin is not changing much at all. Just a few other facts about it. It's changing faster in males than in females, whether you're using chronological age or something that assesses puberty, and it's bigger in the parietal lobes than any place else. But what we're trying to do now is to use the MPF methods to look at quite specific pathways in baby brain to understand the power of conversational turns and the linkage between sensory and motor areas. So we have a study in which we had coached parents with regard to the importance of language input. We have many studies now indicating that the amount, the quantity, but particularly the quality of language input to infants changes the trajectory of everything that we can measure in the brain with regard to language and in behavior as well. So there's a particular style of speaking to babies. You've probably read about it. It's called motherese or parentese, and it has a much higher pitch. If I'm speaking to you in my typical voice, 200 hertz, 200 to 300 hertz. But if I had a 2-month-old baby, I would say, hello, how are you? So there's a huge increase in pitch, very exaggerated intonation contours. And if you give choices to babies between these signals with the high intonation change and the high pitch overall, in any language, they'll prefer to listen to this parentese speech over adult-directed speech. But we're interested in the extent to which this kind of input changes the arcuate and changes the conversational turns and the relationship between them. So here again, methods in which we're starting with a whole brain fiber connectome using AFQ to extract fiber bundles that pass through specific anatomical locations, labeling the resulting tracks, in using ROI-based segmentations, and then taking 100 evenly spaced locations along the tract and calculating either MPF or using naughty methods, neurite density measures. And this is the work of postdoc Libby Huber. And so with 20 kids analyzed from our parent coaching study, so our parent coaching study was a randomized control in which half of the parents were given coaching about the importance of language input to children, and half of them were measured in the same way, but parents were given no coaching. And what we saw is parents increased greatly their frequency of talking to their kids. They use parentese more, they socially engage with the children more, and conversational turns go way up. and language outcomes at 18, 24, 27, 30 months, much higher in the coached group. So here we have, and here's this, there's only 20 kids finished, and so I haven't indicated which are control and experimental kids, but we're plotting on the left 14-month conversational turns and our estimates of myelin density with MPF on the x-axis. And so you can see in the images on the right that we see very nice correlations between the conversational turns that we record. We're recording kids at home every 2 months using little miniature excellent microphones that they wear in their vest pockets. And so we can calculate with quite precise methods how many words, who's talking to the infant, and the turn taking that's going on. So we're pretty excited about these measurements. The study's not done, but it looks very interesting with regard to the MPF and the relationship to conversational turns. And we have a similar interesting correlation. Again, it's the conversational turns, this time at 10 months of age. You see in the left arcuate and the right arcuate, the left SLF and the right SLF, correlations between the number of recorded conversational turns and neurite density. So this is encouraging, it's work in progress, but with the goal to look at the relationship between behavior, structural measures, and functional measures. And in the next round of studies that I'll describe, we're even looking at certain candidate genes that relate to measures of attention. So, but I won't present those today. So here's a second study in which we're bracketing that interesting period, acquiring MPF data at 7 and 11 months of age. Here we have an N of 26, and we were able to collect longitudinal data for 17 of the subjects. And so we have the MPF mapping for those 17 subjects. And then we're looking at language outcomes using, these are very standardized measures in language acquisition research to estimate the development of vocabulary, the complexity of language production in terms of sentences and syntax, and the mean length of utterance. And here we can see the whole brain MPF mapping templates in the 7-monther and the 11-monther. And we're pleased to see how clear they are. And this is the work of research scientist Neva Corrigan in the lab. And here we see the relationship between the MPF results going from right to left brain with sentence complexity at 24 months. So what's interesting here is that in prior measures where we had for the last 5 years, attempted myelin measures, they're difficult, segmentation, everything is difficult with regard to these measures of baby brains. We hadn't seen interesting correlations, and now with this new MPF method, they're everywhere. So it isn't just sentence complexity, it isn't just at 24 months. We begin to see correlations at 18 months, 18, 24, 27, 30 months, and they're quite extensive. And without a deep analysis of where we're seeing them, they're appropriately different for tasks related to language as opposed to executive function tasks in which we're looking at metrics that relate to a baby's ability to switch attention, to do the standard executive function tasks where baby's attending to one set of things with regard to a rule that predicts which stimulus will be active, and then you switch the rules and babies have to switch attention. This switching of attention is a kind of cardinal feature of executive function. And again, we see beautiful correlations in appropriate areas between the MPF measure and our measures of executive function. Now, these are all at 7 months. It's also very interesting to just comment that no correlations were obtained at 11 months. So this is suggesting that these structural changes are preceding the activity that we see functionally. So our myelin measures at 7 months are now predicting our functional activation results at 11 months. And the myelin measures are at 7 months are stronger predictors of eventual language outcome than even our functional measures at 7 and 11 months of age. So I think the bottom line here, I mean, this is work in progress. We're not done yet, but I think that the attempt, while it's difficult to relate multimodal measures of the baby brain with behavior so that we're grounded in what it is that we're trying to explain, the perceptual change, the change in production, the sensory motor coordinations, the baby's ability to take conversational turns and also to imitate, which comes online right at the end of the first year. Babies are busy imitating language in the form of words and longer utterances from the standpoint of its prosodic characteristics. So I think the bottom line here is we're very excited about the combination of methods. And we're also introducing, I said briefly, certain genetic probes. We're very interested in the COMT gene because we'd seen in studies of college students learning a new language for the first time, if you're doing diffusion measures in the arcuate and SLF, while college freshmen are bathed in English in intensive 3 to 5 hour a day social exposure for 3 weeks, and we're imaging the growth using FA measures from DTI, you can see that there's an interaction between variants of the COMT gene and the growth that you see in these tracks as kids are socially exposed to language, even as college students when they're past this so-called critical period for language. So our excitement is to continue that mapping of brain and behavior, to continue to pay attention to what all of you are producing with regard to the most modern methods, and eventually to creating gorgeous visualizations such as you have shown us today. So with that, I'll stop sharing and see if there's any time for questions.
Thank you very much, Pat. That was an extraordinary demonstration of the developing nervous system. Do you happen to have any data on the plasticity of these tracks when there is an injury, for example, stroke?
We don't, but that would be fascinating. It would be amazing. So our goal is to map this out in typically developing infants and in bilingual infants. So we have equivalent studies going on in bilingual infants, and then to examine children with known disorders that involve particular kinds of disorders. So for example, in autism, we would predict based on what we know about autism, the children with autism would probably not have the sensory motor connectivity that we're seeing in typical babies. I think it would be very interesting to look at activation in inferior frontal using MEG. It would be very interesting to look at the arcuate development with language input with children with autism, because there's something about that sensory motor connectivity that's probably, it could be not there. And so we're interested in that population. We're very interested in dyslexia because many of our metrics, these metrics, brain metrics, are very sensitive to, even in the early period at 6 months of age, you can see differences between typically developing children with whose families have no history of dyslexia compared to children who at least one parent of the child has been diagnosed with dyslexia. So we think that these measures will not only be theoretically informative, but really sensitive potential biomarkers of a risk for disease. And of course, in kids with stroke, in an earlier life, when as a speech pathologist, I would deal with children who had been in accidents. So I remember quite vividly a 2-year-old who'd been injured in a car accident with a huge amount of damage in the left hemisphere, and watched her, this was all behavioral at the time, watched her recover language and the use of her limbs on the other side of her body, presumably using the right hemisphere. a left-handed child became a right-handed child. So these measures, I think, are going to be the kinds of things that we'll use clinically and use from the standpoint of early biomarkers that would allow you really early in development to take measures that could help change the trajectory of a child's life with autism or potentially with dyslexia.
Thank you. There's also one question. There are many questions, and if I please take this opportunity to ask the panelists to answer these questions online. One more question for you, Pat, is from Stephen Brem, who's asking, how do you achieve a connectomy in a 26-month-old child? Do they have any sedation? And is this structural or a functional connectome? And if you have the 3 questions in one, and if there are any specific scores of white matter integrity for analysis of the arcuate fasciculus.
Yeah, okay, these are all complicated. The first one is that no, we don't sedate any of the children. We're scanning between 9 p.m. and 2 a.m. We have children fall asleep. We send home what we call the MRI lullaby. It's the sound of the scanner. And mothers expose their babies to it ahead of time so that they're used to it, so that they don't wake up. And we attempt to keep it as quiet as possible and as short as possible. So we're doing, I am doing some functional resting state stuff, but this is all MRI. This is structural MRI. And we try to keep the scan 7 to 9 minutes and do whatever we can to keep the babies asleep. But yes, you have to stay up at ridiculous hours to get these scans. And then for details of the tractography and the way we're segmenting, I mean, maybe David will mention some of the difficulties in segmenting the baby brain in his talk, but I'll refer you, I can send you the papers that go into detail about how we're doing it. It is not simple. And yes, we're going to need, I think we're going to need more sophisticated measures to be really confident of our separation of gray and white matter and our segmentation.
Thank you so much. Now we're going to make a small swap in the order. Next will be Evelyn, and then will be David Van Essen. One of the issues I would like to discuss are around the Broca area, and many surgeons believe that the Broca area as defined does not exist. Other people believe in the Broca area very religiously, and others believe that we have more than one Broca's areas. Evelina Fudorenko is a scientist associate professor at the Department of Brain and Cognitive Sciences at the MIT. And some of her research interests include language comprehension, language production, executive functions, cognition, meaning, and plasticity. And she will be addressing the Broca area dilemma. Thank you very much for joining us, Ev.
My pleasure. Can you hear me, guys, okay?
Yeah.
Yeah, okay, great. And thanks, David, for switching with me. Just I have a 3-year-old who needs attention. All right, so today I'm going to tell you about a line of research that I have now pursued for over 10 years, aimed at understanding how language relates to other cognitive functions, questions that I think are at the core of many debates about human cognition and its origins. And here are the folks that have worked on these questions with me, and I especially want to highlight Edan Blanc, who led big chunks of this work and now runs his lab at UCLA. So as we all know, over a century and a half ago, the French physician Paul Broca reported a case of a patient who suffered from a selective deficit in speech articulation and who post-mortem exhibited a lesion in the posterior inferior frontal gyrus. Broca provided a detailed account of the patient's perceptual, motor, and cognitive capacities and was very careful to highlight its selective nature. So here's an excerpt from the original report. It is only the faculty of articulating words that these patients lack. They hear and comprehend all that one says to them. They all have their intelligence. They emit vocal sounds with ease. They execute with their tongue and their lips movements much more extensive and energetic than those required for the articulation of sounds. And yet the perfectly sensible response that they would want to make is reduced to a very small number of articulated sounds, always the same and always performed in the same manner. Over the years, Broca's area continued to be treated as a key node in the network of brain regions that support articulation. However, in the last several decades, especially since fMRI became a widely used brain imaging tool, this area has also been implicated in a myriad of cognitive functions, going way beyond articulation and even beyond language, including general sequencing abilities. This is just a handful of papers that made claims about what Broca's area does. So like I said, including general sequencing, music perception, tool use, inhibitory control, and so on. Across these numerous studies, two primary hypotheses have been advanced. One is that Broca's area supports combinatorial processing in language or across domains, so combining simpler representations into more complex hierarchically structured ones. And another is that it supports some aspects of executive functions like working memory or inhibitory control. And the term Broca's area is still used by a lot of neuroscientists and clinicians, but I'm going to argue that the very construct of Broca's area and its macroanatomical subregions like pars opercularis or pars triangularis, which roughly correspond to estimates of Brodmann areas 44 and 45, is highly problematic and has led to a lot of empirical confusion and misguided theorizing. So I will briefly talk about the heterogeneity and variability that characterizes the left frontal lobe, which houses Broca's area. I'm going to show you that Broca's area as defined in the literature contains subsets of at least two functionally distinct networks, a language selective network and a domain general network, both of which are distinct from the articulation component that I just introduced to you. And then I'm going to make an argument for moving away from traditional group averaging analyses and toward individual level ones. Okay, so Broca's area is traditionally defined as the triangular and opercular portions of the inferior frontal gyrus. Now, this is a lot of brain we're talking about. This is a huge chunk of cortex, many cubic centimeters of cortex. Moreover, we all know this area is not structurally homogeneous. So this is, of course, the classic drawing based on Brodmann's staining techniques, identifying two sub-areas, BA44 and BA45. And then later work from many different groups suggest that even within these site architectonic areas, there are subregions at a finer grain where cells differ in their cell properties, degree of myelination, number of receptors for different neurotransmitters, and so on and so forth. Now, here's where the problem arises. One, we cannot see microstructure with the tools that are currently available to us. And two, critically, the locations of these microstructural areas vary a lot across individuals with respect to macroanatomy. So particular microstructural areas, which I think could be reasonably assumed to correspond to functional difference in some way, may land on one side of a gyrus in one individual, on another side in another individual. And again, this is not news if you read Brodmann's original descriptions. He talks about that kind of variability extensively. Now, these properties of the brain's architecture have an important consequence. And that is, when in the traditional group averaging approach, when you take a bunch of activation maps and put them together in a common space, any given point in that space, any given voxel now becomes completely meaningless. You're averaging away all the fine-grained structure that is known to exist within this patch of cortex. Let me illustrate. So here I'm showing you three individual subjects. In red are activations for a language task. In this case, it's a contrast between processing sentences and sequences of non-words like Blicket or Florp. And in blue are activations for an executive function task, a spatial memory task, a contrast between a harder and an easier condition. And you can see how variable the precise locations of these activated sets of areas are, in spite of the broadly similar topography. Importantly, within individuals, these activations are remarkably stable, not just within a scanning session over a course of runs, but over time, over spanning many years. So if I bring you today and I scan you on these paradigms and I scan you 10 years later, that architecture will be there and will be remarkably replicable. Now, to drive this point home a little bit more, in different shapes here, we're highlighting areas that correspond to the same voxel in a common brain space, but correspond to different functional areas across participants. So if you focus I don't have my mouse here. If you focus on this little square and trace it across these three brains, you can see that in some subjects, it corresponds to a language region. In other subjects, it corresponds to a region that responds to working memory demands. And this kind of variability can lead to serious issues in group analysis, like you may miss activations because of insufficient voxel level overlap, and you can blur together nearby distinct areas. So a solution I would like to argue for may already be apparent to you. So one point is just do not average brains together. And secondly, although it's really nice to be able to do staining of neural tissue, there is a big advantage to living brains, and that is they work and you can probe function in them. And so the solution I've been advocating for a bunch of years is to basically use function to identify meaningful components of the brain and mind correspondingly, in this case, meaningful components of Broca's area, for example. And then across studies and labs, we can carefully characterize the responses of these regions in an effort to actually understand what computations they perform. Okay, so let me tell you about two networks whose subsets are adjacent to each other within Broca's area. So we have long known that there's a set of brain regions in the left hemisphere that respond robustly when we understand linguistic signals, both in auditory and visual modalities, and when we generate linguistic utterances. These regions that I often refer to as the high-level language regions are distinct from both lower-level speech perceptual areas in the superior temporal cortex and from their motor articulation regions, one of which resides also within Broca's area, as I already mentioned. Unlike those lower level regions that support speech perception and speech articulation, these regions are deeply sensitive to the meaningfulness of the signal. So they respond a lot more strongly when the input is interpretable compared to when it's perceptually similar, but you can't extract meaningful information from it. And similarly in production, they respond more when you produce meaningful sentences compared to if you're just saying a sequence of syllables or something like that. And so these regions, as I already foreshadowed, you can find very easily by contrasting responses, for example, to a linguistic stimulus, like reading or listening to sentences, versus a perceptually matched control, like sequences of non-words or listening to speech in an unfamiliar language. And here again, some sample activations in 4 individual participants. These activations are incredibly robust to variation in the particular materials you use, in the modality of presentation, in what kind of task may or may not accompany the stimuli. And in bilingual and multilingual individuals, they're robust to which language you're probing. More evidence that this network constitutes what I would call a natural kind in the brain comes from work using resting state functional correlation. So this is a study from Rod Braga. Here I'm showing you again 3 individual brains. In a black outline on each brain, you can see a network that emerges from patterns of spontaneous fluctuations during resting state. And it generalizes across other kinds of naturalistic paradigms, but basically the subject is, there's no contrast, there's just how different parts of the brain go up and down during kind of rich mental states. And in different colors in yellow and orange, you see activations for a contrast that my group has been using for over 10 years between sentences and non-words. And there's an amazingly tight correspondence between these two, suggesting again that this network is there. It's part of our brain's architecture. And the contrast that I mentioned is just a way to quickly pull it out of the brain so we can study it. These regions are also highly selective for language processing over many, many non-linguistic tasks, a point that I'll revisit in a couple of slides. And they appear to support both the processing of word meanings and combinatorial semantic and syntactic processing. Again, I'll say a little bit more about this a little bit later. And of course, we've long known that these regions are causally important in that if you damage parts of the system in adulthood, you end up with deficits in comprehension and or production of language. Now, Another system of interest is a bilateral system known as the multiple demand, or MD network. And it's known in the literature by many names. So when people talk about the task positive or the executive control network, that's the same network. So this is a bilateral system spanning frontal and parietal cortices. Now these regions respond robustly to diverse demanding tasks, from solving math problems to deductive reasoning to learning a new task. And the degree of engagement appears to depend on how hard you have to try. So the more you're working, the higher the responses in these regions. And these regions have been argued to basically support goal-directed behaviors broadly, and have been linked to notions like working memory, inhibitory control, attention, and consciousness. And finally, these regions appear to be causally linked to fluid intelligence, such that damage to parts of this system will lead to a loss of reasoning abilities and a loss of IQ points. So now I'll summarize some evidence for a very robust association between the language and the MD networks, including within the inferior frontal areas where they lie right adjacent to each other. And we've been able to find this association using the kinds of individual subject analyses that I mentioned. So in fMRI, these regions show highly distinct functional profiles. I'll show you responses in two regions, one from the language network, one from the MD network, both of which reside in Broca's area. So let's start with the language network. So in black and gray, I'm showing you responses to sentences and non-words. And these are the conditions that were used to identify this language responsive part of Broca's area, but the estimated responses are derived from data that are independent from the data used to define this region, so there's no circularity there. Now, in different colors are responses to diverse demanding cognitive tasks. The darker bars are harder versions of each condition, the lighter bars are easier versions of each condition. And we included the difficulty manipulation because historically, to find these regions, people have used difficulty manipulations. And we wanted to make sure that we're able to find the regions that others have found before. But the critical thing is that in this language region, we find remarkable selectivity for language over these diverse tasks. It's not only that they don't respond to the difficulty contrast. So 0 is here a fixation baseline. So when you're lying in the scanner doing nothing. So this tells you that this region works about as hard when you're solving a math problem as it does when you're looking at a blank screen with no stimuli presented. And we have since shown that these regions also don't respond to things like understanding structure in music, understanding others' actions, including communicative actions like gestures or facial expressions, understanding meaningful, semantically rich stimuli like pictures and videos, or even processing computer code, which has a lot of structural similarities to language. Now, what about MD regions? So here the picture is drastically different. So these regions respond to diverse demanding tasks, and they respond more strongly to the hard condition of each task as has been previously reported. Interestingly, these regions actually respond to the two language localizer conditions, the black and gray bars, in the opposite way from the language region, presumably because the processing of sentences is just more natural and less effortful. Now, there are several other sources of evidence for the dissociation between these two networks that are summarized in various published papers that I can point you to at the end, including double dissociations in brain disorders, dissociations in signal fluctuation patterns during naturalistic cognition, and distinct developmental trajectories. In line with this dissociation between these networks, they appear to contribute to language in quite different ways. So based on what we've learned over the last 10 years, it appears that the language network is a system that stores our linguistic knowledge representations, which I think of as a large set of form meaning mappings. And these, of course, can be used to encode thoughts into utterances, as well as to decode meanings from others' linguistic messages. The contributions of the MD network, this executive control network, to language remain debated, but it looks like it is unlikely to support any core computations like lexical access or structure building. In fact, it appears that this system only comes online during linguistic tasks when there is some extraneous task demand on top of the natural tasks of language comprehension and production. So effectively, when you take a language task and turn it into a problem-solving task, and then the system will come and get engaged. One possibility remains that this network may be helpful in recovering from brain damage when language areas are affected, and we and many others are investigating that possibility. So to summarize, there are at least 3 functionally distinct networks that coexist within Broca's area: the speech articulation network, as originally discovered by Broca, the language selective network, and a highly domain general multiple demand network. And each of these components of Broca's area is robustly identifiable within each individual participant using functional contrasts. So how do the language and the MD networks relate to the 2 kind of main hypotheses that have been around in the literature about the functions of Broca's area, in addition to its contributions to speech articulation, which I'm leaving aside for a second? So the second hypothesis, I think, has merit. There's indeed a region within Broca's area that belongs to the domain general MD network, which is the network that's implicated in the domain general executive control. However, the story about combinatorial processing in language or across domains is not right. In particular, the language selective component of Broca's area is highly selective for language over many other tasks that involve hierarchically structured input, including music, complex arithmetic, reasoning tasks, logic, and so on and so forth. But even within the domain of language, it doesn't appear to be the case that this area is a syntactic hub in any sense of the word, meaning that other parts of the language network appear to contribute to combinatorial processing just like this region does, and all parts of the language network, including the region that lives within Broca's area, is highly sensitive to word meanings. So I have been arguing that combinatorial processing is actually not separable from the processing of word meanings within our language system, which happens to actually align quite well with current linguistic theorizing and with many years of psycholinguistic evidence and with patient evidence. So failure to account for inter-individual variability in the precise locations of functional areas has led, I would say, to a lot of incorrect inferences and a lot of confused theorizing. In contrast, individual subject analytic approaches have allowed us to see a clear and replicable functional architecture within Broca's area and other parts of the brain. And so I would argue that the use of well-validated localizer contrast, these functional localizers, as people call them, which allow you to identify the relevant functional area or network, are quite critical for establishing a cumulative research enterprise where we can refer to the same region or network across individuals, across studies, and across labs as needed to be able to do research in robust ways. Some of this stuff is summarized in these 2 papers, and then you can find all of the other papers on our lab's website. Thanks for listening.
Thank you very much. That was very interesting approach to a very old topic. When you say that there's no individual words representation in Broca area, are you referring to a semantic components of speech?
Sorry, I missed the first part of your question. When I say that,
you mentioned that there's no individual word meaning represented in Broca area. Did I get this right?
No, sorry. What I was saying is that, so a lot of people have argued that this component of the language system is a syntactic hub. So what it does is just puts words together into phrases and sentences. And in contrast to that, I'm arguing that this region is just as sensitive to understanding the meaning of a word, say table or to run, which has nothing compositional about it. And that's a property we find for all other language regions. There seems to be a very tight integration between individual word meaning processing and combining these into larger representations.
Right, thank you. Well, that's great. Thank you so much for joining us. And we will now move on to the next and last talk for the session, for the second session. One of the key problems we have, one of the key challenges we have, as we saw earlier, is to parcellate the brain. And we started with Brodmann with the votes, then to von Economou-Koskinas, then to Bailey with the isocortex of man. And now for the last few years, we had a number of atlases based on fMRI, resting state, MRI, etc. And one of the key components was the introduction of the human connectome project and one of the parcellation maps that David Van Essen and his group has introduced. So it's a great pleasure to introduce David. He's the principal investigator of the human connectome project and has contributed as very few in the field. So David, thank you so much for joining us. We look forward to your talk.
Okay, thank you for the introduction and for the opportunity to present at this exciting meeting about cerebral cortical parcellation and connectivity. Let me start by giving a brief outline of what I'll cover. I want to talk about cortical maps, parcellations, and connectomes in laboratory animals, monkeys, and mice in particular. And I'll talk mostly about human cerebral cortex, covering individual variability again. We've heard about it already. Our multimodal human cortical parcellation, resting state networks, a little more about that, and brief comments on the relevance to neurosurgery. So our brains are 3 times larger than our closest living relative, the chimpanzee, 15 times larger than the macaque monkey, 200 times larger than a marmoset brain, and 2,000 times larger than a mouse brain. You can see at a glance there are dramatic differences in cortical convolutions, and it's these convolutions or folds that made me a cortical cartographer 5 decades ago when I started working on macaque cerebral cortex. So at that time, we used pencil and tracing paper to make cortical flat maps, and onto those flat maps, we represented information from the literature about evidence for several dozen cortical visual areas, some of which were very well defined by all criteria like B1 and the motion processing area MT, but many of the other areas were fuzzy in their boundaries and debatable as to their existence. Dan Fellman and I also looked at the literature and found evidence for several hundred pathways, connections amongst just the visual areas, and proposed that they were organized in what we called a distributed hierarchy. We also took those several hundred pathways and represented them on a 2-dimensional matrix, what we would now call a parcellated connectome, and showed at that time just the evidence for the presence or absence of pathways linking any one area with any of the other visual areas. Fast forward to 3 decades later in 2021, we now have much better quantitative evidence about connectivity, and we can represent it and more accurately on better but far from perfect cortical parcellations. And we can use cortical surface models, the inflated version you see here, and show, for example, from work by Henry Kennedy's lab, that a tracer injection in one cortical area 7A reveals inputs from 55 different cortical areas projecting to it, with a remarkable 100,000-fold range of connection strengths. Doing that for 29 different areas injected, Markov et al. Showed a connectivity matrix with evidence for about 60% of all possible pathways existing. The Rosa and colleagues have done a similar analysis for the much smaller marmoset brain. They used a 117 area parcellation and made tracer injections analogous to what you saw for the macaque, and quantified the connectivity, and did that for 55 marmoset cortical areas, generating a parcellated connectome, similar in spirit, but different in many details from what I just showed on the macaque. Jumping over to rodents, mice in particular have a much smoother cortex, and Andreas Burkhalter has pioneered ways to physically flatten and then tangentially section the cortex and provide evidence for 41 distinct areas in the mouse, much fewer than in the macaque or marmoset. And working with the Kennedy lab, provided evidence for a parcellated connectome that showed nearly all possible connections amongst the areas examined in that study. Lastly, in this collection, The Swanson lab has used a schematic flat map of the rat cortex and nearby regions and generated from data available in the literature evidence for connectivity amongst the 73 different cortical and nearby domains. So similar patterns in all species, difference in many details, but an extraordinarily rich connectivity network. Returning to the macaque for one more moment, we've been working with the Kennedy lab and taking tracer injection data, and instead of showing only parcellated connection strengths, showing continuous maps of connection strengths with the retrograde tracers that we can then compare with functional connectivity data obtained by our colleague in Japan, Takuya Hayashi. And you see here a seed location in the corresponding location 7B, and the functional connectivity reveals high correlations concentrated in the regions where there's anatomical direct connections, but the correlation is by no means perfect, and the functional connectivity, for example, shows moderate correlations in dark red in regions that on the left show no anatomical connectivity whatsoever. And this is the best case story in other tracer injection seeds. The functional connectivity correlation with tracers is even worse at the moment. So I'll return to that at the very end, but let's jump over to human cerebral cortex and look at cortical surface models that can be generated using high resolution Human Connectome Project data, and using Bruce Fischel's free surfer algorithm. We can generate very accurate cortical surface models, and we can spin them around and smooth them out and make inflated maps that show the buried cortex. And if we do this for many individuals, we confirm what has already appeared in many previous presentations today, the individual variability that can be seen here in lateral views of the mid-thickness surface and inflated maps where you can see some regions like the central sulcus are fairly consistent across individuals, but most regions are folded very differently. And remarkably enough, that's true even in identical twins. So there's one twin pair on the top and another on the bottom, and they're very different even with the same genetic construction kit. We also know that cortical areas vary markedly in size, twofold or more across individuals, and they vary in relation to the folding patterns in the underlying cortical sheet. That brings me back to the question of how the cortex folds in a developmental and mechanistic sense, and I'm going to briefly touch upon that in this one slide here. We know that cortical folding occurs mainly in the third trimester of human gestation, and at term equivalent, the newborn cortex is nearly as convoluted as in the adult, even though it's only 1/3 the physical size. Mechanistically, I started thinking about this a few decades ago when Tom Coogan and I discovered that the connections anatomically between V1 and V2 in the macaque form at an embryonic stage that correlates closely with when the gyral fold right at the V1, V2 boundary shows up. And the light bulb moment for me was to mechanistically propose that axonal tension, mechanical tension along the pathways within the underlying white matter brings strongly connected regions closer together, leaving weakly connected regions to lose the battle and allow sulci to form. This remains an attractive hypothesis, but it's by no means proven. And indeed, it's likely to be one of multiple mechanisms. But in reviewing this recently, I published a review and an updated model that has additional features that you can learn more about if you look at the PNAS paper in 2020. Over to the connections and the cytoarchitecture map of human cortical parcellation provided by Brodmann a century ago. That gave us a map with about 50 different cortical areas. But at the same time and in the same institute in Germany, the Cecil and Otto Vogt were using myelin stains to provide evidence for upwards of 200 cortical areas. And that debate as to which is the appropriate range has been informed by many studies over the past century, including more recent studies using functional connectivity and resting state fMRI. But we have inconsistencies across multiple studies, no consensus across studies by the Buckner lab, the Peterson lab, or various others using only unimodal analyses. So when Matt Glaster and I had the opportunity to work with the Human Connectome Project data, we took advantage of the exceptionally high quality of data and high resolution, but also the multiple modalities that let us use information about architecture, topography, connectivity, and function all in the same data set, and to use spatial gradients to find evidence for boundaries represented by sharp transitions and consistency across modalities, as well as the ability to parcellate individuals as well as group averages. So here's one quick example. Myelin maps that we can get from structural images, starting with the T1 weighted image. Matt Glaser, when he came to my lab, had noticed that in the cortical gray matter ribbon, some regions are consistently brighter than average, others are darker than average. And lo and behold, in the T2 weighted images, it's the reverse. What had been brighter in the T1 is now darker in T2, and vice versa for darker and brighter regions. So taking the T1 divided by T2 ratio at each voxel, colorizing it and mapping it to the cortical surface revealed a strong and obvious correlation with classical myeloarchitectonic stains for different cortical regions. So if we use that as a starting point for a cortical parcellation using multiple modalities, with the group average myelin map you see in the upper left, we take an exemplar area 55B, named for reason that you'll see in a moment, as a lightly myelinated region in between frontal eye field and premotor eye fields that are moderately myelinated. Taking the spatial gradient of the myelin map, we find gradient ridges that allow us to identify an elongated strip that's our candidate area 55B, which turns out to be a hot spot with a strip of language-related activation from listening to stories versus baseline using task fMRI, and with the resting state functional connectivity, temporal cortex seed leads to correlations within the 55B strip, whereas a parietal seed leads to low correlation in 55B and high correlation in neighboring regions. So altogether, the functional connectivity gradient, task, and myelin gradients agree with one another and agree with the lightly myelinated strip identified as 55B by Adolf Hope a half century earlier. So Matt and I and our colleagues systematically applied this to all of cerebral cortex and provided evidence for 180 distinct areas in each hemisphere with a remarkable bilateral symmetry in terms of the existence of these areas, which you can see on the map here are colorized to show functional specializations suggested by resting state functional connectivity. So the 180 areas we identified, 83 of them were similar enough that we used the same names as publications from the literature. 97 of them we called newly identified, even though many of them were actually just splitting of larger parcels into finer grain subregions based on the evidence we could provide. The gory details are provided in almost 100 pages of supplemental material in our Nature article, but you can go to our BALSA database and download very easily the entire data set for useful visualization on your own laptop or workstation. We can identify, using an aerial classifier algorithm, these cortical areas in individual subjects with high confidence and probability. So here's probabilistic maps for 4 exemplar areas using one 210 subject data set and a validation data set of independent individuals shows remarkable similarity in the probabilistic maps of the different areas. So you can see both the consistency and the variability on our atlases where the alignment has been achieved using functional as well as anatomical criteria to sharpen the alignment. But the alignment isn't perfect, in part because of the types of variability shown in the next set of illustrations. So here's a group average language network on the left, and you can see the correlation with the blue dots outlining area 55B. In an individual subject, typically the language network correlates very closely with the group average 55B domain. The middle panel shows the lightly myelinated pattern for that individual subject. But here's a subject in which our registration was not able to align the resting state network or the lightly myelinated subregion because topologically this individual had an unusual relationship with area 55B, switched with its neighboring frontal eye field and positioned over more medially. This is another example of an atypical topology. And indeed, about 10% of our subjects have an unusual topology for this area 55B, which is quite striking. If we go back to the group average pattern and look at resting state networks in a broader stroke, the Cole and Antichevich labs have generated from the HCP data a 12 network parcellation of the human cortex that you can see here. If we look just at the language regions, you can see the language networks in temporal and frontal cortex mainly in both hemispheres, relatively, but not perfectly symmetric. And on the right, you can see the blue cross representing the connectivity pattern for the language network. In contrast, the red areas and the red box on the right shows the resting state connectivity for the default mode network nearby, which you can see is anti-correlated with the task-positive singular opercular network shown in the purple region. So this is the functional connectivity network revealed by fMRI, and as I mentioned previously and others have also noted, functional connectivity only indirectly reflects anatomical connectivity. It's by far not perfect. If we go to the tractography approach that we heard from Anastasia and others and say, well, how well does it do and to what degree do we get agreement between functional connectivity and tractography? A recent paper by Rosen and Hallgren took the HCP tractography data and processed it to get the connectivity matrix you see in the lower right. And while there are many similarities with the functional connectivity matrix above, there are actually many differences, and the correlation between the two connectivity measures are surprisingly poor overall. So this establishes that we have a long ways to go to get improved estimates of actual anatomical connectivity using either of these approaches. And as I had touched upon in an earlier slide, we consider the macaque monkey the most promising model system for refining and improving our connectivity method estimates using either of these methods. So lastly, turning to neurosurgery that you heard a lot about earlier today, how do we map the kind of fine-grained cortical parcellation shown on the left to individual neurosurgical subjects? And one option is to take the group average and the probabilistic maps I've shown before, map that onto the patient's individual cortex if we can compensate for the physical deformations caused by lesions or tumors. Another option is to identify resting state networks and cortical areas in individual patients, which is very attractive if you can get high enough quality structural and functional data in individual subjects, which, as we've seen earlier today, is actually feasible. So while there's a lot of challenges, there are exciting opportunities. And to wrap it all up, I'll just summarize by noting that cerebral cortex is a mosaic of areas that ranges from a few dozen to almost 200, depending on the species. Cortical connectivity is extraordinarily complex, but has a systematic pattern related to networks and hierarchies. The Human Connectome project has enabled improved data acquisition, analysis, and data sharing, revealing that our own species has a cortex with about 180 cortical areas per hemisphere that we can identify in individual subjects. And there's great potential for further improving the utility of this for neurosurgical applications. So I want to thank the people in my lab, our collaborators in Japan and France, the wonderful HCP team, our funding agencies, and I'd be happy to answer any questions.
Thank you very much, David. That was an excellent overview of your parcellation efforts, and I think you have contributed tremendously to this field. With regard to the neurosurgical applications, one of the difficulties we have, particularly with regards to the image you showed us, is that the tumors produce a lot of mass effect, which results in geometric distortions. Would you be able to produce some models, some mathematical models to account for these significant distortions of the surface?
I hope there can be progress on that. With smaller lesions, Maurizio Corbetta's group, for example, has been able to achieve cortical surface reconstructions and that preserve the topological configuration despite the large deformations. That's one of the challenges, because if the tumor actually disrupts the cortical topology, it makes it much more challenging to map what is essentially a sheet-like mosaic of areas onto a partial sheet if some of the tissue has actually been degraded or destroyed. But I think, with the sophistication of various registration algorithms continuing to improve, there will be ways to maximize the mapping between cortical areas in a individual surgical or neurosurgical patient, and especially if the high quality neuroimaging data can be obtained in that individual.
Thank you. Are there any efforts to validate clinically these maps? Of course, your map and other parcellation paradigms, particularly using, for example, data from stroke or other clinical conditions?
I hope there are. I'm not personally aware of any recent validation efforts on that, but it's very amenable to, for instance, with very small strokes, looking for subtle functional differences that would correlate with the functional specialization suggested by our parcellation analysis.
Thank you. So I think it's time now to have a discussion with all the faculty. We have about 10 to 12 minutes. I appreciate you running behind schedule quite a bit, but all the talks and all the questions are so interesting. I think we all just being carried away, which is a good thing. So if I could ask all the faculty to turn on your cameras and your microphones, and the two issues I would like to discuss, perhaps 6-7 minutes each. In the background, there have been a lot of discussions in the faculty about the previous question of Professor Ribas with regards to connections of the corpus callosum with the sensory cortices and also with the limbic system. And I believe that Jeremy felt strongly about this, and he also suggested that David has an answer. So Jeremy, would you like to present again your argument, and then perhaps David can comment?
You're muted.
Thank you. Thanks for the opportunity, and what a pleasure and a privilege to listen to everybody teaching us. It's really been extraordinary. What a great session. In the work with Dr. Pandya with isotope injection, we found callosal connections with essentially every region that we looked at, but we did not look at V1. So the work that I was relying on and thinking about that was work that goes back to Siwak and Pandya and the cat. But there's no, as I mentioned in the chat, there's no expert more deeply steeped in this than Dr. Van Essen. And so if the question is, does V1 have connections? I'd love to hear your thoughts about that. But certainly from the motor region, all of the motor cortex has projections across the callosum. And I think that's true for the sensory cortex as well.
I guess what I would add is that V1 is, I think, genuinely special in primates, or at least in the macaque monkey. We had, in 1982, done a study using horseradish peroxidase put into the corpus callosum and found no cells whatsoever coming from V1, even though terminations were sparsely but genuinely identifiable in the V1, V2 boundary region. Jeremy, you pointed me to a paper by Henry Kennedy's lab a few years later, and I'm reminded, and I found the abstract, they actually did find a scattered or a small number of cells in V1 layer 4B right along the callosal or the vertical meridian V1, V2 boundary. So technically there are a few projections callosally, but they are sparse and they're very restricted. So I would still maintain that primate V1 is special with regard to, let's call it, severely hypoconnected relative to essentially all other cortical areas.
Thank you. So I think now I'd like to move to a very important topic, particularly for clinicians and also for neuroscientists, which is to assign function to cortical parcels and subcortical white matter segments. And as I mentioned earlier in my talk, when we operate on brain tumor patients, we have to be quite confident that what we test is accurate, is reproducible, and can preserve function. And looking through the literature, there's been a huge amount of controversy and lack of consensus as to the function. So I would like to first perhaps ask Nina and Craig to make some comments with regards to our degree of confidence on, let's say, what is the function of the Broca area, the pars orbitalis or triangularis, or the supramarginal gyrus. Everybody assigns slightly different function based on different data sets. But perhaps Nina and Craig can start describing what's the take on assigning function in these cortical parcels.
Well, I could speak for Broca's area to begin with. We find that lesions to Broca's area do not result in a persisting Broca's aphasia. Those patients tend to be mute for a while, and then they recover very well. But there we're talking about lesions that are truly restricted to 44 and 45. Now, of course, there's great variation, as was pointed out earlier. But anyway, patients with lesions there just, they do fine. And there certainly have been a number of neurosurgical cases reported as well that have shown that removal of Broca's area, again, leads to sort of a transient mutism. Mm-hmm. That tells me that Broca's area is probably involved in speech production in some way, but that it's not the only game in town. So there are clearly other regions that are helping to subserve speech production. I wonder, I once did a review of a lot of the functional imaging literature, and those that got activation in Broca's area seemed to me to often be accounted for, the activations were accounted for by the fact that the task required articulation or articulation to oneself, sort of inner speech, and wondered whether that could help to explain some of what's going on with what we see in activations in Broca's area. Also, Dean Flinker, a student of Bob Knight's and mine, showed that with ECOG, activation in Broca's area does not occur at the time of articulation. It occurs somewhere where things have been formulated, and it's just before it begins to be articulated. By that time, syntactic processing and lexical semantics and all that has already happened. It's really got something to do with very end stage articulation at least from what I have gleaned over many years. And as we'll see tomorrow, even Broca's cases didn't have lesions right in Broca's area either. So that kind of opens everything up even wider. Greg, you want to add to that?
Yeah, I think that the take-home message is we can't really draw much in terms of conclusions based on a single method. Every method is biased, and we need to look at all the methods together to get a complete picture. Like Nina said, functional activations are going to happen for a lot of different reasons, and it's hard to tell whether you're getting activation in Broca's area during listening to sentences because it's doing some sort of syntactic function or because people are having trouble concentrating in a scanner and repeating the sentence to themselves to perform the task. It's difficult to know. Lesion data, cortical stimulation, and other sources sort some of that stuff out. I think another complication is that, in general, my take is that the two hemispheres are more similar to each other than they are different. Of course, in language, we have some asymmetries, but until you disrupt function bilaterally, you don't typically see major league deficits. Well, speech production is probably one example of that, a high level function that can be disrupted with unilateral damage. But even if you remove or damage opercular regions on the left, there's often some residual speech, and it's not until you deal with bilateral opercular resections or damage that you get something as severe as opercular syndrome, which is obviously very severe. So that's always a complication in trying to reconcile functional activation, cortical stimulation, and lesion data, and it's something that we need to take into account.
So George, I just want to raise the point that we have to keep in mind that the role of an area could change over development, that what we learn about what the role of Broca's area or any particular area is in early development may be quite different than what it is later. As kids are developing the sensory motor loop, as they're learning to not only listen, but to produce, so they're presumably moving on according to a template, a sensory template that they formulated by listening to ambient speech, but to have to coordinate that with an enormously complicated motor system that takes, until kids are 8 before they can produce the full complement of sounds used in the language. And so anyway, the point is just that development is a totally different factor, and we won't know until we've really done a lot of very careful work how these roles of areas and networks change with development.
Well, I'm sure that during the various stages of development and even in the adulthood, there will be different responses to lesioning or stroke in these areas. For us, however, and when I say us, I mean surgeons, we are very keen to have a more clear understanding of the testing paradigms. For example, when we have the pars opercularis, we tend to test phonology and articulation. And when we're looking at the pars triangularis, we're looking at syntax, grammar, and semantic functions. If we don't test the right paradigm in the right area, is the equivalent of testing sensation while you're trying to remove the motor cortex and vice versa. And oftentimes this function is very elusive, but we just try, and this is one of the purpose of the association and of the meeting, to try to put our heads together and understand what is the most likely testing paradigm can be applied for a specific cortical parcel. And as we've seen in the past, the Broca's area is very elusive. As I said, I have resected the so-called Broca areas in many of my patients with awake brain mapping, and they had no deficits. And I believe that Anthony has produced a nice series where it showed how people understand the Broca area. Anthony, do you want to elaborate on this? Some is too big, some is too small, some is completely off.
Oh yeah, I think of this anterior region as sort of being more in the line of sort of the way Thompson-Schill or Petrides have talked about it, as sort of resolving conflict among potentially competing representations. So if it's a potentially competing phonological representation, like you want to say boy instead of toy, there has to be a mechanism for resolving that potential conflict. Or if you have potentially competing semantic representations, you need a way to resolve that in the process of coming to a meaning. So I always think of this region as being situated in the context of its dynamic interactions with other regions of the network, and that together they come to a consistent representation on the way to producing a sentence or understanding a sentence or producing particular words and so forth. So different subcomponents of that inferior frontal gyrus area may be more tuned to resolving competition for phonological connotations as opposed to semantic. And I think there's a lot of TMS work that suggests that as well. Some of the old studies by Joe Devlin, Poldrak, Go, and people like that. So I think that's the function of the region. And this is where I think I would disagree with Ev a little bit. I don't see that as being a multiple demand function. I see it as being a selection function in the context of linguistic or phonological processing. This is where I would agree with Greg. I think in other domains, you get a similar kind of function. So right inferior frontal gyrus may be involved in resolving competing potential action representations. So instead of reaching up, you want to reach down, you've got to resolve the conflict in some way. Or if you want to, you're doing a stop signal task, you don't want to respond in that particular situation, that conflict has to be resolved. And right IFG is involved with the basal ganglia and subthalamic nucleus in determining the output. And that happens in speech, maybe that's left lateralized, and it happens for motor acts, and that maybe that's a little bit right lateralized, although still a bilateral process. So it plays a role in the context of a broader network. And it's really in line with, I think, if you go back and read Patricia Goldman-Rakish's old work in the 80s, it's in line with some of what she was writing about for how she thought of the way the frontal lobe may function.
To that end, we also try to assign function to white matter tracts. And looking at most of the tractography papers, it appears that the function is inferred by the origin and termination. For us, again, it's very important to test the right function. If we're not testing the right function, we might miss the deficit that the patient might develop. And earlier, Juan mentioned that the arcuate fasciculus has been resected, although it did not produce any deficits. And this posed the question as to what function have they been tested. For example, if it is Boston naming task, might miss this. If it is sentence repetition, it might be more sensitive to catch the deficit. So in this line, I wonder if Jeremy can discuss with us how he infers function, at least in his extensive work with the macaque monkeys.
Thank you. You know, I think there was a phrase that one of my former students came up with that's a bit cheeky, but is interesting. It was a corticocentric myopia. And I think that in thinking about language, Mani Neeser some years ago, I think in the 80s, was looking at recovery of language after stroke, and she pointed to what we now think of as a subcallosal fascicle of Muratov as being an important piece in recovery of language function, of articulation. And it gets to the whole business of the cortical-cortical, cortical-subcortical connectivity, and each node having its particular role in subserving a function. I think that, we know you can get an instant dementia from a thalamic anterior thalamic lesion. So we know this is directly relevant for all sorts of cognitive performances. And I think that you're right. I think I agree fully that in thinking about the functions of a tract, we're actually thinking about what are the potential consequences of disruption of communication between two different regions of the cortex or subcortex. And it becomes an inference because of what we know or think we know and are continuing to learn about the different functional properties of the cortex and from animal studies, and then from the magnificent work that Dave is doing in the Connectome project, learning from people about the different functional properties of different cortical areas. So it becomes an inference about the function of a tract determined by what the cortical areas do and how, by their emergence together, they may have some kind of emergent property that comes out of that discussion.
A typical example is the SLF1 connecting the precuneus with SMA and pre-SMA. and we infer that this is higher order level of motor function.
Yeah, George, sorry.
Please go on, Roger.
Just a sort of philosophical point, really, because I suppose the question you're asking suggests that, firstly, that there is a unique function that is not shared with any other parcel, which the patient may be able to use, despite the fact that you are upsetting it or resecting it or whatever. And also that it also suggests that any one parcel has only one function. And I think, some of the areas that we are most familiar with, we now know, we now know, even in primary sensory areas, that there are some important different functions going on within the same cortical parcel. So these are both sort of things that you have to have in your mind when you're thinking about areas, and also the fiber tracts that connect them. And something that I've been very interested in is, how is it that, if you look at something like the corticospinal tract, one of the older studied pathways in the brain, it has a vast range of different size fibers within it that come from a vast range of different cortical areas. So there is almost no point in asking the question, what does the corticospinal tract do? Because there are going to be multiple answers to that question.
If I can add to that, because it's really important, I think Roger stated it well, that the long-standing debate over functional specialization versus distributed function is not going to be completely resolved easily, because I think the truth lies very much in the middle ground overall. There are some areas that are distinctly specialized, particularly in visual and early sensory areas. But as we get farther into the middle domains or up in the hierarchy, there really seems to be much more evidence for distributed and function and partial redundancy built into the system. And when we look at our, with the Human Connectome Project data, we essentially have a more than 100-dimensional feature vector that shows evidence for the degree of specializations or this degree of functional connectivity that is very different between neighboring areas for a small set of sharp contrast domains, but usually is a pretty subtle difference from one area to its neighbors. And so I think part of the challenge that you articulated from the neurosurgical perspective relates to the diversity of sharp contrast in some regions and very modest transitions across many other cortical areas, compounded with what I talked about. The reason I've made the points about anatomical connectivity is the distributed nature of the wires means that when you stimulate a particular fascicle, you're likely tickling, as it were, many different cortical areas to very different degrees according to the distribution of the underlying wiring. So it's hugely challenging for a number of reasons, and we just have to keep pounding on it very carefully and be alert to these issues of individual variability, and the complexity of the anatomical patterns and the wiring underlying it.
Thank you. And if I can ask Anastasia, your take on assigning function to a white matter tracts as shown on your screen.
Yeah, I would echo what the other panelists have said. It's hard enough to assign function to a specific cortical region or a specific, even what are referred to as functional networks. The same sets of regions could come up as co-activated in many different settings and different different functions. So if it's hard enough to assign function to cortical regions, then we can imagine that because these large white matter highways, they have fibers that are projected through them from all sorts of cortical regions, then it's even harder to assign a single function to a whole tract. I also want to point out that there are some tracts that are more topographically organized than others. And so in those more topographically organized tracts, like in the corpus callosum, for example, you have a better sense of, depending where you are in the tract, what part of cortex might be projecting through there. But then there are other pathways like the unsynaptic fasciculus, for example, that are less, they're more disorganized and less topographically organized. And there you might have fibers from, the entire orbital cortex all intermingled in this pathway. So I think there are various also degrees of difficulty in assigning function to white matter bundles.
Perhaps we can take the example of the dual stream language model with Craig, and perhaps we can close this session with this part. Clearly, the dual stream model has assigned categorical functions to ventral and dorsal streams. Craig, what was the process of assigning function to these streams?
Honestly, it was kind of derived from homology, reasoning from homology to visual motor systems, where you get what at the time seemed like pretty clear divisions between what Milner and Goodale called vision for action and vision for perception. And that inspired me and David Popple to think about language systems needing the same sorts of interfaces. So if you think about perceptual speech representations, auditory speech representations, you have to be able to comprehend or map those representations onto meaning. And that is one kind of computational mapping onto conceptual systems. And then you also have to map those auditory representations onto your motor system so you can reproduce those sound patterns yourself to talk. So computationally, they're very distinct. It's straightforward. And then it was just a matter of working out the neuroanatomy, which was fairly well, solvable problem, at least at a course level based on lesion work and so on. So it was that kind of reasoning that led us down that path. I think there's some confusion though, and I'll talk about it a bit. These models, I'll talk about it a bit tomorrow. These models are really models that make sense from a perceptual scientist sense. If you're interested in perceptual science, what are the two kinds of things that a perceptual representation needs to interface with? And those are the two. But if you're studying other aspects of language, like picture naming, it doesn't make any sense anymore because now you're going from concept through lexical to phonological to motor, and that's the entire system. It's not a dorsal ventral stream in the same sense. It's just mapping from a high level to a low level motor plan. It's the same circuit. It's just a different perspective that you're thinking about. And whether you invoke one component of it or not is really a task dependent kind of thing. So there are subtleties in terms of the computation. And I think that we can learn a lot in connecting white matter fibers to different computational mechanisms by looking to motor control homologies, because I really do believe that these high-level language circuits, and by high level I mean lexical, phonological, are evolutionary ancestors of motor control circuits. And they follow the same kinds of computational architectures. And I'm even tomorrow going to extend that idea up to the level of syntax, which is controversial, no doubt. But I think there's pretty good evidence for it.
Thank you. Well, thank you all for taking part in this session. I'm aware that we are running hugely late. So we'll have a 5 minutes break so you can stretch your legs and you don't get DVTs. And then we will join again in 5 minutes with the last session. So see you all in 5 minutes. Okay, we are back. So this is the last session of the day. And today we will be discussing in this session aspects around neuroimaging, surgical technology, and advanced ways to examine and treat the nervous system. I know we're running late, but there's a lot of enthusiasm and interest. And I wonder if the delegates from around the world can send us a brief message on the chat on Zoom, just to say where they're from. People could be from some city in Australia or some small town in North Africa or some university in North America. Just drop us a line and says, I'm John from this place, so we can get our faculty going, because they're getting a little tired. So it's a pleasure to introduce my colleague at Queen Square, the National Hospital, Professor Sotirios Bizdas, who's a professor of neuroradiology both at UCL and also the University of Tübingen. And he has produced very extensive research, particularly with regards to first clinical applications of cutting-edge imaging like the MR PET and ultra-high-field MR spectroscopy, and he's performing quite significant translational clinical research. So Sotirios will be talking to us about multiparametric imaging. Thank you very much, Sotirios.
Thank you very much, George, for this exciting online event. I have watched the whole day. I'm really excited about all this kind of information that has been offered so far. Now, as you said, we will dive into clinical imaging. And in my talk, I will first show clinical exams of multiparametric imaging, and then I will briefly touch upon the emerging techniques in the field. So the multiparametric imaging, of course, spans from the morphology to the biochemistry. It's a wide field, and it has a lot of physiological features in between. It includes perfusion, DTI, fMRI spectroscopy, and of course, when we go on the molecular side, we will meet the PET, which is a key component of multiparametric imaging. I think till 10 years or 15 years ago, it was quite time consuming to acquire all these modalities in a separate appointments for the patient. But since the launch of hybrid and simultaneous MRI PET, we can have this information within 40 to 60 minutes. And this has revolutionized actually the multiparametric imaging and how it can be used in the clinical routine. I will start with very basic paradigms, which I will highlight, however, that we need a shift, and definitely our clinical routine should embrace this multimodality imaging.
So
this is, for example, yes.
Sorry to interrupt. Would you mind go to presentation mode?
Oh, I'm sorry.
That's all right.
I thought it is the presentation mode. I will do that. Is it now the presentation mode?
I'm afraid no, it's still it's the presenter mode.
Okay.
Go back to presentation mode and swap displays at the top.
Okay. New share. I will share it again. Maybe how can do that?
Just start the presentation again, and then there's a button that says swap displays.
Okay, I will do that. So first, and swap displays. I think now it works. Is that right?
Not sharing your screen, I think.
Okay. Too many things at once.
Don't give up, Sotirios.
Share screen. Okay, now sharing screen. Swap, or do you see that? Is that fine now?
Still the same, I'm afraid.
No, try swap.
Try swap again. Now?
There you go. Yep.
Good job. Thank you,
great.
and great.
Thank you. So
is probably very technically difficult to do simultaneous MRI and PET, but it's more difficult to have presentation mode in Zoom. So actually here we have a left frontal tumor, and you see it is not enhancing. That would probably seem to be an indolent low-grade glioma, but we need more information which will guide the surgical planning about the surgical boundaries of resection, about the hotspot that should be biopsied in order to offer a representative histology of this tumor. And so the structural imaging with contrast enhancement is very often very unspecific, and we can enhance our diagnostic confidence with MR spectroscopy or methionine PET MR. Methionine is a radio-labeled amino acid, which is state-of-the-art category of PET tracers for brain tumor imaging. So we see in this case that we have indeed to do with a low-grade glioma, but in this case where we have exactly the same flare and contrast-enhanced T1 features, the spectroscopy and the PET shows high-grade tumor features. And of course, having this information fused into the neuronavigation data set, we can guide a safe and representative biopsy in the tumor or resection. So the multiparametric imaging offers us accurate diagnosis and staging of the tumor and eliminates any sampling error if we go for partial resection. I have another case here where actually shows that the contrast enhancement is very unspecific. This is a corpus callosum tumor with extension into the right white matter. And though the patient or the tumor didn't show any enhancing features, any necrosis, the RCBB, which is the perfusion MRI, demonstrated an extremely high value of intravascular blood volume, and the PET MRI also showed a very avid tracer uptake in this tumor. And such tumors which actually now they don't show malignant features, but they have such an extraordinary high perfusion or tracer affinity. These are now classified as IDH wild type GBMs, and we have now done multiparametric imaging in our institution in more than 200 patients the last 5 years, and we see that this is a consistent feature in these tumors, high perfusion and very pathological PET MRI. Another case where actually the multiparametric imaging helps us to address either bias or shortcomings that each advanced modality might have if we use that in a standalone way, is this one where we have a pediatric patient. We see that there is not a clearly delineated mass on the left inferior frontal gyrus. The methionine uptake is not something which is could somebody tell that it is very high. It is very close to the physiological activity we see in the basal ganglia. However, having DWI and MR spectroscopy in our arsenal of multiparametric imaging, we can easily see that the intensity of the DWI shows us that we have a high cellular tumor. At the same time, the MR spectroscopy offers a lot of information about the choline, which is a cell proliferation biomarker. And because it is a technique that can sample a wide array of metabolites, it has also detected taurine, and this tumor actually fills all the necessary criteria that we need to call it a primitive neuroectodermal. So we are talking about tumors with high cellularity and also very aggressive metabolic profile, including taurine in the sample metabolites. And of course, this has implications for the management of the patient. This is something that is useful to know before we, or when we get or try to consent the patient for the surgery or for the chemotherapy afterwards, and of course to guide the surgical resection. And I just mentioned spectroscopy as a very potent tool in order to understand the tumor pathophysiology. And this is an example where actually in the same tumor, we can create metabolite maps of different metabolites. And each of these map gives us different kind of information, which we need to synthesize in order to understand better the malignant features. For example, we have metabolite maps of N-acetylaspartate, which shows the neuronal density in the tumor. At the same time, we have lactate metabolic mapping, which shows us the anaerobic transformation of the tumor metabolism, glutamine, glutamate, and so on. So these maps are color maps. They can be easily also exported and guide resection. So far, I have shown you about the pre-treatment imaging. Now, in the surveillance imaging, the multiparametric approach helps us to understand what is going in the tumor at the tissue or a molecular level. And I have here an example of a baseline tumor where actually we opted for wait and see approach and examine that 3 months later. At the baseline, we had acquired spectroscopy, which showed kind of in low to intermediate malignancy features. But 3 months later, the contrast enhancement might be less, but the spectroscopy is clearly now showing malignant features with decrease in the NAA and also presence of lactate, exactly highlighting this transformation into more anaplastic and aggressive tumor. And of course, this can be used as a monitoring tool in order to understand which is the right time point to initiate the therapy. Pseudoprogression is also something which is clinically always a diagnostic dilemma and a challenge. Multiparametric imaging achieves, in my opinion, the highest accuracy rate in order to stratify the patients correctly, avoid unnecessary treatment, avoid under or over treatment at the same time. This is a patient where he received irinotecan and bevacizumab is very notorious for obscuring the contrast enhancement in these tumors. So the classical T1 post-contrast images cannot be used for reliable disease staging. For this reason, again, multiparametric metabolic imaging with PET and MR spectroscopy showed that in this increasing flare hyperintensity on the follow-up, we don't have any underlying disease. That's pure pseudoprogression. At the same time, pseudoprogression might have also been manifested with contrast enhancement, with increasing contrast enhancement and increasing edema, as in this case. But a reliable differentiation from recurrence could be also obtained by using K-trans, which is a permeability biomarker and is elevated in neoangiogenesis. And of course, we see the excellent spatial agreement that perfusion has with the methionine PET. So having two techniques showing positive findings, we increase the diagnostic confidence in our clinical work. On the other side, we can have the postoperative surveillance with increasing contrast enhancement and edema, but before the patient has received stereotactic radiosurgery, and this raises the possible differential diagnosis of radiation necrosis. We see here a patient who had developed edema and contrast enhancement in the internal capsule, highly eloquent region who cannot do more than wait and see. And at the baseline, we acquired perfusion and PET. We showed negative findings, and we were very actually confident that the increasing enhancement 6 months later is nothing else than radiation necrosis. It took a while till this contrast enhancement resolved, but the high negative predictive value of multiparametric imaging on the baseline avoided an over-treatment of the patient with more chemotherapy or stereotactic radiosurgery. Here I have a similar scenario where the patient was treated with multiple therapies, developed a quite nasty enhancement in the parietal lobe. Again, we utilized all the available MRI modalities at that time, and we see that spectroscopy shows low metabolites in edema. However, we see very low, actually almost at the level of the noise, concentrations of NAA and choline in the enhancing lesion. That actually tells us that we have necrotic tissue, and there is no viable tissue there. And of course, DWI confirms these findings. Talking about DWI, I think that it's worth mentioning that DWI has been revisited the past few years, and now for diagnostic purposes, we go a step further and we try to analyze DWI in a way that offers us not only information about the tissue cellularity, and this can be elucidated by the so-called slow diffusion, but we also now capture using appropriate techniques the fast diffusion of the water molecules, and in that case what we do is a surrogate biomarker for the capillary perfusion. So there is a small variation in the acquisition technique, but more importantly, there is a different mathematical approach. The technique is called intravoxel incoherent motion, captures the fast moving molecules in the capillary network. And we see here a non-enhancing insular tumor, which however shows red areas here, which are increased capillary flow areas. And these are hallmarks of anaplastic transformation and malignancy. Then the patient was found to have a WHO grade 3 tumor. At the same time, DWI can offer us a lot of information about the slow diffusion on the other spectrum of the DWI values. And by using, again, an appropriate mathematical modeling, we can calculate the so-called kurtosis DWI, which is a surrogate biomarker for the heterogeneity within the tumor mass. And actually, if we have low kurtosis, we have low-grade gliomas, and this is actually well correlated with the IDH mutation. And when we go for higher kurtosis, which denotes a higher degree of disorganization and higher heterogeneity in the tumor, we essentially have to do with grade 4 tumors and GBMs. Now, another emerging technique that I would like to draw your attention, because it will be a major player in metabolic imaging and in neuro-oncology the following years, is the so-called amide proton transfer, chemical exchange saturation transfer. So shortly said, APT-CEST, and actually captures the chemical exchange between the bulk water and the endogenous mobile proteins and peptides. So it's a true alternative to radio-labeled amino acid PET. And you see here in comparative studies we have done in PET MRI, we see that essentially the APT-CEST in this tumor on the left insula shows no increased amount of proteins and peptides. And this is very well in high agreement with the FET PET, which is the fluoroethyl tyrosine PET. And when we have here a tumor where actually supposed to be an IDH mutant grade 2 tumor, we see that the amount of proteins is very high, reflects very well the results in FET PET, and actually it was found to be a W8 grade 3 tumor. That is really a huge technological and clinical advance, because the APT test is acquired within 8 minutes. It doesn't have all the logistic hassle of the FET PET, and of course, it's magnitudes cheaper than an FET PET. Talking about metabolic imaging, I will just again remind you that spectroscopy, by giving us information in certain metabolites, certain peaks that can be identified in the acquired spectrum, we have the so-called oncometabolic correlation with certain tumors. So if we find alanine, it's a central neurocytoma. If we see glutathione, then it is a meningioma. And of course, the holy grail was when we managed to measure in vivo the amount of 2-hydroxyglutarate in IDH tumors. And this enables us in a non-invasive way to differentiate between the IDH mutant from the IDH wild type tumors. And of course, this has tremendous clinical implications for the patient management. And one of the last examples, this is here a tumor with faint enhancement. It is in the frontal lobe, a loquent area involving also the middle frontal gyrus. By using appropriately the perfusion imaging, we see that the posterior part is the hyperperfused, and if it is about here resection or it is about biopsy, this is what we firstly need to sample. At the same time, by using spectroscopy, we can identify another oncometabolite, which is glycine, and this has been correlated in a high, very statistically significant with GBM. And talking a lot about resting state fMRI today, I would just put a little bit of flavor of resting state fMRI in my talk, but I would approach it not only on the preoperative, but also in the intraoperative setting. So we have quite a lot of cases investigated the last years with resting state fMRI. We see a very high agreement between the localization of fMRI eloquent functions with the ones on the resting state fMRI. Now I have here an example of sensory motor activity on the resting state fMRI, which correlates well with the clinical validation of finger tapping, and another case with language fMRI, which showed us the correct lateralization of the language, and the patient had no language deficit. I think here what is revolutionary is to use in an intraoperative setting resting state fMRI. And so acquiring the baseline and then on the intraoperative setting, we showed that the Z scores between the, or the differences in Z scores correlate well with the outcome and the degree of paresis in cases where the tumor was near the motor strip. And to recap, I think that doing only the conventional diagnostic approach, we capture only the tip of the iceberg in neuro-oncology, and multimodality imaging is needed to shed light into the tumor physiology, because this is essentially what is the decisive information for tumor staging and also for surveillance of the lesion afterwards. There are established techniques like PET, like spectroscopy and perfusion, but also there are new techniques, emerging techniques, and I would highlight again here the APT test. These techniques can be easily implemented. We need to do some preparatory work on that. You need to discuss that with a neuroradiologist, but these are things that are clinically available, and there are clinically certified and commercially available sequences to do that. And this will actually be the cornerstone of personalized medicine. And I think it is worth investing in it. Thank you very much for your attention.
Thank you very much, Sotirios. That was excellent review. And this is an example how in clinical practice, we have to superimpose multiple layers of information with regards to the pathology, to histology, to metabolic imaging, to functional imaging, including the cortical anatomy, the mapping paradigms, and the surgical techniques. So these are all very important pieces of puzzle we have to put together to look after our patients. There are lots of questions on the Q&A section. So because we're running so hugely late, I wonder if Sotirios, you would be able to answer those in writing. You can type directly. So thank you very much for that. Also, thank you very much for those who have been sending your regards from various parts of the world, including Ajay in Pittsburgh, Sizwe in South Africa, Vinkes Waran in Kuala Lumpur. It's now nearly 5 o'clock in the morning. Thanks for staying with us. Kelly, pediatric neurosurgeon in Oregon. Manhar here in Japan is now 5:30 in the morning in Japan. So please keep sending the messages and your questions. Now we are moving to Mr. Kevin O'Neill, who is a consultant neurosurgeon at Imperial College in London, and he's got very extensive research in the brain tumor management. He's also head of research, and he has published extensively, particularly with regards to spectroscopy. Also, he has recently joined multiple efforts to promote neurosurgery and safe tumor resection in the UK. Kevin, thank you so much for joining us today.
Thank you, George. Thank you very much. I'm just going to share my screen and get the presentation underway. I have to say that once again, this now being the 5th year in succession, this course just gets bigger and better. And being part of a faculty with so many legends within their field is really quite humbling. So thank you very much for that invitation. And in the words of Monty Python, now for something completely different. Although having said that, can you hear me okay? Yeah.
Yes.
Yeah. Having said that, it's very similar to Sotirios' talk, although it's an optical version of it. But this is about clinical spectroscopy as a technique to solve problems, and particularly pertaining to clinical problems around neurosurgery and, in this case, brain mapping. And it's funded by, well, it's part of a big research program funded by Brain Tumor Research and the Brain Tumor Research Campaign. So spectroscopy is defined, and it's derived from both Latin and Greek, and it means looking onto the spirit or the ghost, which is actually quite irrelevant because this really applies to the initial definition of spectroscopy, which was looking at the different frequencies of light as it interacts with matter. In fact, now it refers to a plethora of different techniques that deal with measuring, interpreting the interaction, the spectra that are formed by the interaction of EM radiation as a whole with matter, concerned with the various aspects of that, including absorption, emission, and scattering. And there are different types of study of this interaction, but in simple terms, these types either involve the transfer of energy from a photon to the matter or no transfer. And each of these properties have some information to impart in their own right. But if you look at the side, I think spectroscopy, clinical spectroscopy actually includes the interaction of any wave, even outside of the light and EM radiation categorization, and that includes sound and gravity. And sound has been obviously highly utilized with ultrasound in its reflective nature in matter, but I think there's going to be a lot more heard about the lower frequency information that can be gained from the penetrative aspects of sound, and that will be hopefully an aspect of a talk as I'm looking into it in the future. I've also included mass spectroscopy, or really better term spectrometry, which is a form of spectral separation of ionized matter according to its mass charge ratio, something that we all know pretty much a reasonable amount about. But I'm going to focus on a few aspects. I'm going to talk about absorption, photoluminescence we know a lot about because of the luminescent effect of UV light on protoporphyrin produced by 5-ALA that we give to high-grade glioma patients, but actually a lot of tissue will autofluoresce with certain frequencies, and this is a property that we've underutilized and certainly something that will become more and more useful in the future. Regarding mass spectroscopy, this is really about the eye knife, which many of you probably heard about, which in itself is essentially an atmospheric interface between tissue, or indeed the vaporized smoke that we produce from electrocautery or indeed laser ablation, and the mass spectrometer. And I'll explain that in a second. Regarding absorption spectroscopy, this is an aspect of all frequencies, but many frequencies of EM radiation are potentially harmful to biological tissue because they're ionizing. So we really focus on those that aren't and that are non-ionizing. And you've heard a huge amount about radio wave absorption in the form of nuclear magnetic resonance spectroscopy, which is the basis of MRI. And you've heard a lot from Satiris about the expanding aspects of spectroscopy in that field that will become more and more useful. But I'm going to speak about Raman in particular, which is a part of infrared or near infrared spectroscopy. And infrared spectroscopy is something very similar. It's complementary, but I'm not looking into that. And also the visual part of the spectrum in terms of multispectral or hyperspectral imaging. So before I go into that, I wanted to just talk about the philosophical aspects of using spectroscopy clinically. And it's really, spectroscopy will give you a new view on things. It will give you new information, but it will also give you holistic information. So this comes down to the argument about reductionist versus holistic approaches to problem solving and research. Most of research that we do is reductionist in that we tend to break it down into its component parts to understand its mechanisms, and then correlate that back to the clinical problem, which of course is difficult because any clinical biological system is complex, particularly the brain, and you will often be missing something that's very important. You often will not be able to see the wood for the trees, and it's important to actually see interrelations with other things to understand them better. And in this case, you can see the forest and its relation to a beauty spot nearby. But in terms of brain mapping, you need to know, how the arcuate fasciculus relates to the visual system, for example. And clinical spectroscopy is a form of systems biology approach, which has been made feasible by advances in technology in parallel with advances in computational power. I remember years ago in the 90s looking at putative spasmogens in vasospasm and thinking, how the hell are we going to solve this problem? And thought, well, maybe the answer is in CSF. If we could look at its component parts, that might give us an answer. So I did all sorts of spectroscopy way before my time, very difficult, because the computational power we had at our fingertips were 33 megahertz computers at that stage. So we did find some very odd things. One of them was the IL-6 story, which is now part of a trial, and various other things which I should have published, but there we go. But the whole point about this is that technology these days can interrogate tissue in very detailed ways, providing huge amounts of data in the attempt to answer biological questions. And with computational analysis, including algorithms and AI, we can then produce at least new hypotheses and new insights into that biology to answer those questions. And as you hone down, the cycle hones you down further and further until you get closer to the answers. And this is what I think clinical spectroscopy is doing. And in terms of its context with pathology, brain tumors is a perfect example. When you look at the molecular and pathway aberrations that occur in a brain tumor, they're vast. We've found many main drivers, which you can see as the red dots. But if you try and block one of those, there will be another pathway or an aberration which will negate the effect of that blockade and limit your success in dealing with that tumor. And although a reductionist approach has been very useful in terms of really honing down on what's going wrong at the molecular level and giving us a better idea how to classify these tumors in relation to how they're going to behave and how they're going to be vulnerable to treatments, we still have made limited success. So many of the new treatments have been very promising, but again have only had limited success because we're usually probably missing something. For every good aspect of a treatment, there may be another aspect that is counterproductive. So to look at it more generally, which you have to, it's not just about gene expression. It's not just about signaling. It's more about the system as a whole, the microenvironment, the immune system, the physiology of that organ, even down to what the individual does to affect his metabolism, for example, diet, even the individual's environmental characteristics that might affect them. So it's very complex. And we're often going back to the blackboard to try and fill in those gaps. So this is the philosophy of the spectroscopic approach. And this is mirrored in brain mapping, in that there are trends in our understanding of brain function pathology are similar. We're trending from an understanding of the gross anatomy to more and more detail about the connectome and the microscopic level. Even now theories about the bipolarity of electron spin accounting for the huge amount of memory function that we are capable of. But we're using more and more elaborate tools to correlate anatomy and function, and the constant debate in this meeting has been about that correlation. And a lot of these include spectroscopic techniques, mainly MRI techniques. But I wonder whether or not, certainly at the surgical level, whether our understanding of brain function and that correlation of function to anatomy could be answered by some form of clinical spectroscopy. So again, sorry, to continue with the philosophical thing, because spectroscopy gives you a broad view on a complex biological system, it can be broadly applicable to all disease states of that system. And because it's adding another dimension of detail to the clinical data, that detail is fundamental because it's molecular. And as a result, it'll be fairly precise, fairly sensitive, and hopefully very specific. And if it's very specific, it will have a tendency to reduce uncertainty in your clinical decision making. And it's also likely, because it's filling in gaps, pick up most changes and define differences. For example, the difference between normal and pathological brain. And it will be able to, hopefully, with the advent of more and more precision medicine, match the correct treatment to that particular molecular phenotype and monitor treatment response. And because, again, it's filling in gaps of our understanding, it may then provide novel routes for investigation, novel routes for research, and new correlates to the disease, new biomarkers, new insights. So that's the advantages of it. Currently, what we're using is all the aspects of nuclear magnetic resonance spectroscopy, which is MRI in its various forms. And we're looking at the intensely interrogating that form and the imaging manifestations of that, including the Brownian motion that's incorporated in tractography, and the proton, the difference in the effects of protons in deoxy and oxyhemoglobin ratios to look at function and perfusion. But you know, how do we get that into theatre? We bring it in terms of imaging for neuronavigation. Some of us are lucky enough to have intraoperative MRI, but many of us don't. It's very expensive, it's quite cumbersome, and it's difficult to incorporate in that setting. But we do use standard things. Here's an awake craniotomy separated by that sterile drape, with the patient being, again, task-driven, interrogated with neurophysiological monitoring. We've got three-dimensional ultrasound going on in the background. But at the end of the day, when we look at that, what looks like normal brain, we still have to take a leap of faith in intervening based on imaging, perhaps intraoperative imaging and neurophysiological monitoring. So can we do something else? Can we add some other dimension to our understanding? So I'm going to talk about how we've incorporated mass spec technology in the form of iKnife. And just a brief understanding of that is, you know, whatever your subject matter of interest has to be vaporized and then accelerated into a very strong electromagnetic field, where those vaporized particles will get separated according to their charge mass ratio and give you a spectrum, a molecular fingerprint of those molecules, a spectrum, if you like, of that vapor. And that vapor is constantly being produced in surgery through electrocautery or laser ablation. And here's a picture of the prototype of the iKnife, which is still a big mass spectrometer on wheels. But the key element is this atmospheric interface, which is this thing down here, which draws in smoke from your surgical field and then passes it through further processing straight into the mass spectrometer. And this is how we've done it. We reversed irrigation bipolar forceps and used that channel to suck smoke. And the smoke that you produce by electrocautery is already partially charged. And that will go into that atmospheric interface, which has to be specially designed so you don't contaminate it as you heat the components up into that mass spectrometer, which will then produce a spectrum of molecules or clusters of molecules coming from that smoke, coming from that patient. And this slide illustrates on the bottom there a spectrum from the tumor itself, the tumor bulk, and then the one above from the periphery in the white matter and the periphery towards the gray matter. And they are all slightly different. And just to show you, there are at least 4 or 5 major differences, which if you put through a mathematical modeling will differentiate those two and identify them against the classifier. So the way that we build a database up using this technique is that we take the sample, acquire the spectra, we correlate, we validate that histologically, and we store that in a database. And in that database, it will undergo multivariate modeling, mainly principal component analysis, lead differentiate analysis, but some other algorithms, which will then allow that machine to identify the smoke coming from a similar type of tumor and classify it according to and it will basically learn from that database. So when it's exposed to smoke from a GBM, it should recognize it. And here is an example of that. Here is the screen printout. We're working on a GBM. We're acquiring spectra. We're sucking the smoke from the, and there it is, identifying it as a grade 4 tumor to 94 and now 95% specificity. So that's what that can achieve. And you can also differentiate it out and spatially, and you can look at where these samples are taken from, and here is the region of tumor. You can see there's a bit of variation in terms of the signature that you get, and this represents the heterogeneity that you see within these high-grade tumors. And you can see that it will also separate out the different types of tumor. But one thing you'll notice is, although the region of normal gray and white matter are separate, there's only few of them. And the reason for that is that we can't ethically go into normal brain tissue and destroy it for this purpose. It's a destructive technique. So we wanted to find an alternative which was more Star Trek-y and was non-destructive and hopefully a bit more robust, because actually that Reams technique, that evaporative mass spectrometry, was quite variable and quite a dirty technique, and it's much better with laser than it is with electrocautery. So we opted for Raman spectroscopy, and Raman spectroscopy again is an interaction of light and photons with the electron cloud of molecules, such that when light hits the electron cloud, a photon hits it, most of it will be scattered and it won't change energy state, won't change frequency, but a small, very, very small part of it will either impart or lose energy and change the vibrational status of that molecule, not its electron ground status, but it will then change frequency. So if you can pick up that frequency change, that frequency change will be specific to that molecule. So if you can filter out the incidental light and pick up those frequency changes, you'll end up with a spectrum of different frequencies representing the spectrum of different molecules within your target subject matter. And we wanted to prove this first in man, and we did. This is the setup. There are problems with it because the amount of Raman signature you get back is something like 0.01% of the incidental light. So you have to filter out all sources of infrared radiation in the environment and use LED displays and LED light. But it is achievable. And because we showed this back in 2015-16, the BBC got hold of it.
Shines a near
and we're
infrared
quite
light
excited
onto
about
the
it.
brain. The pattern of the scattered light which bounces off differs between healthy and abnormal cells, so surgeons know whether to cut or spare the tissue.
So there we go. So I'll come on to why that was exciting in a second, but the workflow, because we combined the two techniques of iKnife and Raman, were that you identified your sampling point, your tumor sampling point. You did your non-destructive sampling with Raman. You localized it on your 3D ultrasound, your MR navigation system. You then did your iKnife sampling, then you take the biopsy, and that way you've got a streamlined workflow. With the spectroscopy, you can see the unsubtracted signal above, that big curve. That's a fluorescent signal superimposed with the Raman signal. If you take away the fluorescence, you get this very nicely defined peaks and trough Raman signature, which correlates to the various different molecules within that system and that tissue. And that actually enables us to do some differentiation. So the first question was, could we differentiate between tumor or non-tumor and normal surrounding brain? And we could do that to our heart's content because it was a non-destructive technique. And we found that we could do that very nicely, again, to a 95% specificity. And the green curve shows the average normal, and the red, the average tumor, and the blue curve down below shows the number of differences, and the red dots are where those differences are significant to P being less than 0.05. And we can also differentiate the tumor types. Here's a low-grade astrocytoma, again, different from normal brain, and a GBM. And you don't have to actually put this through any analysis to see the difference between the grade 2 and the GBM. In fact, the normal brain looks more like the grade 2 astrocytoma. And you can do spatial differentiation as well in terms of defining the normal brain brain tumor interface. What this demonstrates is that there are a couple of points here, the S14 and the S4, which were taken from the borders or the tumor resection margin bed, the margin, if you like. And when we looked at histology, this one that's well within the low-grade astrocytoma area, the tumor area, as opposed to the normal brain area, was actually had some gliosis, but did actually have quite a lot of tumor there. This one, S14, was predominantly normal stroke gliatic brain, but there was single cell infiltration, and the system picked that up, which was quite sensitive. So this is an indication of the sensitivity of this technology. Now, we want to put it through its paces and see whether it could go further and identify against other classifiers. So identify the molecular fingerprint in relation to common mutations that would normally take a few days at least, or if not a week or so, to get something back, or at least immunohistochemistry now. So we looked at IDH mutations, and we found that there was a clear difference, and this could be clearly identified, along with ATRX and MGMT status, to the point of 98% specificity for MGMT and 94% for IDH. And when we looked at it, obviously IDH mutations are associated with the accumulation of the oncometabolite 2HG, and you've seen from Sotiris's talk about the MR spectroscopic manifestation of that. We can see that this forms a large part of that signal that differentiates the IDH1 mutant subtype. So when we combine the technologies, we see that we could separate out very quickly the high grades and the low grades and the outliers of the oligos, which are much rarer. But the grade 3s, they would span both those high grade and low grade phenotypes. And that is because these match the molecular phenotype much better than the histological. This is based on histological classification, which we know is inherently inaccurate. And these now that we're going to a molecular classification of tumors, this is a much closer representation of that. And this was data that we'd acquired prior to the latest WHO change in classification. So this is a very accurate way of molecularly defining the tumors. And bear in mind that this happens we can get this data within seconds. This will definitely potentially shorten the diagnostic pathway of these patients. But is this benefiting us in any other way? Is it enhancing precision neurosurgery? So here are some of the questions we wanted to be answered. Can it provide a molecular profile of the tissue in the live patient during surgery? Well, we can see that it can. Can it detect the difference between tumor and normal brain? We think that it can, and we can see that it can. Can it detect the cell density better than any other modality? We've seen some evidence, yet to be fully proven, that it can detect up to single cell infiltrations. Could it identify significant pathway aberrations, mutations? Well, again, we can see that it's very sensitive at doing that and giving us a molecular profile. And we hope that at some point we can get a readout of the molecular phenotype that close to something like next generation sequencing. Could it determine the tumor's likely behavior, well, if in response to treatment and beyond? This is something that needs to be answered, but if it can do what it's done before, potentially yes. And if it can, then it potentially will be able to inform the surgeon's decision whether to fully go for full resection. And the classic example for that is the melanoblastoma in children, whether they've got the sonic hedgehog mutation or not, and whether you should chase the tumor or not and run the risk of neurological deficit. But even with IDH1 mutation, it might be, if you're in an eloquent area, a decision to say, well, let's hold back and avoid going too far because we know that they're going to respond better. But the most important thing in terms of precision neurosurgery and precision medicine is where we're heading, is understanding the molecular phenotype of that tumor will allow us to therefore bespokely give the correct molecular treatment, molecular solution. And that's the subject of another talk, but we'll come on to that in a little bit. The other thing from a research point of view is that, is this not the best model for studying brain tumors? We take tumors and culture them in the lab and analyze them in various models. But here we have the live tumor and the live patient with a way of interrogating the molecular phenotype of these tumors. And as Raman technology moves on with the various different types of it, there is, and the increasing use of high iteration lasers, that you could potentially study chemical reactions in real time. However, could it detect the nature and function of normal tissue? If it can detect differences in pathological tissue, perhaps it could detect differences in the nature of normal tissue, but we haven't proven that. And the problem with this is it's a point analysis so far in our iteration of it. It doesn't give you a field view. There may be an answer to that incorporating other optical spectroscopic techniques, namely hyperspectral imaging and fluorescence, which I mentioned before. So hyperspectral imaging is basically a multispectral image that captures the image data, i.e. the absorption stroke reflection pattern of the tissue or the matter from a single frequency. And what you then do is you layer up the same pattern in an image cube by shining different frequencies, one on top of the other. And in that way, you get a composite, multi-parametric, if you like, signature of that tissue. And that can be done by using sets of filters that filter out all other frequencies when you're shining white light onto a subject matter, or through using detectors that just detect specific frequencies. And it's been used already in other arenas, particularly when studying the cosmos and the sun. When you filter out the different frequencies of the sun, you can see different aspects of its chemical composition, its gaseous composition, etc. And when you look at multispectral imaging of agriculturally, you can see how well your crops, for example, are irrigated and how well they're doing. We cottoned onto this because we wanted more of a field change, and our colleagues in the field of general surgery, laparoscopic and endoscopic surgery were characterizing it to look at the features of the different organs and see if they could see characterization of normal and pathological tissue in this darkened arena of the abdomen with the laparoscope. And the promising results showed, I won't go into the model of this is actually a graph model of the sheep uterus, but it shows you that which is really exciting for us and me, is that you can actually detect the differential levels of oxy and deoxyhemoglobin in tissues by using this technique. And you can see, going from left to right, you can see the well-perfused donor, the less well-perfused recipient, and the very poorly perfused graft, which has not got a good blood supply. But theoretically, what we thought, if we could apply this to neurosurgery and brain mapping, prove the concept that we get a field of view of optically enhanced images and obtain multispectral signatures, which will delineate normal and maybe pathological brain, but more importantly, will show us perfusion, blood vessels, microarchitecture, but even more importantly, potentially an optical version of BOLD, functional MRI. Ultimately, if we get this down to a fine art, potentially give us an insight into functioning brain in real time at surgery, which would be the holy grail for the neurosurgeon. we've got the setup, we've got the camera, we've got it all ethically approved. We just need to start the process, which has been hindered by COVID. So in conclusion, we are moving forward with this, and it has potentially huge clinical applicability. But can it provide any more solutions? I mean, one of the things that I discussed in previous talks was rather like you're forming an atlas, a functional atlas from awake craniotomy stimulations. Can we now, with the data we've got, start to form a spectroscopic atlas based on optical, molecular, and functional data, and then develop a real-time optical identification system? This kind of thing, because it's optical, does lend itself to technology that's already out there, and we've already seen from Stanford.
Sorry to interrupt. You have another minute, I'm afraid.
Okay, that's right. I'm just so enthusiastic. That's the end of it. So the last slide is basically that, you know, this is heading towards precision medicine or precision surgery, because it's really showing us that we can use this technology to molecular profile and improve our diagnostic and prognostic pathways. I always use this slide at the end because it's all about matching what we achieve with patients' expectations. Thank you for your attention.
Thank you very much, Kevin. That was amazing talk. And as you said, this could be the holy grail of neurosurgery, to combine these technologies together. So we are very grateful for your talk. That was slightly over 20 minutes. And people who want to ask questions, please feel free to type the questions, and Kevin O'Neill will be happy to answer them in writing. So moving on, I would like to present my good colleague, Andrew McEvoy, a consulting neurosurgeon and recently professor of neurosurgery at UCL, who's got a very extensive experience not only in tumor surgery, but also in epilepsy surgery. And he has the biggest epilepsy surgery practice in the UK. Andrew is involved very actively in research and also in new technologies. And today he will discuss robotic brain surgery and also technology-related advances. Andrew, thank you very much for joining.
Thank you very much, George, and wonderful course, great day, fantastic. Good evening, everyone. So I'm going to speak this evening about two projects that we've been working on over the last 8 years or so, EpiNav, which is a navigation platform, and a robotic platform. And I hope you'll see what happens when a multinational commercial organization gets hold of one of our ideas and what it can lead to. And as George says, I'm primarily a brain tumor and epilepsy specialist, and I'm speaking to you from central London, from Queen Square, which has been central to brain tumor and epilepsy. Svitman Godley, who performed the first operation anywhere in the to resect a brain tumor, was born in Queen Square, a future president of the Royal College of Surgeons here, and the founder of mine and George's unit, Sir Victor Horsley, was the first man in the world to perform an epilepsy surgery operation, which he did here in this hospital in 1887. And this is the hospital on the corner of Regent's Park, about a mile from where I'm speaking to you, where the first brain tumor operation took place. And this is Horsley, who worked, of course, with two fantastic neurologists, Hewlings Jackson, one of the fathers of epilepsy, and David Ferrier. Now, Horsley himself was a man of many firsts. As I said, the first man to do an epilepsy surgery operation, the first man to do, he was doing awake craniotomy, he was taking pituitary tumors out, spinal tumors out. But this is what is going to be the crux of my talk tonight. He was, along with Clark, in 1906, the first person to invent a stereotactic frame. And this is Tippu Aziz, the professor in Oxford, holding that frame, which was used for animal experiments to accurately get around the brain of monkeys. And that sits in the Science Museum here in London. And you'll know, I think, that certainly during my career in surgery, we've been using stereotactic frames to accurately get around the brain. This is the original Talairac frame for stereo EEG, and this is the grid that the electrodes were put through. But there are all the problems of frame-based surgery with restriction of access, the problem of having to transfer patients just... Sorry about that, everyone, that's not from here. And we've all moved over the years onto frameless procedures. And this is an example of a frameless stereotactic procedure going on. This is us performing stereo EEG. And This is Horsley operating, but now, of course, what's happened with surgery over the last 50 years, certainly in stereotaxy, the workflow has not really changed. We're still left with the problems of image fusion. We're left with all the problems of brain shift. We're left with all the problems that you're operating on patients in different positions that they were imaging in. And often you're using images, most often you're using images acquired pre-surgery. But now with intraoperative imaging, while we're operating, you've got the facility to do automatic registration to reduce these errors that we have in our surgery that lead to complications. So these automatic registration systems will give submillimetric accuracy in the registration. And it's becoming very commonplace now to have mobile scanning units where you can position your patient in theatre and simply bring the patient into the room and bring in a scanner and automatically map the patient to your navigation. So what surgeons have been looking for is to see whether that safety and that accuracy can be increased even more with robotic platforms. This is being used across surgery. In the UK now, more prostate operations are done with robotic aids than are done open these days. And I think it's widely accepted that robotics is better than human when it's a space-limited procedure, when you need it to be highly accurate, when it's repetitive and tedious and time-consuming, and when it's intensive and requires a lot of concentration. So a lot of surgeons are looking at robotics. And you can easily see the sorts of procedures we might translate this into, whether this is biopsies and tumor biopsies. You've had a lot of this today, ablations, and we're using increasing lasers and things to do that, catheter placements for drug delivery, stereo EEG, which I'll use as an exemplar in this talk, and stimulators and a variety of things such as DBS for movement disorders. Now, there can't be a field of surgery or fields of surgery outside of tumor and epilepsy, which lead to more multimodality imaging, and you're hearing so much about that today, whether that's structural imaging, vascular imaging, SPECT imaging, looking for the source of seizures, or PET imaging, looking for the source of seizures or the activity in a tumor, DTI and fMRI, looking to make the surgery safer, MEG, looking at function, EEG, fMRI, again, looking at seizures or electrical source imaging. And I've left the dates on these scans because you can see the fMRI focus here relating to this tumor of motor hand and motor leg. And we've had these static pictures for a long time. This is the optic radiation in relation to this tumor. But the question to all of us as surgeons is how we can use this data operatively. How can we get all of the imaging data that's been so eloquently demonstrated today into the OR to help our patients and improve the outcomes of our surgeries? So I'm going to talk to you about EpiNav, which is a navigation package that we've been developing here over the last 8 years at Queen Square, and how that's used in neuronavigation and how it's been integrated into a robotic platform, and hopefully demonstrate how we've taken that forward. I'm going to use the exemplar of stereo EEG. For those of you in the audience who don't know what that is, it's a way of implanting multiple bolts into the skull of a patient with epilepsy. You put electrodes deep down into the brain with multiple contacts on, and then you wake the patient up and you record where their epilepsy is coming from. It has a number of advantages. It's a closed procedure, so it's very well tolerated. The patients are very well with it, but it's a blind procedure. And so it's clearly high risk. You're passing multiple electrodes deep into the brain, and you need to do that safely. These are elective operations. So I'm going to go through, essentially in this talk, 2 of the central features of the stages of this, the planning of it and the surgical procedure. And I'm not going to talk much about postoperative imaging today. We need to be using patient-specific data. You know, in the old days when we were doing stereotactic procedures, we were using the data that was acquired from atlases and dissections of cadavers. But nowadays, we can have beautiful patient-specific data. Look at the cortical extraction on this picture here of a scan. And this is a representation of where the surface EEG electrodes were in relation with the stereo EEG electrodes. Look at the vascular anatomy that's demonstrated when that can be morphed onto the cortical parcellation. I mean, beautiful imaging. And of course, as you subtract away the cortex, you see down here just how difficult it is for us to plan these electrode trajectories safely. It really is trying to direct your electrode through the eye of a needle. But what I'm going to demonstrate to you is how this software package has been developed, which along any particular trajectory will give you a risk profile as the electrode proceeds into the brain. It doesn't need to be an electrode. It could easily be a catheter for a shunt. It could easily be a biopsy needle for a tumor. Look at the representation of the venous anatomy. I'll show you the bridging here of the veins mapped onto an operative photograph showing exactly the same picture that you see here. So this has been the result of a number of years' work and 3 very large program research grants. And the first grant essentially allowed us to develop the platform to basically upload all of these different imaging modalities into a platform that we could interact with remote from theatre. So we could plan these trajectories in the weeks coming up to operations. We could find the safest trajectories with the minimal number of electrodes to cover any particular plan. You'd get a better spatial understanding of where the epileptic focus sat in relation to the motor strip or speech areas or the corticospinal tract. You might choose to do your definitive operation in a different way depending on that result. Maybe do it awake, maybe do it in interventional MRI. But when we went back to get the second grant after 4 years, we wondered, could you teach the computer to automatically find the safest trajectories? I mean, teaching it all the rules to avoid the blood vessels, find the easiest drilling angle through the bone, find the greatest gray matter contact of each of your electrodes. Could it be taught to do that in a matter of seconds? Anna Mizrachi, my surgical colleague here at Queen Square, and I would take 6 to 8 hours to plan these cases. What if the computer could do it in a matter of seconds? What if you could put those plans into practice in the OR on a robotic platform to sub-millimetric accuracy, again, much quicker than we were doing before? Now, I'm not going to talk very much about the other aspects there about visualizing the spread in the software, but I hope you can see the wide applicability of this to all forms of cranial neurosurgery. This is a single electrode just demonstrating the pathway here and what happens. And as it scrolls down, this electrode, you can see it gives you a risk profile along of how close you get to any particular vessel as this electrode goes deep into the brain. And clearly, if your registration is not very good, you would adjust the acceptability of how close you want to get to any of these critical structures. And if you think about it, we're doing the same thing over and over again, often in neurosurgery, especially doing these sorts of biopsy procedures and stereo EEG. You're using exactly the same rules over and over again, and it lends itself to automated planning. You want to find the avascular trajectories. You want to find the easiest drilling angle through the bone. You want to avoid the peel boundaries, avoid the ventricles, have the greatest gray matter contact. You don't want the electrodes touching each other in the brain. You want to be able to export these plans easily into your navigation package in the operating room. And if you think about it logically, if you choose a target deep in the brain and you limit the way you can attack that target by restricting or with all the rules I've just given, there's usually only a limited number of places it can come in from. And clearly, if you're putting multiple trajectories into the brain, the computer can work out all of the risk much greater summed together than any surgeon can. So this is an example of how the package works. And you can see here that this is the interface. You're going to see a series of targets here. These actually are electrode targets, but they could be tumor biopsy targets. It's clearly on the left side of the brain. So you want to restrict the access so that these trajectories can only come in from the left side. So you give it a mass. And then you simply add all of the imaging structures that you want to restrict the access to the trajectories. So you put in the veins, you put in the arteries, you put in the peel boundaries, you put in the ventricle, you put in whatever you want the computer program to avoid. And the program will automatically segment the structures. So when we're doing epilepsy, we're often choosing specific targets that we want to hit, such as the amygdala or the anterior third of the hippocampus or the middle third of the cingulum. It will automatically segment those so that you can just say to the program, look, I want to hit the inferior frontal gyrus and I want to hit that from the superior frontal gyrus, and it will automatically find the safest trajectory to that target with the easiest drilling angle. And when we looked at the results, it became obvious. This is in blue, the amygdala, in red, the hippocampus, in yellow, the middle temporal gyrus, that we were nearly always, the surgeon has his or her preferences. So they tend to always come in from the same area. And if you choose, that happened with all of our targets to all of our structures. So if you were looking at wanting to hit the posterior cingulum, we were nearly always coming in from the supramarginal gyrus. If you wanted to hit the anterior cingulum, you were nearly always coming in from the middle frontal gyrus. And the computer will learn that. It will use a machine learning approach to learn the safest trajectories and any particular surgeon's preference. So as I said, we can do this very safely in a frameless way. I mean, we get all this data from the EpiNav package into the OR, but how do we do the operation safely when we've got all of this data? How do we actually do the operation? Well, we can do it in a frameless way, and that's indicated here. This is the stealth system, the precision aiming device. And we've shown and demonstrated with a series of publications that the workflow of this is very safe and it can be done very accurately. But are there more accurate ways to do it? So certainly I think epilepsy surgery leads the field in neurosurgery at using robotic platforms for these sorts of operations. Now, there are some fantastic solutions out there on the market. This is the Renshaw robot. There's a Rosa robot. These are very large pieces of equipment, as big as fridge freezers in the OR, but they're very accurate, they're very robust, and they're very good. But we wanted to see if it was possible to develop a different robotic platform to achieve the same sorts of results. And we were very fortunate to be given the opportunity to use a very small robotic device which had been developed in Vienna, Austria, at the University of Vienna. And two surgeons, Christian Dorfer and Stefan Mulzberger, had developed this and they'd used it. This is Stefan Mulzberger using it for a stereotactic biopsy. And you can see, compared to the other thing that's a fridge freezer size, this is the size of a paperback book. And it's been run off a standard commercial Medtronic navigation package. And you can see, it's a very appealing piece of equipment. And we were able to get access to this. And they demonstrated in some pilot work that in stereo EEG and in tumor biopsies, you could actually find the entry point and the target points of your trajectories 60% more accurately than you could in a manual way, and you could do it 20% faster. So this is the workflow that was used, and you can see the great advantage of this particular software package and this robot is that it's using things that we use all the time, the Medtronic standard navigation, the Mayfield pins. We're all used to using these Medtronic arms for navigation. The robot itself is two platforms that slide over each other. It gives about 30 degrees of freedom in each direction, and it works off the standard Medtronic Vertex probe. So we were all used to using this sort of technology, which made it very appealing. They developed a series of surgical adjuncts to make it more accurate. So they developed a technique where you passed a K-wire in through the skin and into the bone, and then introduced an oversleeve which tapped into the bone to secure the device before you drill, to stop drilling, to stop slippage on the bone. And a technique where the electrodes were also passed once the little screws were placed down exactly the same trajectory. They compared the precision aiming device that we were all used to using for the Medtronic system, and they compared it with this robotic device. And you see here the result of that there was a significant increase in both the target and the entry point, how accurate it was, and it was significantly quicker. And this shows the scatter of that. This is the entry point and the target point manually, and these are the entry points and the target points with the robot, which were statistically much more accurate. So we were lent this device to do a randomized controlled trial. I'm not going to go through the details of this workflow, but just to say that for our registration, we used bone fiducials, which were implanted into the skull in both arms, the precision aiming device arm and in the robotic arm, and then a preoperative CT, and then we fused all the EpiNav data to that. They were well matched, these patients. There were 16 in each arm, and equally well matched in different types of epilepsy, 328 electrodes in total inserted. And this is the workflow. So just to go through this, what you needed to do with the robot is put it roughly, you'll see this in a video in a minute, roughly in the position of where the target is, and you lock the arm. Then you simply allow the robot to find to submillimetric accuracy the trajectory. You then tap the device through the skin. You put the securing device for the drill into the bone. You then drill, you place the screw down exactly the same trajectory, and you can either put the electrodes down through the robot itself, or you remove the robot and put the electrodes in later. And so this is Anna and I doing one of our first cases here. And you'll see where this blue target here, you just need to be able to lock the arm so that it sits within the green circle, the blue circle. And you can get to different trajectories if they're in about a 4 centimeter square area. But you can see once you've locked it, it takes just a matter of seconds to lock onto that trajectory to submillimetric accuracy. You then just tap a little divot into the bone to secure your drill. So it makes a little indentation in the bone. You then drill down into that divot, and everything is maintained in the same line, the same trajectory. Then down exactly the same trajectory the bolt is going to go. And then, like I said, either you remove the robot and put all of the electrodes in, or you can pass them through. And here's us finishing off the implantation. Now, I'll come on to this in a second, but the primary endpoint of this study was to demonstrate that it was quicker, and it was significantly quicker. It was 3 minutes quicker for every single electrode to implant here. So there was a 30% time reduction per electrode in implanting with this robotic device. But interestingly, the accuracy overall was not improved. And I think there are a number of reasons for that. It was very similar at the entry point, and it was slightly worse at the target. And clearly these electrodes do bend in the brain and can move in the brain, and that might have had something to do with it. But I think overall, probably it just demonstrates that Anna and I had done thousands of these electrodes with the precision aiming device, and it shows the learning curve of surgeons with new technology. We'd optimized all the protocols with the precision aiming device. We got exceptionally good. We were using trajectory guidance on the stealth machine, so we were using guidance of both the entry point and the target, as opposed to just the traditional target guidance. And I think another aspect of it was that we were, and something that you'll see again in a minute where this is being developed, is that we were able to use tracking, which you can see here, on the device that we were drilling through all the way in the manual cases, but not in the robotic cases. And so we did check just to make sure that we weren't, we hadn't making errors here, systematic errors, and we were well out compared to other published studies. So we compared it with a systematic review and meta-analysis of all the other trials in the field. We looked at other robotic trials, other frameless trials of stereo EEG, other frame-based trials, and you can see these are our results here, well within the range of the accuracy of the standard trials. So it was certainly accurate, it was quicker, and just that this is that was the entry point, this is the target point. Now, many of you will know, of the surgeons in the audience, this has clearly been now taken forward by one of the major manufacturers in the field, Medtronic. This is what that robot has become, the AutoGuide. You can see here the use of the O-arm so that you can automatically register this patient in any position in the operating room. It clearly has all the interface that's standard with the stealth machine. It's got all of its own tools now, as you can see, all developed by Medtronic. Some great advances. This particularly, you can see that now the robot is handheld by the consultant who just looks at the interface on the device itself. So you control it yourself as the single surgeon. Can you see now that the surgeon, when he's drilling, actually still can track the robot, which is what we couldn't do in our preliminary study. You can see all these lightweight drills that have been designed that just use like a pencil to go through the bone. I mean, beautiful equipment, and a tiny surgical footprint in the OR. This is actually showing a little bolt being put in for the laser. Stereo EEG, the laser, catheters, whatever that might be. And you can see what these prototypes have now been developed into. So just a couple of last slides. I think there are some good developments that the commercial companies put into this, especially that it can be navigated while you're drilling. These lovely lightweight electric drills will be fantastic. It'll stop slipping on the bone. It'll stop the pressure on the device when it's working. And it comes with a beautiful now gauge so that you don't pierce the jaw and go too deep in any particular moment in time. I think these are all fantastic advances. We've just been given now the proper auto guide commercial robot to run another trial, and we're going to trial that doing our stereo EEG and doing all our tumor biopsies. So myself, Anna and George will be doing that over the next couple of years. And on that point, when I've hopefully demonstrated how we've been involved in the commercialization of that product, I'm going to say thank you very much.
Thank you very much, Andrew. That was an excellent review of the recent advances in neurosurgery, and I'm very glad that you and Queen Square is at the forefront of these advancements, so good job. Again, due to lack of time, we have to move on, but please do send your letters to Professor McEvoy, and he will respond in writing. Now, moving on to Edward Chang, who is Professor of Neurosurgery and new chairman at the UCSF, the University of California, San Francisco, taking over the department from Mitchell Berger, a legend in brain mapping, who will be with us tomorrow. But Edward Chang is also extremely talented, very knowledgeable, very, very intelligent. He has published very extensively in Science, Nature, Cell, Nature Neuroscience, Neuron, and he has a quite unique understanding of language, function, and testing. What we have asked Eddie to present today is his experience with maps produced from direct electrical stimulation. Thank you very much, Eddie, for joining us.
Can you see me and hear me?
Yes.
Okay. And you see my slide here?
Not yet.
You don't see my slide.
Let me stop Andrew sharing. Okay, can you try again, please?
How about now?
I'm afraid not yet. Just give me a second, Eddie. Yeah. You're on. Thank you very much.
Okay. You see my slide, not the navigator, right?
We can see your slide.
Okay. George, thank you so much for inviting me to be part of this fantastic course. You've really lined up an amazing group of speakers, and really honored to be part of this. So not enough time really to address mapping and functional mapping in brain surgery, too big of a topic. So I thought I'd focus on some new work that's unpublished and share it with this audience. Really excited about this new work. It does have to do with brain mapping, a combination of both physiology, stimulation, and resection, a multimodal approach to understanding the functional organization of the human auditory cortex. Many of you know a lot of our work relies on electrocorticography recordings. These are electrode recordings, typically in the setting of epilepsy monitoring, that we use to localize seizures and do brain mapping. And historically, most of our work is really focused here on the lateral superior temporal gyrus and also some of these speech areas in the suprasylvian cortex. Over the last 10 years, we've had some really unique opportunities to actually study the extent of the auditory cortex, the entire extent of it, as it relates to areas that are within the sylvian fissure. So in certain cases, about 12 that we've studied over the last 10 years, we've had indications to approach the insula or the temporal plane for localization of seizures where there was a hypothesis of seizure origin there, which could not be well sampled from the lateral cortical surface. And this shows a rendering of the electrodes that we can place laterally, as well as within the sylvian fissure, covering Heschel's gyrus and the planum temporale. And one of the basic questions that we wanted to address with the scientific questions was really just trying to understand the information flow that goes through the auditory cortex. Previously, there's a discussion of streams, in particular the ventral stream, but the auditory cortical processing. And a lot of the classical way of thinking about this is that there is a feed-forward serial processing of sound information. So many of you know the auditory thalamus is the medial geniculate body. And that this projects to the A1 core auditory cortex. that's located in the posterior medial aspect of Heschel's gyrus. It's not the entire Heschel's gyrus, but in fact, the posterior medial aspect of it. And there's this basic thought that this projects basically to the superior temporal gyrus, which is known as the belt and parabelt auditory cortex. So this is the standard classical model of auditory organizations, heavily inspired by visual processing, not so much by brain anatomy, as I'll explain later, but something that, pretty much 99% of how we think about the auditory processing. This is an illustration. So the core auditory cortex, as I mentioned, is in the posterior medial temporal plane, the very back medial corner of Heschel's gyrus. And here are some of these other acronyms: the planum temporale, the planum polare, the anterior superior temporal gyrus, and the posterior. And there's a reason I'm making a distinction between the posterior and the middle or anterior STGs, because it has really dramatically different functional properties. So for the neurosurgeons in the group, this is not anything new, but to get electrodes in this area safely requires a lot of peel dissection. Obviously we can't just slip electrodes in there. We actually have to dissect this space. And these are under very unique settings. And when we do this, we can put something like a high density array, in this particular case, it's a 32 channel mini grid from the temporal plane to record from all of the medial areas, and then in addition, simultaneously from the lateral, to get complete coverage of the human auditory cortex. So one of the first things that we studied, and again, we're looking at this basic question: to what extent is there a feed-forward serial organization of sound? And there's this idea that in the medial part of the core auditory cortex, it goes from a simple sound organization, kind of like we have in the cochlea, which is tonotopic, organized by sound frequency, and then this gets converted over this temporal plane into sound elements, the phonological representations of speech and other complex sound processing that's involved with music and whatnot. So the basic idea in the serial feed-forward model, again, very much modeled after the visual cortex, is that it goes from this medial to lateral. So one of the key and easy things to look at is the latency. If you look at the timing of activation when people are listening to speech across dozens of electrodes recording from the temporal plane, both medial and lateral, what we found was not a steady progression of sound latency, response latency from medial to lateral, but in fact, we found that many areas of the posterior superior temporal gyrus, in fact, had very, very short latencies, just as short as the medial Heschel's gyrus. And you can see this here in the posterior STG, the latencies are less than 100 milliseconds, as they are in the posterior medial Heschel's gyrus. Also extremely short latencies in the planum temporale. So the idea here is that this whole posterior quadrant of the human auditory cortex has very short latencies. It's not suggestive of something that is happening serial, but something that's happening almost in parallel. And then as time goes on, you start to see activations of the anterior lateral Heschel's gyrus, some of the slower latency responses in the surrounding parts of the posterior superior temporal gyrus, the middle superior temporal gyrus, and the longest ones are in the planum polare. So these are where the different anatomical areas are outlined in the latencies of responses. And again, the conclusion from this is that the earliest activations occur simultaneously in the posterior auditory cortex across the core A1, but also non-primary auditory cortex and the lateral quote-unquote parabelt areas in STG. This is a map basically of time, latency time. And the shorter latencies are purple, the longer ones are green. You can see, again, the back part of the auditory cortex is where you have the shortest latencies that are nearly equivalent, and more anterior areas are longer. This is more consistent with more of a posterior to anterior flow of information. But it's not as simple as that, and I'll explain that in a second. Here's some correlation with elements of the functional myelination that was described by Dr. Van Essen in the Glasser paper. As he described, looking at the T2 signal, you can actually derive the the myelinated cortex using MRI. In their studies, as well as we validated this ourselves independently, has observed that it's not just the core auditory cortex that's heavily myelinated, but in fact, this whole posterior area of the superior temporal plane. And this is why we think that it has very short latencies, because it's a heavily myelinated part of cortex. Some more things. So we characterize the difference in activation to these areas to speech and to pure tones. And what you can see is when you record from Heschel's gyrus and the planum temporale up here, you can get these very nice, well-tuned, what we call auditory receptive fields. So basically, this is sound intensity as a function of sound frequency. And this is an example of 3 electrodes right next to each other that have tonotopic organization. It's going from low to middle to high frequency. And we use a method called a spectrotemporal reverse correlation method to estimate receptive fields from speech, looking at the frequency content in speech, and this matches what you get with pure tones very well. So these, again, are tonotopic from, but listening to different stimuli. What you see when you record from the superior temporal gyrus is completely different. You can see that this part of the brain does in fact have very strong responses to speech, but they're not very well tuned to simple pure tones. And when you estimate the receptive fields using reverse correlation, you can see that they're broadly tuned to spectrum in very complex ways. And on top of that, the responses are much stronger to speech as they are to pure tone. So tones versus speech are shown for each of these areas, including the planum temporale, the core auditory cortex, and the posterior medial Heschel's gyrus. You can see it largely have similar responses to tone and speech, but in the lateral areas, there's a huge divergence that becomes much more selective to speech. This raises a simple question. Given the latency and the selectivity that I've described here, is this evidence of serial processing, meaning feed forward, where one area is passing on information to the next, or are these independent parallel inputs? And the next set of results I'm going to show argue for more of a parallel story. So one thing that you can look at outside of just the timing and the selectivity of different sounds features, stimuli, is to look at actually what aspects of speech are actually being encoded. And so if you take the auditory waveform for a sentence like this, it had gone like clockwork. This is the acoustic spectrogram, frequency as a function of time. And you can label all of the different, what we call phonetic features. These are the subpart of all the consonants and vowels that linguists use to describe the different consonants and vowels. There's only about 12 of them that give rise to all the consonants and vowels. And then also other acoustic attributes, things like the pitch, the absolute pitch, the relative pitch, which just means the pitch that's normalized by the speaker's voice, and also a derivative of the pitch, which is essentially the pitch change. And these have different relevance in terms of processing sound. And what we found when we took this, what we call encoding model approach, whereas we're trying to look at each one of these electrodes that we've reported from the temporal plane and STG, we've tried to figure out which electrodes are tuned to what sound properties. And what you find is that the areas on the Heschel's gyrus, not surprisingly, are tuned to things like absolute pitch and pure tone frequency information. But out here, you start to get much more complex features, like these areas out here are not sustained, but are highly responsive to the onset. And because of that, they only respond to the onset of a sentence or a phrase. And because of that, these cannot be inputs to these other areas that are coding things like these phonetic elements. These are the things that are the elements of consonants and vowels that are represented more laterally. So this is a more distributed representation of all of the elements of sound and speech, where the medial aspect of the auditory cortex is highly tuned to simple sound processes, where its lateral part is complex. If you look at the response properties, some of these cannot feed this because of the representational content. I'm not going to get into that too much here, but one way that you can essentially look at this causally is to apply stimulation to these parts of the brain independently. And what I'm going to show you in a video, but also in the summary data here, is that we found a double dissociation when you stimulate from the medial and lateral auditory cortex. So our hypothesis is that if this was like a serial processing system where information was going from medial to lateral, that if you stimulate in the medial heschel gyrus, that you would change what people hear in terms of the sound quality, the words, maybe the words sound distorted, or that you may interfere with it. But that's not what we found. We found something that was quite different, in fact, a double dissociation. So when you stimulate in the medial heschel gyrus, you evoke a sound hallucination. People will hear a sound, and almost universally, it's contralateral to the side of the stimulation. And interestingly, and we tested this quite carefully, there is no effect on speech perception whatsoever. There is no distortion of the speech quality. People hear it superimposed on top of the hallucination. That is to say, they hear both the artificially evoked sound from the stimulation on top of what appears to be very normal speech perception. And then what you see out here when you stimulate in the lateral STG is something exactly different, which is you cannot typically evoke sound hallucination. And then number two is that you have a very distinct and robust and very repeatable impairment of speech perception when you stimulate on the lateral STG. So this is just a video, and it's going to show you some examples of stimulation. This is in the setting of epilepsy patients that were implanted with either these electrodes over the temporal plane or with depth electrodes to heschel's gyrus.
I heard a sound of I heard a sound of blah, 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 type thing over here. Yeah. Here's what it sounded like waves.
So I'm gonna, for the sake of time, I'm gonna fast forward to different part of this, which you can see that okay, here we're testing comprehension. When you're stimulating, can the patient comprehend what you're saying? Is there any change in the sound quality?
Busybody. Creativeness. Ukulele. Yes, I was able to hear all of the noise, values.
So patient hears the evoked sound as well as what the person is saying.
Biographer. Applicator. Exhalation. Watercolor.
So you hear the sound, but you also hear the sound. Do the words say sound to you, or they just you hear the voice?
Sometimes the sound is a bit overtaking you when it comes to words, but besides that, I can hear you both quite well.
Okay, and then when you stimulate in the left STG, in the parabelt, at rest, basically, patient has no perception whatsoever. No conscious perception of stimulation.
Asparagus. Sounds, I'm sorry, I couldn't really hear you. Cotton. Only a bit, but not very long. No. So I have like a caterpillar. Yeah. Mutation. Okay. Mutation. Something like that. I heard some echo.
So these are just some video examples of this dissociation. Again, when you stimulate the medial auditory cortex, it really has no effect on speech perception. And the last two things that I want to show you are that if you ablate the primary auditory cortex in the Heschel's gyrus, it basically has no effect on speech perception. This was a case that one of my friends at Montreal Neurological Institute shared with me. It was a patient who had stereo EEG and then basically isolated the seizures to this area. While he was awake at the bedside, did radiofrequency ablation of the Heschel's gyrus while the patient was awake doing language tests, and there was really no effect on his behavioral language, both during and after this procedure. This is one of my cases of a patient who has had a recurrent tumor basically in the posterior STG area and underwent a resection there and basically developed a Wernicke's aphasia that lasted for at least 2 to 3 months. So one of the arguments from this, and this is provocative, is that the left STG is necessary and sufficient for speech perception, and the Heschel gyrus is actually not a critical input. So one of the models that we're going towards is a really different kind of way of thinking about how the auditory system works. The classic model is one that's really serial feed-forward based, but I think a lot more data is becoming more consistent with a parallel and distributed kind of processing that has different pathways. There's a lemniscal system that projects and is tonotopic to the primary auditory cortex, but there's a non-lemniscal that actually has different inputs from different aspects of the thalamus that project directly and bypass the primary auditory cortex that goes to the STG and middle STG. And these are the ones that are actually more important for speech processing. So this model that we're working towards is this idea that the human auditory cortex is not a serial feed-forward hierarchy, that there is a posterior to anterior activation with the posterior STG with primary-like fast latencies, and it's not tonotopic. There's a distributed map of speech features and coding, not a simple hierarchy of sound representation from A1 to STG. And a claim here that the medial and lateral auditory cortices are independent and parallel systems, which is very, very different than the visual system. The lateral STG is necessary and sufficient for speech perception, and the core auditory cortex is neither. So with that, just wanted to thank everyone for the attention, and thank you very much, George, again, for having me participate.
Thank you very much, Eddie. That was a great talk and great results. In the interest of time, we will move on, but please feel free to write your questions to Professor Chang. We have 2 more great talks for the next 40 minutes. The first one is from Anthony Dick, who is director of the professor of cognitive psychology at the International University of Miami, and he has done very extensive work on tractography and produced a number of excellent original and review articles. Anthony first got my attention when I read an amazing review article on brain, on consensus and controversy on the arcuate fasciculus. And he kindly visited us in London last year when we had the course in face-to-face. It was actually the last course before the COVID pandemic erupted. And I had the pleasure to meet him in person. He's got a great insight and a very general overview of multiple problems around tractography. So Anthony, thank you very much for joining us today. We look forward to your talk.
Thanks. You can hear me okay?
Yes.
Great. All right, I'm just going to jump right in. So George wanted me to talk about benefits and challenges of diffusion-weighted imaging for language mapping. And let's see, there we go. So I'm going to focus on two benefits that I think we can use diffusion-weighted imaging for, and then some specific challenges to each. So the first one, definition and verification of new pathways. And if we start with the sort of classic model of the language connectome that was known in the 20th century, we've started to really ramp up and modify that connectome mapping, mainly using a lot of different procedures. One includes the diffusion-weighted imaging procedures. So we've really revised this language model over the last probably decade and a half or so. A lot of these pathways were already identified, but one that has only been recently identified in the last decade is the frontal slant tract. I'm going to use this as an example fiber pathway. It's for a couple of reasons. One, I really am interested in the function of the fiber pathway, but another reason is it should be a relatively simple fiber pathway to try and track, and it turns out it's not. But if we start with the simplest fiber pathway and see what the challenges are, we can sort of extrapolate to more difficult fiber pathways. So what is the frontal slant tract? It sits right here within the broader language connectome, connecting generally inferior frontal gyrus to the medial frontal cortex. And when we study it, we usually identify two main regions of the inferior frontal gyrus, the opercular and the triangular region, connecting to pre-SMA and SMA of the medial superior frontal cortex. But there is argument that the pathways may extend beyond these specific regions, and some of the tractography might be more predominantly represented in certain regions as opposed to others. Functionally, it's associated with a number of language, speech, and executive functions. This is a listing of the languages functions it's associated with. We had a review of these things a couple of years ago. There's a really great review that just came out. The research on this tract is so rapid that even within 2 years, we need a new review. So I urge people to go check this one out. A couple of things, though. It's important to dissociate the frontal slant tract from neighboring fiber pathways. So if you're interested in exploring its function and you want to track it and relate it to behavior, or if you're interested in tracking it in preparation for surgery, you're going to want to dissociate it from the neighboring white matter, and that's a tricky thing to do. So this is just an example of a study trying to do this with tractography, dissociating the frontal slant projections in orange here from the frontostriatal projections shown in blue. And the reason you want to dissociate those is because the proposal is that they're associated with different functions. And can you do this with diffusion tractography? It's also important to define precise connectivity of the frontal slant tract. So this is much easier to do in animals. So this is the macaque from the original study of Petrudes and Pandia, which I think is one of the first studies to suggest you had this connectivity. Here's the injection in inferior frontal cortex of the macaque. And then you can see where all these axons project to, including the medial superior frontal and additional areas outside of that. And obviously, Dr. Schmamann mentioned this earlier. This is more like the gold standard because we can actually say these are the origins and terminations of these tracks. The question is, can we say the same thing when we're using tractography with diffusion-weighted imaging and even with blunt fiber dissection? So this is laws from 2008 using tractography and blunt fiber dissection. And this is before this pathway was even called the frontal slant tract. And you can see delineating the boundaries of it is rather tricky. even again in this sort of simple fiber pathway that we're calling the frontalis land tract. So you can see where are the origins and terminations using diffusion weighted imaging here. Now this is some years ago, and the methods have improved, but some of the problems remain. Here's some attempts to more robustly identify the origins and terminations using diffusion weighted imaging. So this is a study by Zmuda and colleagues using diffusion tensor imaging, showing that their proposal is that predominantly lateral frontal terminations are in pars opercularis here in green, with some in pars triangularis. And then in the medial cortex, studies trying to identify what are the terminations and boundaries of the tract here. This is a recent study suggesting that beyond the pre-SMA, the frontalis land tract may extend more anterior. And we would want to identify if this is the case, because if it's beyond pre-SMA medially and it goes more anterior into the frontal lobe here, we might suggest it be involved in other higher level cognitive functions beyond what we associate with pre-SMA and SMA, for example. And that's what they proposed, showing it may be involved in working memory. And the anterior boundary they've identified here is just right at the genu of the corpus callosum. But the same anterior boundary can be identified for pre-SMA. So anterior genu of the corpus callosum for pre-SMA. So we might want to know, look, is this really a projection to pre-SMA, or is it more anterior in superior frontal gyrus, because that would tell us something about the potential functional association of the fiber pathway. Can we do this with diffusion weighted imaging? It's not a simple answer. And then just one final example. Here's a paper proposing that the frontalis land tract has contralateral projections through that fiber pathway called the cross frontalis land tract. So one might ask, is it meaningful to call these frontalis land tract projections, or are they simply transcolossal fibers? And can we answer a question like that using diffusion weighted imaging? So to put it bluntly, should we trust the connectivity profiles from DWI? It depends on a lot of things. Three that I've identified here: acquisition parameters, post-processing decisions, and atlas definitions. And a lot of these Dr. Udiki talked about earlier, and I think this will reinforce some of these issues that we see. So for example, it depends on the acquisition parameters whether we should trust these connectivity profiles. So this is showing vendor differences with matched parameters, whether you run your diffusion scan on GE or Siemens. They have different settings. Even if you put the same number of B values, the same phase encoding direction, and so forth, you will get different distortion profiles. And then even the choice of the phase encoding direction can have pretty significant impact on the reliability of the diffusion scan and how it overlays on the actual anatomy that you're interested in looking at. And one of the interesting things about the choice of the phase encoding direction is that can affect the calculation of laterality in these diffusion tensor imaging tracks, which obviously is something that might be important for language mapping, for example. It depends on post-processing decisions. And I think this is one of the most important things that people don't really pay as much attention to. There's an assumption, I think, if you put it through the right post-processing software, it will work out. But some of these decisions have a pretty marked impact on what your tractography is going to look like later. So let me just parse this out. So here's the T2 weighted structural scan you're trying to make sure that your diffusion scan overlays on. And if you do diffusion weighted imaging, you're probably used to seeing these kinds of distortions. But if you don't, you may be surprised to see that the images, because of the type of acquisition, the images tend to be very sensitive to susceptibility artifact. And a lot of that depends on what phase encoding direction you use. So here's a right to left phase encoding direction, which pushes and smears the brain in this direction. Anterior, posterior, this is going to push the frontal lobe out. It will stretch and shear the brain. It's going to make alignment to the T2 difficult. And different corrections, which there's 3 of them shown here, will be differentially effective. And this affects tractography. This is just an example showing another scan where you have the distortion and then after correction, and does it overlay on the anatomy. It does affect the tractography. So here's 2 examples of where the ROI placement is identical. This is before distorted data, and you can see that this tractography tells you there's some part of this cingulum that goes below the corpus callosum here. And this corrected data shows that that part doesn't exist, at least in the diffusion scan. I think the even more stark example is an ROI trying to identify the corticospinal tract. And before distortion, you get a wildly different tractography profile than you do after distortion correction. And then finally, this was touched upon earlier, the choice of reconstruction algorithms significantly affects tractography. So this is an extremely busy slide. So let me just point out a couple of parts of it. So here's the ground truth that the reconstruction algorithm is trying to identify. And here's the diffusion tensor reconstruction of that ground truth. And you can see the diffusion tensor is not very good. And a number of other reconstruction algorithms from the buffet of reconstruction algorithms that are available get better, but they do show differences. So the one that you choose is going to affect what your tractography profile is going to look like. And Anastasia's talk earlier, I think, really showed this effectively by showing how the algorithm is going to choose which direction to go in can markedly affect what your track will look like. And then finally, the degree to which we trust these connectivity profiles is going to depend on atlas definitions. I think this is the most stark example of how what looked like pretty reliable tracks can actually be pretty unreliable. So this is a group, Schilling is the first author on both of these papers. It's Max Descotteau's lab at Sherbrooke Connectivity Lab. And you can see that when you give the same data set to 42 groups and you ask them to dissect 14 white matter bundles, and some of them are like the ones we're very familiar with, like the arcuate fasciculus, the corticospinal tract, you get wildly different outcomes, which is bad for replicability. But the good news is if you start with a very reliable and gold standard fiber pathway map, and you apply some of these constraints, you actually can get replicable tracking. But this is in the macaque, and we don't really have this gold standard in the human to the degree that we do in the macaque. And again, Anastasia showed this in some other studies earlier, suggesting that it's not all bad, but there are significant challenges that we need to try to address. Okay, so the take home on the first part, should we trust the connectivity profiles we get from DWI? I would say at this point, very dependent on user acquisition and processing decision. And that's where we need to do a lot of work in formulating some rules and boundaries. And I think that will happen with additional study, and it's already happening. Now, the second part, we want to use this tractography, and we want to use these diffusion weighted imaging scans to study disease, to plan for surgery, to study development of white matter. Like, for example, Dr. Kuhl presented a really interesting talk earlier looking at development of white matter. And the goal there is to measure some sort of direct property of the tissue we're interested in, like myelination or neurite density or something like that. But obviously we know from the diffusion weighted imaging scan that this measurement of tissue properties is very indirect. So just to give an example, we conducted a study in 4 to 7-year-old children. We're interested in the function of the frontal slant tract as it is associated with executive function and with speech. In this part of the study, we looked at the syllable repetition task to see if we could associate quantitative anisotropy of the frontal slant tract, which uses this more complicated crossing fiber sensitive reconstruction. We wanted to see if we can predict performance on the task with the quantitative anisotropy. So we did what were recommended practices for reconstruction. We did recommended practices for ROI to ROI tracking. And we controlled for a number of things that we think are important, especially for the diffusion metric in the whole brain and for movement, which is especially important when you're dealing with children. And we did find that there's an association between left frontal slant tract quantitative anisotropy and performance on the syllable repetition task. So what does that mean? Well, it just means that we've identified some sort of measurement of individual differences in diffusion properties of the fiber pathway that are associated with behavior. But we want to get more into what the individual differences might be measuring. So there are newer measurement models to try to study various aspects of the properties of the tissue, for example, neuronal morphology. So there are a number of multi-compartmental models that can be used to do this. It's just like the buffet of imaging methods to reconstruct for tractography. And here are 4 of them that are used most often, I think. And just to give an example of what these are doing, they're trying to separate different parts of the signal to get more at the morphology of the tissue that you're interested in. So the NODI model would separate intra-axonal space from extra-axonal space in the salmon color here, and then also try to separate signal from CSF as well. So it's a more complicated model attempting to divide up the signal in a particular way that would be useful. And these models tend to be sensitive to specific morphological properties. So for example, sensitivity to axon density. So the parent fiber density model seems to be most sensitive to what we find in postmortem histology. So this is from the Aboides paper looking at fiber density across the longitudinal axis of the corpus callosum. So here's the genu all the way through the body of the corpus callosum to isthmus and then to splenium. And as you can see, the fiber density of these sized fibers increases until the 3rd component of the body and then starts to drop off before popping up a little bit again along that longitudinal axis. And the best methods seem to be the apparent fiber density. And then we've looked at, in children, this restricted diffusion imaging metric that was developed by Frank Ye and his colleagues. Here in the Human Connectome Project sample showing the same sort of pattern. And then in a child sample, a separate child sample showing the pattern where you get the increase and then the decrease and then the pop-up. So these do seem to be sensitive to axonal density. And the NODI model has also been proposed to be sensitive to neurite density. So for example, you see increased NODI signal on the left here with more dense populations of neurons and dendrites and other processes off the neuron, as opposed to on the right. So we wanted to look to see if neurite density in the regions that are connected by the frontal slant tract are sensitive to individual differences on the task that we were looking at. So this is the syllable repetition task. And we find that for the right, there's a significant effect and then approaching significance on the left in the same direction, which is encouraging. And this was for the pre-SMA. So together, using these diffusion properties, we're trying to get at whether these morphologic differences can actually help us predict some performance on a behavioral outcome. Much the same as what Dr. Kuhl was trying to accomplish when she spoke about the younger children earlier. But the issue is we don't know, again, whether this measurement of tissue properties is actually giving you the kind of microstructural differences that you think it is. Again, the tissue property measurement is really indirect. So all we're measuring is water diffusion, and we're trying to separate the signal into different components. And the assumption is that these will be related to neuronal differences. And there's some evidence in postmortem histology to suggest that's the case, but a lot more work needs to be done to make sure we can actually relate these behavioral outcomes to actual differences that are in the tissue. And we need to try and figure out what that would mean. So just in summary, I do think diffusion weighted imaging is an important tool in the toolkit for addressing language organization of the brain, and it's important for improving precision mapping of the fiber pathways and for identifying individual and developmental differences in fiber and neural structure and relating these to behavior. But there are a significant number of limitations that I don't think are often appreciated. And I do believe we'll mitigate or eliminate some of these with improvements in methodology, with more reliable rules for how we would actually go and try to meet some sort of gold standard anatomical definition of these fiber pathways. But I don't think we're quite there yet. And I think the great thing about this set of talks today is showing that there's still some very significant questions outstanding in the field about what the connectivity profiles of some major fiber pathways are. For example, the inferior frontal occipital fasciculus and the arcuate fasciculus. The frontal slant is supposed to be an easy example, but even it has challenges. And there are some fiber pathways that we say they exist based on diffusion weighted imaging, but there is a lot of disagreement about whether that's the case. So for example, earlier today, there was discussion about whether the superior frontal occipital fasciculus is a real fiber pathway. And some of the issues I brought up today are the reasons why it's so challenging. So for people who work in this area, we are generally aware of these issues, but if you don't work in the area and don't use diffusion weighted imaging data, the decisions you make in the process of trying to get to the final result that you may relate to behavior or that you may use during neurosurgery or during neurosurgical planning, those decisions are extremely important and have profound implication to what the fiber tracks will actually look like. All right, let me just say thank you. I want to give the last speaker an opportunity to talk. So thanks so much.
Thank you very much, Anthony. That was a great overview. Talking about the Aslan tract, what is the view of the posterior Aslan tract, which was referenced earlier on today?
Well, that is, I think of it as being essentially similar to the posterior part of the arcuate fasciculus, but I was interested to hear that view is not really shared. And I think that I don't necessarily have a problem with this. I think one of the great things about this evolving literature is that as we start applying and combining different methodologies, we're going to come to some different definitions. So I would say it's this, basically they are the same thing, but I'd be willing to hear otherwise. I know there's some thinking that the posterior part of the arcuate fasciculus, as defined by Catani, is not the same thing as this parietal aslant tract.
Thank you. I hope we'll have more time to discuss this tomorrow. So moving on to the last talk of the day, I'm very pleased to welcome Firouz Tarapor, who is a professor of neurosurgery at UCSF. And I had the pleasure to meet him in person a few years ago when I visited the UCSF. Firouz has worked with basic scientists and has introduced very important concepts on navigated transcranial magnetic stimulation. And at least within the neurosurgical field, I cannot think of anyone more experienced to talk about language assessment with TMS. So Firouz, thank you so much for joining us today.
Thank you, George. Thank you to all the other panelists, and thank you to our audience who has displayed a real interest in this topic today. And I know that for many of you, it's either very late or very early. So my thanks goes out to everyone and to the whole team that put this on together. I know it was not an easy thing to do. So thanks to all of you. Well, I'm going to switch gears just a little bit and talk about language mapping with navigated TMS. For those of you that may not be familiar with navigated TMS, I'm going to describe exactly what it is and then talk about the state of the technology at present day and review the literature that's relevant. So as I said, we'll discuss some of the methodology and then talk about the utility of it. So transcranial magnetic stimulation uses the principle of induction. There are a number of ways to do it, but the most common is with a figure of 8 shaped copper wound coil with a rapid electrical discharge that goes through it. And the flux in the electrical field results in a rapid magnetic field as well, which is then able to induce an electrical current and a depolarization in the underlying neural tissue. So for the purposes of most language mapping, we're trying to induce what's called the temporary lesion effect in the underlying tissue. And you know, this is basically where it all started. About 30 years ago, Dr. Pasquale, who was one of the leaders in TMS, figured out that it was possible to induce speech arrest and counting errors with a rapid pulse sequence of transcranial magnetic stimulation. It was recognized and reported on, but not really applied in the neurosurgical field or in the brain mapping field for almost 20 years, until there was the advance of basically merging a transcranial magnetic stimulator with a frameless stereotactic navigational system. And for those of you that aren't familiar with that, a frameless stereotactic navigational system is something that we commonly use in the operating room. It is basically a way of identifying a patient's anatomy and localizing a patient's anatomy and referencing an MRI scan, which is displayed on a monitor. So we can point a pen to particular parts of the patient's anatomy, and the MRI on the screen will rapidly reflect exactly the portion of the patient's anatomy that we're pointing to with the pen. So merging a transcranial magnetic stimulator with this technology is understandably makes it much more powerful, because we can induce the lesion and know exactly where the lesion was induced. Thereby, we can do a non-invasive language map or motor map, whatever the goal may be. So this is the resulting type of map. This was published in that original paper I showed. You can see here the left side of the patient's brain. The orange dots here and here in a cloud over and around the sylvian region reflect each stimulus with the transcranial magnetic stimulator. And the red dots here with the arrows pointing to them are reflective of those sites that resulted in a speech arrest or a counting error. So move forward again by about 5 years, and now we start to go past just doing speech arrest and counting, and we start doing some of the more sophisticated mapping techniques. In this case, we're talking about object naming, which is probably one of the more commonly used tasks for intraoperative language mapping. So in this article and in a few other articles here, for example, we found a good correlation between intraoperative direct cortical stimulation and preoperative navigated transcranial magnetic stimulation generated language maps. And we started to see some trends here. We started to see that the technique was highly sensitive and very good at identifying all positive language sites, but it was not terribly specific, meaning that there were a number of language sites which seemed on transcranial magnetic stimulation to be important, but then in the operating room with direct current stimulation, we did not see correlation. Some of the issues that we worked out here were with regard to the pulse onset and the stringency of the language error determination, whether there was a speech and language specialist doing assessments or not, whether there were multiple people doing independent assessments or not. But still, even with the most stringent of determination algorithms, it turned out that the best we could push specificity was about 60%. So at the same time, navigated transcranial magnetic stimulation based language maps have been looked at for other purposes. And in this case, it seems that they are particularly useful in identifying seed regions for performing subsequent fiber tractography. So in this paper, Dr. Solman and Krieg in Munich demonstrated the feasibility of navigated transcranial magnetic stimulation-based fiber tracking for subcortical-based language pathways. And essentially here, what we're showing, what they were able to show is the ability to identify 9 language-related subcortical tracts. You can see in this 3-dimensional image, the orange volume represents the tumor. The tracks are obviously in multicolors, and the language sites are represented in purple dots. So those positive language sites were used to seed fiber tracking algorithm. Subsequent study in 2018, this is a particularly important study by Dr. Raffa in Italy, which took a slightly different approach, but also used postoperative aphasia, in this case as determined by the Western aphasia battery, to determine the accuracy of preoperative navigated transcranial magnetic stimulation and fiber tractography in brain tumor patients. These were all patients who were unable to go intraoperative direct cranial stimulation for a variety of reasons. So they demonstrated excellent correlation between the preoperative TMS maps and development of the postoperative deficit. In this case, they had 100% sensitivity and a specificity, again, that was about 57%. So here you can see in the top right image, I apologize, the spots are a little bit small, but you can see that the TMS language mapping sites showed some spots inside the lesion. And as a result, this lesion was classified as most likely a truly eloquent language lesion. Here's an example of a case with a non-eloquent lesion. You can see the TMS spots are outside the tumor volume, which in this case is left frontal. And in this case, the tumor was classified as non-eloquent. We've also looked at navigated transcranial magnetic stimulation-based fiber tractography for performing prognostication in determination of preoperative risk stratification of patients with tumors in language eloquent regions. So in this study from 2020, the lesion to tract distances on fiber tractography were measured. And it was found that they were predictive of postoperative language deficits in patients with left-sided hemispheric language dominance. So in this case here, the lesion to tract distance of less than 8 millimeters for the arcuate fasciculus and 11 millimeters for other language-related tracts were found to be good cutoffs for surgically induced permanent aphasias. So I'm going to show you a few videos here of exactly what it looks like when we do transcranial magnetic stimulation with language mapping. So here you see the patient. You can see the reference arc, which is this device attached to the head. She is looking at a monitor, which is displaying this picture here. The picture is of a bone. And on the left is going to be a control, and on the right will be the trial stimulation. I'll play that again. Bone. So she says the word bone. Bone. And here you can see she's unable to produce sound during the train of pulses. Bone. 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 Here's another example. Bench. So she says the word bench. Bench. And again, a long delay until the train is complete. Guitar. She says the word guitar. Violin. And here she says violin. And again, that response is after the train has completed. So it's really, in this case, a combination of speech arrest and a semantic paraphasia. Here's another patient. You'll see on the left a control and on the right the stimulation. And this patient was bilingual in both English and Tagalog. So I'm going to show a couple of examples in each.
Ball.
So here he says the word ball.
Ball.
And here he says it correctly.
Coin.
He says coin. No response.
Ball pen.
So he says the word ball pen, which is the Tagalog word for pen.
Oh, pencil.
And here he actually says the word pencil, which is obviously the English word for pencil.
Grapes.
The response is grapes.
Juice.
And it's hard to hear there, but he says the word juice. So this is an example language map. This is what the prior patient's language map looked like. You can see the area of the lesion here in the temporal region and the entirety of the cloud of TMS response points surrounding it. And this is what the intraoperative map looked like. Despite those numerous positive language points, the only intraoperative speech arrest site was actually at this location, which is where the number 1 is placed. So these efforts have resulted in a protocol that was put together a couple of years ago for best standards and practices in motor and language mapping with navigated TMS in both patients and healthy volunteers. This was a group effort by a number of folks who have been working on this for years and decades. I invite anybody who's interested in incorporating this technique into their practice or into their research practice to take a look at this paper, because it really does detail the best practices for conducting this sort of mapping. So in conclusion, I would say that NTMS-based language maps offer really valuable insight into the functional relationship between eloquent brain and a lesion. They are particularly useful when applied in conjunction with fiber tractography and might even be helpful for prognosis after surgery. They are very useful in patients unable to undergo awake surgery. And just keep in mind that the technique has a very high sensitivity and negative predictive value, but negative mapping is feasible, and positive mapping, you have to be aware of false positives because they happen a lot with current practices. So that will bring me to the final slide. It just thanks to the whole group and to all of the audience for sticking with it to the end here, and I'll turn it over to George. Thank you very much.
Thank you very much, Firoz. That was an excellent review of navigated TMS. I have tried this myself for about a year, and we also tried language like yourself and some vision. Some of the difficulties my team had was how to handle the magnet, which looks quite bulky. I think it plans to make this slightly smaller and therefore